Good morning, everyone. Welcome to day 3 of the AMALathon. And this specifically is the AQA session and the question answer session from the AQA session. So if anyone has any questions, just drop the answer, drop the question into the live chat, and I'll do my best to answer it. Now, there was a request yesterday to go through some 6-mark questions, so that's what we're going to start with. And like I was saying to you, and I've reiterated multiple times, that AMAL method really makes a big difference. And this is one example that I'll highlight that. Because the first thing we've got to do is activate our brain. We've got to understand everything. And the best way to understand, and this question is an example where you have to have understood. After that, past paper practice ties everything together. So my advice to everyone, and I'll repeat this when we've got more students on afterwards, is that once you reach a point, so if number one, you've got to understand everything. So that's what we're doing during the AMALathon. So during the AMALathon, we're going to understand everything in the best detail we possibly can. And if anyone doesn't understand anything, all they've got to do is ask. And I can repeat things, go over things again. We've got time from quarter to 10 till 10 o'clock. There's also actually time from 9 a.m. So if anyone wants to repeat any topics, let me know and I'll come on from 9 and I'll repeat those topics and I'll go over them again. So this time is yours in whatever way you want to use it. So if people want to go through even more questions, I can come on even earlier. Now, this question that's in front of us now, this is a 6-mark question. And let's quickly go through it. So it says particles can move into and out of cells by different processes. So we'll come back to that. And it says figure 2 shows different particles inside and outside a root hair cell. Explain the processes by which the different particles would enter the root hair cell. So what we need to look at here is the keywords. So first thing, if anyone wants to answer the question and type it into the live chat, and I can read through the answer, that's not a problem. But here, our keyword is we're trying to explain. So that means we've got to give a scientific explanation as to why something's happening. Now we know things enter. So now we're looking at the processes by which different particles will enter the root hair cell. So we have got diffusion as one of the processes. We've got osmosis. And we know active transport. Now this is where some memorization has to come into place. We have to be able to write down what diffusion is, what osmosis is, and what active transport is. So we know diffusion is the movement from high concentration to low concentration, and it is a passive process. I'm just going to put a P in brackets for passive. Osmosis is from the movement of water from dilute to concentrated. Remember what we said yesterday, when it moves from dilute to concentrated. And if you ever forget what the direction is, it's Washington, D.C. Dilute to concentrated. And that is also a passive process. And osmosis occurs through a partially permeable membrane. So I'll just draw a dashed line there to show partially permeable membrane. Or better, it would have been if I did it here. And then we've got active transport. Active transport is the movement of particles from a low concentration to a high concentration, and this requires energy. This requires mitochondria, where respiration occurs, and that releases energy. Now we need to, because it's an explanation question, we're going to apply what we know to our understanding. We've got to, so what molecule there is diffusing? So actually the easiest one would be to start with osmosis. We know that only occurs with water molecules. So when we're talking about osmosis, we would specifically say the water molecules go from a region of where it's dilute. So that means the soil outside is dilute. And we know that because if you count these white circles, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. But on the inside, there's 1, 2, 3, 4, 5, and 6. That means we've got a higher concentration of water on the outside than the inside. So which way is the water going to move? It's going to move inwards due to osmosis. And osmosis is the movement of water particles from a region that is dilute to concentrated. Now let's look at the phosphate and the magnesium. Now, if you look at phosphate, which is the greyish, this one here. So I've still got it on eraser. If you look at phosphate just there, we only have 2 phosphates on the inside, but we've got lots and lots of phosphates on the outside. So we have a higher concentration. Yeah, well done, Dean. What you've written in the live chat is absolutely correct. Phosphate, we've got more phosphate ions on the outside, the inside. So that means our phosphate ions are going to enter through diffusion. So they're going from a high concentration to low concentration, and it's a passive process. Now with magnesium, there's lots of magnesium on the inside. That's these ones with a little plus sign in the middle. There's very few on the outside. So the only way we're going to get the magnesium inside is through active transport, moving it from a low concentration to a high concentration. And that requires energy. So that requires mitochondria, where respiration occurs, and that releases energy. Now if you compare what we've just discussed with the mark scheme, you're going to see that is 6 marks out of 6. But the whole point here is many students will have looked at this diagram and they would not have known which one does diffusion, which one does osmosis, which one does active transport. So this is where understanding comes into play. And then also knowing what diffusion, osmosis, active transport, that's where memorization comes in. And this past paper practice shows you how those things tie together, the understanding and the memorization. That's why past paper practice is really important. So my advice to everyone, and I'm going to keep reiterating this across the 6 days, across the whole AMALathon, is number 1, understand. Number 2, memorize key facts. Number 3, lots and lots of past paper practice. So as soon as the AMALathon is over and your understanding phase is finished, my advice for the rest of these holidays is to try and do 1 past paper a day, or at least a third of a past paper a day. We've got 4 weeks left, so you want to do your best. You want to have gone through all 7 or 8 past papers which are available before your exam. Okay, so let's look at the indicative content. Water is absorbed by osmosis. So tick. Water is a passive process. Water in soil is at a higher concentration. Yep. Water moves down the concentration gradient. Now we didn't mention that. It goes through a partially permeable membrane. Phosphate ions are absorbed by diffusion. Diffusion is a passive process. Phosphate ions are in higher concentration in the soil than inside the cells. Magnesium ions are absorbed by active transport. We mentioned that. Magnesium ions are lower in the concentration in the soil. Magnesium ions move from an area of low concentration to an area of high concentration inside the cells. Magnesium ions move up the concentration gradient. Now we didn't mention this. So we didn't mention the concentration gradient bits. And the process requires energy from respiration. We even mentioned mitochondria. So automatically now, we've added all that content into our answer. So we've automatically guaranteed ourselves 6 marks, depending on the quality and the way we've written it as well. Right, now this question is a 5-mark question. And when this question was originally asked, it's a question that really that stumped a lot of students. Because like I said, or like I've said in previous sessions, students who are likely to get grade 9s or who are very good at science don't mention the obvious. Now, plants need nitrate ions in order to make proteins. A plant is growing in soil flooded with water. Explain. So we need to explain, to bring our scientific explanation, why the plant cannot absorb enough nitrates. So, first things first, if we're struggling to answer this question, at least write down initially what we know. And what is it that we know? We know that the only way nitrates enter the plant is through active transport, through the root hair cells. So we know we've got our root hair cells, and we've got more nitrates on the inside than on the outside. So what process do we require? So we've mentioned root hair cell, and it is active transport. Active transport is when the nitrates will move from a low concentration to a higher concentration, meaning they go against the concentration gradient. So it's like moving something uphill. So that's that diagram I've drawn just to represent that is going against the concentration gradient. So we've mentioned active transport, we've mentioned root hair cell, active transport, low concentration to high concentration. We mentioned that it's going up the gradient, so it's going against the gradient, and it requires energy from respiration. So respiration releasing energy. Now let's break down respiration for a second. Respiration is the use of glucose. Now, does the plant have glucose? Yes, because it photosynthesizes. But where does the oxygen come from? Now, for the root hair cells, some of the oxygen will be through the soil. And the soil does have air inside it still. Now, if it's waterlogged, the pitch, that means there'll be less oxygen. So that means we have an issue. We have less oxygen because the soil is flooded with water. Now, let's just assume we didn't know that the soil being flooded with water equals less oxygen. Not a problem. We still mentioned everything else. So we mentioned the fact that for those nitrates to enter, we have to have active transport, low concentration to high concentration. We need respiration, we need energy from that, but respiration requires glucose and oxygen. If we mentioned everything else, if we don't mention this one point because we missed it, we still should get most marks. So let's have a quick look at the mark scheme. Ions are absorbed by active transport. So it's a movement of ions against the concentration gradient. Active transport requires energy from respiration. Respiration requires oxygen. Now, this is the bit that many people missed. There's no or little oxygen or air in the waterlogged soil. Now, we mentioned this, however, a lot of students didn't. But what that still does is it still gets you the original 4 marks. And what do we get the 4 marks for? For writing down what we already knew. We know that nitrates are absorbed through active transport, and then just explaining what active transport is. So the first thing you do in a question, which might seem difficult and worded in an awkward way, like this one is, or worded in a way that the question doesn't fully make sense straight away, is we write down what we know. In this case, we know that nitrate ions only enter due to active transport. Now, then it says, explain why the plant cannot absorb enough nitrates. So what would stop a plant from absorbing enough nitrates? It would be not doing enough active transport. So that means something's stopping this process from happening. In this case, it's not having enough oxygen. So again, look, this shows that, number one, to answer this question, we had to understand. there had to be an understanding of how nitrates enter the plant. If you didn't have understanding, you can't understand the question. So that's why the activate phase is really important. Then you need from beforehand to have already memorized what active transport is. And the third part, by doing this past paper question, it helps us to understand what the question actually means and what the examiner wants from the question. So these three phases are always important. Like I said, that's what we call AMAL: activate, memorize, apply. And if you bring all three things together, then you've learnt. So just quick answer to a question, and it's a really good question. That there are lots and lots of mitochondria, but if there isn't sufficient oxygen, the mitochondria won't be able to do the respiration. Okay, for those that have just joined us, welcome to day 3 of the AMALathon. Thank you for those that have been joining from day 1. Thank you for those that have been watching the recordings. I really appreciate everyone's support. We were really surprised by the uptake and how many people have been watching our videos. Our first one, our day 1 video has already gone over 1,000 views, and our second day video is close to 700 now. And I really appreciate those people that have been sharing, liking, and the ones that have subscribed as well. If you haven't subscribed, please do subscribe. Okay, what we're going to do today is we are going to finish off topic 2. So now we're on schedule. So we finished cell biology, and we didn't start set organization yesterday, but we're going to do our best to finish it today. In fact, we are going to finish it today, and then we'll start infection response tomorrow. So we're a little bit off schedule, but not massively. So, cells. Cells are the basic structural and functional units of all living organisms. They carry out the essential life processes and can exist as single-celled organisms or as multicellular organisms with specialized functions. Now, if you get a group of cells working together, then we have tissues. So this shows muscle tissues. Each individual cell, you've got lots and lots of these individual cells working together to make a tissue. Lots of different types of tissues working together to perform a specific function we call an organ. So you'll have lots of different tissues within the lungs, allowing the lung to do different, allowing the lungs to fulfill its function. And if you have lots of organs working together, fulfilling a vital function within the body, for example, the digestive system. So if you have lots of organs working together, we call it an organ system. So for example, the digestive system, which processes food, the respiratory system, which enables us to breathe. So an organism, if you now bring all these organ systems together, what you have is an organism. And an organism is a living being that can function independently. It may be single-celled, like bacteria, or multicellular, like humans, animals and plants, with complex organ systems working together to sustain life. So a group of cells work together. So cells are the building blocks of life. A group of cells work together are tissues. A group of tissues work together are organs. A group of organs working together are organ systems. And a group of organ systems work together, it makes up an organism. Now, the role of the digestive system. Number 1, it breaks down food. The digestive system breaks down large insoluble food molecules into smaller soluble molecules. Now, if you consider the whole, the purpose of the digestive system is to take a large piece of food. And this is, I mean no offense to anyone who's a vegetarian here, but if you have a piece of chicken, that piece of chicken has got nutrients inside it that need to reach our cells. Now, the only way the nutrients in that piece of chicken can reach our cells is through the blood. Now that piece of chicken is not going to fit into our blood because it is it's a large insoluble food molecule. So that means it has to go through a whole process, as in we chew it up into small pieces, it enters our stomach and it's churned, it enters our small intestine where enzymes break it down further. And we have enzymes in the mouth that break it down, we have enzymes in the stomach, but the main part is within the small intestine. And that process breaks it down into smaller soluble substances, so those smaller soluble substances can enter our blood. Then our blood can take it around the body and take it to the cells that require those nutrients. So that's what's explained here. Nutrients are absorbed into the blood through diffusion in the small intestine, but first that food has to be broken down and made smaller and soluble, and then it can be utilized by the body. The body uses these nutrients to build new carbohydrates, lipids, and proteins. So the whole point is, if you've got a large protein, they are broken down with protease enzyme into individual amino acids. Then once they enter the cell will join them back in different form to make a different protein. And that's protein synthesis, which occurs in our ribosomes. Energy production. So glucose from digestion is used in respiration to generate energy. So when we break down carbohydrates, it's broken down into glucose. When we break down proteins, it's broken down to amino acids. Each thing that's broken down, our body uses for different functions. Specifically, glucose is used for respiration. So nutrients like carbohydrates, they provide energy for bodily functions, and we find them in bread, rice, pasta, potatoes, and fruits. Proteins we need for growth and repair and maintenance of tissues. We find those in meat, fish, eggs, beans, lentils. This is not an extensive list. There's much bigger lists. Lipids and fats, they are stores of energy. They insulate the body and protect the organs. If anything, when we say it's a store of energy, we go into our lipid store of energy when our carbohydrates run out. So we initially use carbohydrates and then we start using our fat store. And fats provide twice as much energy as carbohydrates, but they take longer to release that energy. What are enzymes and how do they work? So enzymes are biological catalysts. So we're going to study this in chemistry as well. A catalyst speeds up a reaction without being part of the reaction. And that's exactly what an enzyme does. It helps break down proteins and it helps break down carbohydrates, lipids, which are fats. And it's essential for process like digestion, respiration, and metabolism. So how do enzymes actually work? Each enzyme is specific to its substrate. So there you can see here we have the enzyme, we have the substrate, and here where I'm just drawing my blue line, that is the active site. Once the substrate attaches itself to the active site, weak intermolecular bonds are formed between the two, and that makes it easier to break that substrate down into the products. That means that one enzyme will only specifically work with one substrate. So protease will only work with proteins. Carbohydrates will only work with carbohydrates. Lipase will only work with lipids. And the enzymes are used in things like digestion, respiration, and DNA replication. So this is what I've just explained. We've got an enzyme, the substrate attaches to the active site. Then you have an enzyme substrate complex. The products are made, they're released. So now we've got our products. And the enzyme is left unchanged, which means another substrate can now attach and that whole process can start again. And we call this the lock and key model. But why do we call it lock and key model? Because one substrate is specific to one enzyme. Let me rephrase that. One enzyme is specific to one type of substrate. Just like our key for our front door only works for our front door. It's not going to work for one of the doors at school. It's not going to open up the neighbor's door. So that's why we call it a lock and key model, because you have a specific substrate which makes the enzyme substrate complex with a specific enzyme. So explain the reason why no product will be formed. So this is a past paper question, and I know the answers just come on the screen, but we're just going to quickly go through it. And the question was really simple. Why is there no product formed here? And simply because this substrate is not specific to this enzyme. So substrate A is specific to the enzyme, so it will make us enzyme substrate complex, but substrate B will not. Now, what is a denatured enzyme? A denatured enzyme is an enzyme that has lost its shape, making it non-functional. The enzyme cannot regain its shape. So here is the original enzyme, and because of an increase in temperature, so enzymes don't work well beyond a certain temperature or beyond a certain pH. They have optimum temperatures and they have optimum pHs. So here, because of the increase in temperature or optimum or increase in pH, the active site has changed shape. Essentially what's happened is the increase in temperature or pH has caused some of the bonds to break. And when those bonds are reformed, they've reformed in a different active site pattern, meaning that substrate will no longer fit. So there's therefore no longer a reaction. Now, explain how boiling the enzyme stops the protein being digested. Now, if you look at this answer, these are 3 points that should be memorized and always written down whenever the word denature comes up. Because once an enzyme is denatured, what happens? The active site changes shape. If the active site changes shape, that means the substrate will not fit into the active site. So it's these 3 steps over and over again that need to be remembered. Now, in questions, you might be asked to describe, or you might be asked to explain the link between the impact of increasing temperature, pH, or substrate concentration on enzyme activity. So there's a difference when you answer a description question and a difference when you answer an explanation question. When you describe something, you say what you see. In other words, what do we see here? As the temperature increases, the enzyme activity increases. There's no scientific explanation there, we're just saying what we see. This point here is when the enzyme activity is at a maximum, and that's what we call optimum temperature. So enzyme activity is the maximum at optimum temperature. Again, we're just saying what we see, we're not explaining why that makes a difference. Now, as the temperature continues to increase, what happens to the enzyme activity? It decreases. As the temperature continues to increase, the rate of enzyme activity decreases. Now, the reason for that is the enzyme denatures, but it's a described question. So there's no need to write down the why. You just write down what you can see. And this is, again, one of those things that students who are intelligent enough to get grade 9s always get confused, mixed up on, because they think it can't be that simple. It can't be that I just say it goes up and it goes down. So they start to explain. And usually these are the kind of students, when a teacher's marking their papers, they've got answers on the side. On the other side, it says, please see the back. And because it wasn't enough just to describe it. So you have to, when you go through past paper practice, start to see the difference between describe and explain. And when you do more and more past paper practice, you'll start to see a description question is not as complicated as you may have thought. It is just write down what you can see. And an explanation is where you need some memorization so you can actually explain the science. So now let's look at the explanation. So as the temperature increases, so in the explanation you should still start with the description. So when you're writing an explanation, still start with the description. So this part here is the description, this part here is the description, and this part here is the description. So as the temperature increases, the rate of enzyme activity increases. Why? Because particles are moving faster and so collisions are more likely. And the collisions are more likely between the enzyme and the substrate. As the temperature continues to increase, the rate of enzyme activity decreases. Why? Because the high temperature has caused a change in the shape of the active site. So the high temperature has caused a change in the shape of the active site, meaning it's caused it to denature. Now if it's denatured, the active site has changed shape. If the active site has changed shape, the substrate will no longer fit in the active site. If it's not fitting in the active site, then there's no reaction. Okay, so now let's describe the impact pH has. As the pH increases, the rate of enzyme activity increases. So pH is increased, enzyme activity is increased. What we've got here at the top there is when the enzyme has enzyme's optimum pH. Then as the temperature continues to increase, the rate of enzyme activity decreases. So again, what we've done here is we've just written down what we can see. We've compared our x-axis to our y-axis. Now if we're going to explain this, so we've got our description here, here, and as the pH continues to increase, the rate of enzyme activity decreases, and here. But why does the pH, as the pH continues to increase, why does the rate of enzyme activity decrease? Why? Because the pH has caused a change in the shape of the active site, meaning it has denatured. Now if it's denatured, what's changed shape? The active site. If the active site's changed shape, the substrate can no longer fit. If the substrate can no longer fit, then there's no rate of reaction, and the rate of reaction starts to decrease. Okay, so these are common enzymes that are linked to digestion that you need to know. Amylase, which is found in the salivary glands and the pancreas. What does it do? It breaks down starch into glucose. And the best pH for amylase is 7. Then we have protease, which is made in the stomach. And if it's made in the stomach, it's made in the pancreas and the small intestine. Now, it turns proteins into amino acids and it works best in acidic conditions. Then we've got lipase. It's made by the pancreas and the small intestine. And it breaks lipids or fats into fatty acids and glycerol, and it works best at pH 8, which is alkaline. Now if we look at these, protease works best, it's made in the stomach, and because of the pH that it works best in, it works best in the stomach. The small intestine is slightly acidic, so amylase works best in the small intestine, and the lipase works best in the small intestine as well. Now, why are enzymes important? Because they break down large molecules into small molecules that can dissolve into the blood, be carried to our cells, and then our cells can use them to make new molecules. Now, what enzymes do is they lower the activation energy because they're catalysts. So everything a catalyst does, our enzymes do. They lower the activation energy, meaning less energy is needed for the reaction to occur between the enzyme and the substrate. The less energy is needed to break down the substrate, They speed up reactions without becoming part of the reaction. They're specific, which means each enzyme works on a specific substrate, ensuring the correct reaction takes place. Enzymes help control and coordinate metabolic pathways. and they enable key functions like digestion, DNA replication, and energy production. Now, those doing separate science, DNA replication, when we cover that topic, you'll see how enzymes are involved. So, how enzymes breaking down starchy foods? How do enzymes break down starchy foods in the mouth? So initially the teeth chew up the food. This is mechanical digestion. It breaks the food into smaller pieces. Then after that, the salivary glands release amylase. And amylase is an enzyme that starts breaking down starch into sugars. And enzyme producing glands. Enzyme is produced by amylase salivary glands, by the pancreas, small intestine, and lipase is produced by the pancreas and the small intestine. So that question has been written slightly incorrectly, but you need to memorize where the enzymes are actually made, which glands produce them. Now, describe how protein and starch are actually digested. So protein is digested by protease, and it converts it into amino acids. Starch is broken down by amylase, and it breaks down the starch into glucose or sugars. And lipids are broken down by lipase, and the lipid is broken down to fatty acids and glycerol. And as we've already covered this bit, it's important to know where each one is made. You'll note that all three are made in the pancreas, and all three are made in the small intestine. It's just the protein is also made in the stomach, and the starch is also made in the salivary glands. So if you remember small intestine and pancreas, you've got all three covered. So how are fats digested in the body? So initially, mechanical digestion happens first in the mouth and stomach that breaks the fat into smaller pieces. The enzyme lipase is released by the pancreas and small intestine. Lipase breaks down fats into fatty acids and glycerol. The digestion takes place in the small intestine. Now, prior to that happening in the small intestine, the liver produces bile. Now, bile is stored in the gallbladder and it's released into the small intestine. Bile neutralizes stomach acid, because remember, carbohydrates lipase works best at pH 8. So bile neutralizes the stomach acid, creating the right pH for lipase to work, and also emulsifies fats. So what it's essentially doing is it's taking large bits of fat and it's breaking them down, so it increases their surface area. If you increase the surface area of something, you increase the rate of reaction. Okay, I'm just going to remove that line because I've not drawn it very straight. Okay, why are enzymes important? So this is what we've already covered, but we'll just quickly reiterate it. Lipase breaks down lipids into fatty acids and glycerol. But there's a two-way process. It can be used in the other direction as well. It can be used to make lipids. Now, amylase. Now, DNA polymerase is something that we'll cover in more detail when we cover protein synthesis. Okay, so this is a quick recap of what we've already covered. Salivary glands produce amylase. Stomach secretes protease, and the pancreas makes the majority of these digestive enzymes. So it produces and releases amylase, which is a carbohydrate, protease, and lipase. Now, from a labeling perspective, that usually comes up in foundation, but in the higher paper as well, you can be asked to write down where certain enzymes are produced. So the role of bile. We have covered this in a little bit of detail but we'll go through it one more time. Now, bile is made by the liver and it's stored in the gallbladder. When it's released, it's released into the small intestine, and bile is alkaline, so it neutralizes the stomach acid. And most of our enzymes work best at between a pH 7 and slightly alkaline environment. And the final thing, which is really important, it emulsifies fats. So it breaks large fat droplets into smaller ones, increasing the surface area for enzyme action. Okay, this is the question that came up before, but that slide should have been deleted. So describe 2 ways the mouth can break down starchy foods. We chew up the food physically and break into small pieces, and then amylase enzymes begin breaking down starch into simpler sugars. And explain the role of stomach in digestion. The stomach secretes protease, and protease breaks down proteins. Now, hydrochloric acid creates an acidic environment for enzyme activity. Now this, if you look at this question, it says explain. This one says describe. So it's important to start to understand the difference between what's needed for description and what's needed for explanation. So in description, we're writing down what actually happens in the mouth. And what happens in the mouth, we chew the food physically into smaller pieces, and our salivary gland releases amylase that breaks down starch into simpler sugars. It doesn't say how the process occurs or why it occurs, it just says what's happening. Now here we're saying explain the role of the stomach. So the stomach secretes protease, that's what it does, so that's a description, but why does it do it? The why is because the enzymes break down proteins. Hydrochloric acid creates an acidic environment. Why? Because for enzyme activity. Now that could have been elaborated further to say that protease works best in an acidic environment. So when you're explaining, you've got to answer the why. You still put the description, but then you put a why straight after it. How does bile help fat digestion? Bile emulsifies fats. Now, when we say how, that still comes under the category of explain. So what does it do? This is description. It emulsifies fats. It breaks them into smaller droplets. Why? Because that increases the rate of reaction. It's better enzyme action. Now again, we could have elaborated here with the why part and written down the fact that by emulsifying the fats, we increase the surface area, which increases the rate of reaction. Oh, actually, I've spoke too soon. Now, bile also neutralizes stomach acid, and that's what it does. And the why, to provide optimal pH for digestion, because lipase works best in a pH of 8. Why is the small intestine important for digestion? It absorbs nutrients into the bloodstream through the villi. That's not something we've covered in enough detail yet. And it secretes enzymes to complete the breakdown of food molecules. Okay, before we can test the substance, so this is one of the required practicals. Before we can test the substance, we need it to be in solution first. So when we're testing something like milk and juice, they're already in a solution state. So they can be tested as they are. However, something like bread isn't. So what you've got to do with bread is you've got to grind it into a paste with a pestle and mortar, add water to it and stir. And that turns it into a solution. So now we can test for starch. Now if you add iodine to a food sample, now with all of these, the color just stays the same. If there's nothing there, then the color will stay the same. So with iodine, it tests for starch. If it turns blue-black, then we know we've got starch. And just in case this kind of question comes up in your GCSE paper, iodine is harmful. So safety goggles should be worn, and you should avoid contact with skin. And when we test for glucose, we use Benedict's reagent. If there's no glucose, it stays blue. But if there is glucose, it will turn red, green, yellow, orange, brown. Now, red means it's got lots of glucose. As the color becomes lighter, it means it's got less and less glucose. I forgot to say, you have to heat Benedict's reagent for it to work. Protein, you add biuret solution or burette solution or burette reagent to the food sample. Now if there's no protein, it stays this bluish color. Otherwise, if there is protein, it turns to like a purple mauve kind of color. Now burette is harmful, just like iodine, so you need to wear safety goggles. You need to wear that with Benedict's reagent as well, and you need to avoid contact with your skin. Testing for lipids. So you can add ethanol and distilled water to the food sample and shake. Now this only works with a solution. Now if there's no color change, there's no lipid. But if it turns to a cloudy white color, then it means there's a lipid. Now if you've got something that won't turn into a liquid, so adding ethanol and distilled water doesn't allow it to mix, it doesn't mix in together, then instead you can just rub that piece of food on paper. If it leaves a greasy mark, it means it contains fat. Now, these kind of questions are memorization type questions. So you need to know the name of the reagent, you need to know the positive outcome and negative outcome. How can we investigate, and this is another core practical, how can we investigate the effect of pH on enzyme activity? Now we need to understand this in a bit more detail before we continue. So it says here, add a few drops of iodine solution to each dimple in the spotting tile. So this whole thing's called a spotting tile, and each one of these is like a little groove inside that spotting tile is called a dimple. Now in this experiment, initially what you get is some starch solution, so amylase solution, pH buffer, and you put it into a water bath at a set temperature. And that's because we're using amylase. Amylase is an enzyme, so we want everything to be at the same temperature. If our starch solution is too hot, it could denature the amylase. If our pH buffer solution becomes too warm, it can denature the amylase. So everything starts at one temperature, and that's also one of the variables that we keep the same. So our constant variables. Now, if you look at these different solutions, we're looking at the effect of pH. That's why we have pH buffers. Now a pH buffer is a solution that you add that keeps the pH at one level. So you can use a pH buffer of 3, the pH will stay at 3. A pH buffer of 7 will just keep the pH at 7. The main thing we're looking at then is how does that pH impact on amylase solution? Now what is it that amylase actually does? Amylase breaks down starch and it turns it into glucose. So amylase breaks down starch and turns it into glucose. What that means is, in this experiment, we take our starch solution and we add it to our test tube or beaker. We take our amylase solution and we add it to the beaker. We also add our pH buffer. So we might, for example, this time start at pH 3. We've added them together. Now, every 30 seconds, using a pipette, that's a pipette there, it's just a basic pipette, we take a small sample from the test tube and we add a drop to our iodine. So we add a drop of iodine. Now after 30 seconds, is it likely that the amylase will have broken down the starch to glucose? No. And what color does iodine change to when it's mixed with starch? It turns blue-black. Now 30 seconds after, it still turns blue-black. That means this is still full of starch. Then 30 seconds later, it turns blue-black. Now let's say we come back on the fourth 30 second and it doesn't change color, it stays orangey red, the iodine color, like a brownish reddish color. What does that tell us? That tells us the amylase has now broken down the starch into glucose. So we now know it took, for pH 3, it took 90 seconds. So now what we do is we repeat the experiment, but now we use a pH buffer of 5. Then we do the same thing for 7, 9, 11. Now ideally, we do each experiment 3 times. Why do we repeat the experiment 3 times? So 3 times at pH 3, 3 times at pH 5, 7, 9 and 11. We do it 3 times so we can remove anomalous data. So anything that doesn't make sense, we can remove. How could we make this experiment even better though? We could, once we've understood, so our best results are going to be around pH 7 and pH 9. So we could repeat the experiment using smaller pH intervals. So we could do it at pH 7.5, pH 8, pH 8.5, pH 9 to find closer, to kind of find the optimum pH as well. So why do we use a water bath? The water bath is used in enzyme experiments to maintain a constant temperature. What does that do? It ensures fair and reliable results. It means all our solutions start at the same temperature. Now, it's what I said before, that's important because we don't want the temperature to be too high and we don't want something to cause the enzyme to denature. We're not investigating temperature, we're only investigating pH. Okay, heart and blood vessels. So the lungs are spongy organs in the chest that facilitates gas exchange, allowing oxygen into the bloodstream and removing carbon dioxide from the body. So trachea is the first part of the organs. And it's the main airway that carries, so the air enters through the mouth, it goes down the trachea, the main airways that carry air from the mouth and the nose. So let me give this guy a nose as well. And then the trachea breaks up into bronchi. And then the bronchi break up into bronchioles, which break up into more bronchioles. They're smaller airways, they branch. Okay, I didn't mean this to look like a weird monster or anything like that. These are supposed to be lungs. I am trying to draw a human being here. And the bronchioles, at the end of each bronchiole, you have an alveoli. So the alveoli are tiny air sacs where gas exchange occurs, allowing oxygen to pass into the blood and carbon dioxide to be expelled. This is something I explained on day 1 when we went through when the alveoli came up. When I was covering the topic of diffusion, sorry. Now capillaries around the alveoli. So if you look at this image of the alveoli here. Now when I explained it to you, I drew it a little bit like this. In reality, this is the alveoli. And if you look at all those red and blue lines, they are the capillaries. So you've got all this massive network. You've got this huge network of capillaries that are taking oxygen from the alveoli, and carbon dioxide is being diffusing back into the alveoli so we can breathe it out. So these small blood vessels around the alveoli where oxygen enters the blood and carbon dioxide is removed. There's a lot of capillaries there, and they're only 1 cell thick. They only have enough room for 1 red blood cell to move through at a time. That's to ensure our body gets enough oxygen. So this is what I was trying to draw. I maybe should have just moved forward 2 screens instead. So we've got our trachea, then we've got our bronchi, and then look, the bronchi break up into bronchioles, which break up into more bronchioles. And at the end of each bronchiole, we have alveoli. Okay, the heart. The heart is a muscular organ that pumps blood throughout the body, supplying oxygen and nutrients while removing waste through the circulatory system. So what is the role of the heart? The heart is a pump, and its role is to pump blood around the body. The right side pumps deoxygenated blood to the lungs, and the left side pumps oxygenated blood to the body. The heart contains valves to prevent backflow of blood. Now the way the valves work, because we have valves in our legs as well. When I say in our legs, we have it in the veins which are within our legs. And that's about to come up in a few slides, but I'm just going to explain it now because the word valves has come up. As soon as the blood pushes through the valve, it opens the valve up. So as in it temporarily, the valve opens and the blood goes through. Then, as the blood goes through, it hits the valve from the side and it closes the valve. So now the blood can't come back into this initial region here. So next time the heart beats and it's pumping the blood through, this part of the blood here goes through to the next section. So you have lots of valves stopping our blood flowing back to our feet. And the same thing happens in the heart. The blood enters this part here. Once the heart contracts, it pushes through the valve. But once it's pushed through the valve, the blood fills up and it helps close the valve. So it prevents backflow. Now we've got a double circulatory system. So mammals have a double circulatory system. That means the blood travels through the heart twice in one circulation of the body. What that means is our right hand side sends the blood, the right hand side of our heart sends the blood to the lungs, and the left hand side sends the blood to the rest of our body. So this is the right hand side, so it's as though someone's facing you. This is the left hand side. So the right hand side sends the blood to the lungs, the left hand side sends the blood to the rest of the body. So, and this happens at the same time, so it's double circulatory system. So half is sending in one direction, half is sent to the other direction. So let's quickly go through the different parts of the heart and which ones we need to know. So the atria are the upper chambers. And that receive the blood. The ventricles are the right and left lower chambers that pump the blood out. So this is the atria, labelled 2 and 6. These numbers are not linked to the words above. The ventricles are the lower chambers. So number 1 is 2 and 6. Number 2 is 3 and 7. The aorta is number 8. So the aorta is the main artery that carries oxygen-rich blood from the heart to the body. The pulmonary arteries is number 1. That takes deoxygenated blood from here no apologies, that's number 4 in this picture, and it carries it to the lungs. The pulmonary vein brings the blood back from the lungs to the heart. Then we've got the vena cava, which is number 1, which is the largest vein that brings the deoxygenated blood back to the heart, which is this one. And then we've got these valves which prevent backflow. Now, this is the septum, this middle part here, but you don't need to know about the septum. And coronary artery is actually the artery that supplies the heart muscle with oxygen-rich blood. So even though the blood's inside the heart is supplied has its own blood supply. So we've got So these are the different valves in the heart. So when blood initially enters here, the heart contracts. So the blood initially enters the atria, the heart contracts, and the blood goes into the ventricles. But then the heart contracts again, and the blood on this side goes through to the lungs, which will be through here, to the lungs to get oxygen, and the blood that's on the right hand side will go through here, and that will go to the rest of the body. So blood flow. On the right hand side, the oxygenated blood passes through the vena cava and into the right atrium. So it goes through this blue part here, the vena cava, and it enters, the blood initially enters here. I'm just going to show this as a one direction flow. Now that it's in the atrium and the blood fills up in the atrium, the blood moves from the right atrium to the right ventricle, passing through the tricuspid valve. So it goes through the valve and it enters here. But this blood is deoxygenated. So where does it need to go? It needs to go away from the heart. So it goes through the artery, and it's pumped through the artery, and it goes through the pulmonary artery, and through the pulmonary valve, and it goes to the lungs. Now when the blood goes through the lungs, the blood goes through capillaries, and it takes up lots and lots of oxygen as it passes by the alveoli. That blood then comes back through the pulmonary artery, so the pulmonary vein. So the pulmonary vein brings the blood back to the right atrium, it's the left atrium, which then goes through the valve when the heart contracts into the left ventricle, and when the heart contracts again, it sends the blood through here, and it sends it out through the aorta to the rest of the body. So from the aorta, the aorta will become arterioles and eventually capillaries, and that will then give all the cells in the body the required oxygen that they need. We also have something called a pacemaker, which is a small electrical device that helps control your heartbeat. Now, if the pacemaker stops working, then it can be replaced with a small machine that is called a pacemaker that helps the heart regulate. Because what actually happens is, what the pacemaker does, it sends out an electrical impulse, and as that electrical impulse travels downwards through these external muscles, it causes them to contract. So first this part contracts, the top half, the atria, and that pushes the blood through the valves into the ventricles. Then the ventricles contract, and then that pushes the blood from the right hand side to the lungs, and the left hand side, it goes straight to the rest of the body. So what are the different types of blood vessels in the body? What are the different types of blood vessels in the body, and what can they do? So we've got capillaries. That's the site where molecules are transferred between cells and the bloodstream. So the capillaries are very thin. Veins, they carry deoxygenated blood at low pressure back to the heart. And arteries, they transport oxygen-rich blood at high pressure from the heart. So the artery, now this is a very common question where you compare blood vessels. So it's definitely something that should be memorized. So artery carry blood back to the heart. No, they don't carry blood back to the heart. They carry blood away from the heart at high pressure. They carry blood from the heart at high pressure. Now, arteries have got thick muscular walls, and now that's a description. Thick muscular walls. Why? To handle high pressure. That means it's got a small lumen, so a narrow space inside. So it's got a narrow lumen. Now that's an over exaggeration of how narrow it is, but it's just to show that outside it you've got thick muscular walls and you've got thick elastic walls. So you've got a muscular region and an elastic region. Now it's got elastic fibers. Why? To stretch and recoil. Now if you can feel your pulse, that pulse is because as the blood comes from the heart, from the left ventricle, and it goes into the aorta, into the artery, what happens is the blood goes into the small lumen and it causes it to extend. Why can it extend? Because of the elastic fibers. So it extends, and then anything that's elastic like a rubber band, what happens? It recoils. So that aorta recoils. Then as more blood comes into it, it expands or extends. And again, because it's got elastic fibers, it recoils. That extension, recoil, because of the elastic fibers, is what we feel as our pulse in our wrist, or just by the ear in the neck. You can feel it there as well. The thick muscular walls are just there to handle the high pressure. So you can see here in the structure we've got what it does, a description of what it does, and we've also got the explanation of why. So it's important to understand it from both perspectives. Okay. The vein carries blood back to the heart at low pressure, so this one is correct. They've got thin walls because the pressure is low. So with veins, they've got a large central area here. The lumen is really, really big. So in an artery, the lumen is very small. In the vein, it's got a large lumen to help blood flow easily. And it's got valves to stop the blood flowing backwards. So this is what I was explaining to you guys before. If you look at the leg, and you've got blood going all the way from the feet all the way up to the heart, what's going to slow things down? Gravity. So once the blood flows past this one valve, so it starts here, it flows through, it starts filling this area up, that helps close the valve. So now all the blood has moved from this initial position, and now the next time the heart beats, the blood that's in this initial section will push through that valve and it'll start filling up this section. So now the blood continues to move from one section of valve to the next section where there's a valve, and that stops the blood from flowing backwards as well. Capillaries. The function, it allows exchange of substances like oxygen and glucose between the blood and body cells. It's got very thin walls. Why does it have a 1 cell thick wall? Because then it provides a short diffusion pathway for the oxygen and the carbon dioxide, which are transferring from the alveoli and the red blood cells. So the oxygen is going from the alveoli to the red blood cells, and the carbon dioxide is going from the plasma, the blood plasma, and going straight to the alveoli. They're very narrow, so only one blood cell can pass through at a time. Okay, explain 2 adaptations of alveoli that allow efficient gas exchange. So thin alveolar walls allow only 1 cell thick, which gives a short diffusion pathway. Look, this is an explain question, and what we're doing, we're giving the why. So this is the description, it's 1 cell thick. Why is it 1 cell thick? Because it gives a short diffusion path for gases like oxygen and carbon dioxide. It's got a good blood supply from capillaries. Why? Because that helps to maintain a steep concentration gradient, allowing gases to diffuse quickly. Now here we've got 3 different blood vessels. We've got arteries, so that's an artery. This is a vein, and this is a capillary. Now we know it's a capillary because you can see these cells, it's only 1 cell thick. Describe the job of blood vessel B. A named substance like oxygen, carbon dioxide, or glucose diffuses between the blood and the surrounding tissues. Okay, now this is all a recap of what we've just gone over through, so I'll leave it there just for a couple of seconds. So if you're watching the video afterwards, because after as we've done the last couple of days, everything's uploaded straight away. So you can pause on these kind of questions and really have a good think about them and try to properly understand. Don't forget, this is an explained question. So you write down the description and then you answer why. Why does it do that? So arteries carry blood away from the heart at high pressure. They've got thick muscular walls. Why? To withstand pressure. They've got elastic tissue. Why? To stretch and recoil. Veins carry blood towards the heart at low pressure. They've got thinner walls. They contain valves. Why? To prevent backflow of blood. Okay, I've already covered this left hand side flow of the blood. Okay, moving on to the blood. So blood is a vital fluid in the human body that transports oxygen, nutrients, hormones, and waste products. It consists of red blood cells, white blood cells, platelets, and plasma, playing a crucial role in immunity, clotting, and maintaining homeostasis. So blood carries substances around the body. It transports useful substances such as glucose and oxygen. What do we need glucose and oxygen for? For respiration. And what does respiration release? It releases carbon dioxide and water. Carbon dioxide is a waste product. Too much of it is poisonous for the body, so we have to remove it. Now, just to clarify, not all explained questions are 6 marks. It can be a 2-mark question, a 3-mark question, 4, 5, or 6. It just depends on how big the question is. So some of the explained question that we went through before, because it covered osmosis, diffusion, and active transport, that means you're explaining quite a lot. That's a 6-mark question. The question that we just looked at a second ago, explain how the different structures of arteries and veins relate to the different functions. That's more likely a 4-mark question. So 4 marks because you describe something and then you explain it. So you describe the artery and explain the function. Then with the vein, you describe a part of it and then you explain the function. So you're describing and giving the why, describing it, giving the why for both. So it's most likely a 4-mark question. So the blood is made up of red blood cells that transport oxygen from the lungs to the body tissues and carry carbon dioxide back to the lungs for exhalation. Now, the red blood cells don't carry the carbon dioxide. It is the plasma that carries it. The white blood cells defend the body against infections and foreign invaders. That's something that we'll go into a lot more detail in topic 3. And the liquid part of blood that carries nutrients, hormones, and plasma is the liquid part of the blood that carries the actual red blood cells. So the red blood cells and white blood cells are carried within the plasma. It also carries the carbon dioxide, nutrients, hormones, and waste products throughout the body. And then we've got platelets, which are also carried by the plasma. They help in blood clotting. So if someone gets a cut, it's the platelets that start to seal that gap. So the hemoglobin in the red blood cell only attaches to oxygen. The carbon dioxide is carried specifically in the plasma. What does attach to oxygen is carbon monoxide. So when there's a fire or whenever there's incomplete combustion, the carbon monoxide attaches to the red blood cell, but it attaches irreversibly, meaning the red blood cell is then destroyed. The body has to destroy it and replace it with a new one. Okay, we'll stop there and we're going to continue tomorrow with biology from 2.2.4, which is coronary heart disease. So we're going to take a short break now and we will continue at 5 minutes past 11. Okay, thank you everyone. I'll see you in a few minutes. Okay, welcome back everyone. We're going to start our chemistry topic 2. And again, we are aiming, so let's just quickly see where we're up to. We've done really well, so we've completed cell biology. Just get the pen open. And we have actually completed cell biology, which is fantastic. And we've almost finished organization. So tomorrow we'll be finishing off the organization section and then starting infection and response. So we're not really too far behind. Still doing really well. With chemistry, we are on target. So we finished atomic structure. We have finished periodic table, and we're now moving on to C2 bonding. Now the aim is hopefully to finish C2 bonding off today. But if we have to move it on to tomorrow, that's not too much of an issue either. So I am doing my best to stick to the timetable, but we knew there was going to be a little bit of back and forth, and what we might end up doing is using some of the morning times on Wednesday and Thursday. So if we are behind on a subject massively, we'll probably start the Wednesday, Thursday sessions at 9 a.m. Now, just a reminder to everyone, those that are doing separate science chemistry, tomorrow the session will start at 9, and most likely it'll take between 9 and 10. If I can finish by 9:45, we will still have the question and answer, but please log on for 9 a.m. Now those that can't, not to worry, it'll still be covered. It'll still be recorded and it'll be uploaded, so it's not too much of an issue. Okay, so let's get started. There's 3 types of chemical bonds: ionic, covalent, and metallic. So ionic, the particles are oppositely charged ions. So that happens between a metal and a non-metal. Covalent is a shared pair of electrons, and it happens between when non-metals combine, or non-metals make molecules. Now in a GCSE question, if it says what is a covalent bond, you have to say it's a shared pair of electrons. That's what's going to come up in the notes as well afterwards. Now metallic bond, the particles are atoms it only happens in metals or metal alloys. It's atoms with shared free electrons. So this is where there's delocalized electrons. So metals usually have delocalized electrons, and it's the attraction between the two. So let's start with ionic bonding. What is an ionic bond? So examples of substances with ionic bonds include sodium chloride and magnesium oxide. So an ionic bond is a force that attracts oppositely charged ions. So it is the electrostatic attraction between a negative ion and a positive ion. And an ion is a charged particle formed when an atom gains or loses electrons. So when an atom gains an electron or loses an electron, it becomes positive or negative. And that's the first thing we're going to look at. Now the slides that come after this focusing on how is a positive ion formed and how is a negative ion formed. But I'm going to explain that here before we move on, because it will help make sense of everything else as well. So let's look at sodium as an example. So sodium on the periodic table looks like this. That means it's got 11 electrons. So its electron configuration will be 2, 8, 1. So sodium has 1 electron in its outside shell. So I'm only going to show sodium as having its 1 electron on the outside shell. Now sodium wants a full outer electron shell. So at the moment it's 281, but it wants a full outer electron shell. So it's got 2 options. It can gain 7 more and become 288, or it can lose this 1 electron. So the easiest thing is to lose the electron. So if it loses this electron, then it becomes sodium has just an electron configuration of 2,8. But now, because it's lost an electron, it's become positively charged. But let's compare the two. Why is sodium now positively charged? And that's because the sodium at the top as an atom has how many protons? It has 11 protons. How many electrons does it have? Exactly the same amount, 11. So that means we've got 11 positives and we've got 11 negatives. So what's the overall charge? 0. Now with the sodium plus, remember it's lost an electron. So how many protons does it have? It still has 11 protons because it hasn't lost or gained a proton. And atoms can't lose and gain protons. So we've got 11 positives. Now in terms of electrons, it did have 11, but now it's lost an electron. So now it has 10 negatives. So what's the overall charge? If you've got 11 plus, take away 10, we have a charge of +1. So our sodium has a +1 charge. Now, this is an atom because it has the same number of protons and the same number of electrons, meaning it has a charge of 0. This is an ion, specifically a positive ion, because it has a different number of electrons or protons, because it has lost an electron. So if something loses an electron, it becomes a positive ion. It's same as human beings. If we lose our negativity, so if we're surrounded by people who are a little bit negative, if we've got a friend that's a little bit negative, and we spend a little bit of time away from that negativity, what do we become? We become positive. So human beings and atoms, we behave exactly the same. Now, well, not exactly the same, but there's this one similarity. So now let's look at chlorine. Actually, let's look at fluorine. That's how it would look on the periodic table, which means it has electronic configuration of 2, 7. So it's got 7 electrons on the outer shell. So let's draw fluorine now. 1, 2, 3, 4, 5, 6, and 7 electrons. Now, what does it want? It wants a full outer electron shell. That is where an atom's happiness lies. It wants to be happy, it wants a full outer electron shell. So where does the electron come from? It gains one, most likely from something like sodium. So it's going to gain an electron. So right now, it gains the electron, and let's redraw it now. And let's just draw its outer electron shell. So it's got the original 7, 1, 2, 3, 4, 5, 6, and 7. And now it's got that one gained electron. So now its electronic configuration, it's gone from electronic configuration of 2, 7 to 2, 8. And because it's gained that electron, when a human being is surrounded by negativity, or if you're surrounded by negativity, then we become negative as well. Same with atoms. If they gain that negative electron, they become negative. So let's compare the two. The fluorine atom has how many protons? 9. So it's got 9 positive charges. And electrons, it has exactly the same because it's an atom. So it's got 9 negative electrons. So the overall charge, because it's an atom, is neutral. Now if we look at our fluorine ion, it's just gained an electron. So now protons, it hasn't gained or lost any protons, it can't, so it's still got 9 positives. But electrons, it's gained 1, so now we have 10 negatives. So what's the overall charge? -1. So we have a 1 minus charge for that fluorine ion. Now, if I just rub out some of these things that are on the slide, when we say ionic bonding, what are we actually talking about? We are talking about the attraction between the positive ion and a negative ion. So the positive ion on the left, the sodium plus, and the fluorine minus ion on the right hand side, we have an electrostatic attraction between the two. And that electrostatic attraction between the two is what we call an ionic bond. So now what I've just covered there is, how is an ionic bond formed? Well, if it loses an electron, it becomes a positive ion, like sodium. If it gains an electron, it becomes a negative ion, like fluorine. And then we have an attraction between the two. And we call that an electrostatic attraction. Now when it forms, it will form a giant lattice. So if I show you in a two-dimensional manner, one positive, one negative, one positive, one negative. So if you were to draw this three-dimensional, that positive ion in the middle would have a negative ion attracted to it in every direction. So that's why usually, if you look at a salt crystal up close, it makes like a kind of a cube shape. Now, if I clear this screen, something that's really important to understand. And if you are someone that's going to forget, if you understand it, you don't need to worry. If you're going to forget, then just memorize it. Now, group 1, if you think back to what we've already done, group 1 has 1 electron in the outer shell. So if it loses that 1 electron, it becomes +1. Group 2, for example, beryllium and magnesium, have got 2 electrons in the outer shell. If they lose both electrons, then they become +2. Let's look at fluorine. Fluorine gained 1 electron. It already had 7. It gained 1 more, so it became -1. Let's look at oxygen, which is group 6. Oxygen in group 6 has got 1, 2, 3, 4, 5, and 6, so it gains 2. And when it gains 2, it becomes -2. Now, boron and aluminium, they're in group 3, so they're going to become +3. And nitrogen is in group 5, so that's going to become -3. So what I've just covered on this one slide is many of the slides we're just about to go through. So I'm going to read through them, and if anyone has any concerns or any questions, then you've just got to ask, and I'll go back to that slide. So positive metal ions are formed when metals lose electrons. You need to draw a square bracket around the ion to show the charge on the top, at the top left or the top right. So this is showing lithium. It's lost its outer electron and it's become positive. Now, positive metal ions form when metals lose electrons. So here, fluorine has gained an electron. And because it's gained that 1 electron, it's become negative. And this is what we went through before. Lithium is in group 1. So when it loses 1 electron, it becomes +1. Calcium's in group 2. So when it loses 2 electrons, it becomes +2. Oxygen becomes oxide. It gains 2 electrons. That's in group 6. Because it gains 2 electrons, it becomes -2. Chlorine, which becomes a chloride ion when it reacts, it's in group 7, so it gains 1 electron, so it becomes at -1. Now, I've gone through this myself, and I'm going through this a second time now as I'm going through the slides, because understanding ions is really, really important. It's important because it comes up in ionic bonding, it comes up as part of electrolysis, redox reactions, it comes up again and again. It is one of the fundamental foundation topics. So what happens when a metal and a non-metal react? You end up with a positive ion and a negative ion, and what do we get in between the two? So here, the sodium has given its electron to chlorine. So sodium has, because it's lost an electron, it's become positive. Chlorine has gained an electron, so it's become negative. And we have electrostatic attraction holding them together. So a little bit like magnets being attracted to each other. Now, I'm going to leave this slide here for you to have a look at later. There's just a few questions and answers, and you can check that you've done it correctly. And same with this exam style question. Explain what happens to cesium atoms and oxygen atoms when cesium reacts with oxygen to make cesium oxide. In fact, I will quickly go through this. So cesium atoms, what does cesium's in group 1, so it loses 1 electron. So now it's become a positively charged ion, Cs+. Oxygen gains 2 electrons to become a negatively charged ion, O2-, which means the Cs+ and the O2- ions attract each other and combine to form Cs2O. And that's because oxygen is 2 minus. Cesium is only 1 minus. We have to balance the charges. So for that reason, we add another cesium. And that's why it's CS 2 and 1 oxygen. Now this is a redox reaction where cesium is oxidized, as in it loses electrons, and oxygen is reduced, it gains electrons. Now that's something we'll cover in more detail afterwards, but for those who already understand it and aiming for that higher level, it's good for you to understand it now as well. Ionic compounds. So a large structure made of positively and negatively charged ions that are held together by strong forces. So here you've got the 2D model, the one I used before, a ball and stick model. So you can see that central blue negative ion is surrounded in every direction. It attracts a positive ion. And then you've got your 3D model as well. Now each one has a limitation. So a 2D model, it doesn't show the 3-dimensional arrangement of ions. In a ball and stick model, the sticks don't accurately show how the forces of attraction work in all directions. It also suggests that there's empty space, but there isn't. And 3D representation, it does not indicate the forces of attraction between the positive and negative ions. If you look at that 3-dimensional shape there, it almost looks like the positive ones are next to each other. And if the positive ones are next to each other, what would actually happen in reality if it looked like that? They would repel each other. So that 3-dimensional shape, the representation just shows an impossible structure. Covalent bonds. So before we move to covalent bonds, if anyone does have any questions on ionic bonding, please ask. I will go back and I will go through it. Ionic bonding and ions really needs to be understood to a really good standard. And if anyone's watching this afterwards, if you want to ask any questions in the comments section below, you're more than welcome to, and someone from the AMAL team will do their best to answer it. Or we could make a short. We are on Instagram and we will be on TikTok soon as well, so we will start making shorts to answer people's individual questions. So substances with covalent bonds can be made of small molecules. Now it doesn't say it here, but it should. When atoms share a pair of electrons. It's a shared pair of electrons, and that's what we call a covalent bond. And for example, let's look at nitrogen. Let's look at chlorine. Chlorine, we'll look at drawing this in a second. Chlorine's got a pair of electrons that are being shared. Now you have a positive nucleus in the middle, and you have an attraction between those. That shared pair of electron area there, that's what we call an electron dense region. There's lots of electrons there, and so there's an attraction between the positive nucleus and that dense region of negativity, and that's what holds the atom together. So these bonds are very, very strong. Now what I've just described and explained to you there, you don't need to know for GCSE. I've just said it because it helps to understand it better. So what are covalent bonds? Substances with covalent bonds have very large molecules like polymers. Polymers are made of monomers. So this is something that will come up afterwards. It's something that you definitely need to know for separate science. So here you can see you've got these repeating units. The repeating part is the monomer. And when covalent bonds form between the monomers, you make a larger molecule called a polymer. And then you have these giant covalent structures like diamond, which is what's shown here. And then we've got silicon dioxide, which is sand. So here you've got an example of lots of different covalent bonds. And these are the kind of covalent bonds that you might get asked to draw. So what I'm going to very quickly do is go through a few examples and explain a really simple process on knowing how to draw them. So let's have a look at the one I was just showing you, chlorine. Now chlorine is from group 7, so it's got 7 electrons in its outer shell. So how many more does it need? It needs 1 more electron. Now, however many more electrons an atom needs, that's how many electrons it will share. So if we're making Cl2, because it will tell you chlorine, if it says chlorine, you have to automatically know, and this is a side point now, which I'm just going to reiterate: hydrogen, oxygen, nitrogen, and all of group 7 are diatomic. They do not exist as single atoms. So if you remember Hun7, or Hun7, then these are always diatomic. So if it says to draw the covalent bonding in oxygen, automatically you're drawing O2, nitrogen, N2, hydrogen, H2. Something from group 7 like chlorine, Cl2. So it's just something to keep in mind if you don't already know it. Now, chlorine needs 1 extra electron. We've established that chlorine is Cl2, so we know we are drawing 2 atoms. So let's draw our 2 atoms. Let's make them nice and big. So let's look at chlorine number 1. Chlorine number 1 has got 7 electrons. From those 7 electrons, we're sharing 1. And the remainder, so there's 1, the remainder will go on the rest of the shell. So there's 1, 2, 3, 4, 5, 6, and 7. And chlorine number 2 has also got 7 electrons, so it's going to share 1, and the remainder will go on the outside. 1, 2, 3, 4, 5, 6, and 7. That's it, done. If we now count how many electrons each chlorine has, they've both got 8. So we've got 1, 2, 3, 4, 5, 6, 7, and 8. And the other chlorine has 8 as well. And we can represent it like this. This bond in the middle represents a shared pair of electrons. So that line in the middle represents a shared pair of electrons. You don't have to draw the dots, but that's what it represents. Okay, I am going to do 3 different examples. So now let's move on to nitrogen. So nitrogen, we know as Hun7, so we know it's N2. And we know it's in group 5. So if it's in group 5, it's got 5 electrons in the outer shell, but we need 3 more to get to 8. So how many will each nitrogen share? This time I'm going to do this quite quickly. Each nitrogen is going to share 3 electrons. So nitrogen on the left is going to share 3, 1, 2, 3, and the remainder go on the outside, 4 and 5. The one on the right is going to share 3, 1, 2, and 3, and the remaining electrons from the 5 that it has go on the outside, so 4 and 5. Now if we were to count, we've got 6 in the middle, 7 and 8. 6 in the middle, 7, 8. So now they've both got 8 electrons because of the 6 that they're sharing. Now how would we draw this? Remember a covalent bond is a shared pair of electrons. So a pair of electrons. How many pairs do we have? 1, 2, and 3. So we have a triple bond. Well done if you've already understood this, or you've understood as I'm going through it. Now, slightly trickier one. Let's look at the examples. Would be NH3, CH4. So let's go through one of those. Let's go through NH3. Nitrogen has got, as we already know, nitrogen has got 5 electrons, but it needs 3 more. And we've got 3 hydrogens. Hydrogen's got 1 electron, and it needs 1 more. But we've got 3 of them, because it will tell you NH3, although we should know ammonia is NH3 anyway. So each one of these hydrogens has got 1 electron and it needs 1 more. and they all need one more. So let's quickly draw this. Let's put nitrogen nicely in the middle. Add our 3 hydrogens. Each hydrogen only has 1 electron. And each one of these hydrogens is going to share that 1 electron. And if hydrogen has shared 1 electron, nitrogen needs to share 1 electron too. So it needed 3 more electrons. So it's going to share 3 electrons. So it shared 1 with this hydrogen, 1 with this one, and 1 with this one. And it's still got 2 more. So they're going to go there. So now hydrogen has got 2 electrons. Each one of these hydrogens, there, hydrogen's got 2 electrons. There, it's got 2 electrons, and there, that hydrogen's got 2 electrons. So those 3 hydrogens now have full outer electron shells. Nitrogen's got 2, 4, 6, 8. So it's also got a full outer electron shell. So all 4 of them are happy. Remember, what do atoms need to be in a state of happiness? They need a full outer electron shell. You've got the examples there, so you can go through them in your own time as well. So how do metallic bonds form in metals? So it's the sharing of, now it says free moving, but the better word would be delocalized. So they've got delocalized electrons. And that creates strong metallic bonds. Now what you've got, let's look at a metal. For example, copper. Copper's got 2 electrons. Well, not always, because it's a transition metal, but let's just assume copper has 2 electrons in the outer shell. If it loses those 2 electrons, I'm using copper as an example because we usually use copper in wires. Copper does not necessarily have 2 electrons in the outer shell. Now, let's just assume it loses these 2 electrons. Now, we've got copper 2+. So let's assume every one of these ions is copper 2+. Now, those electrons are free. They're delocalized to move around. So the electrons are free to move between the atoms. So I'll just draw little negative lines. So the positive ions are arranged in a layer or lattice, and they're surrounded by a sea of delocalized electrons. It's the attraction that occurs between the two, between the ions and the negative electrons, by the sea of delocalized electrons, that holds the metallic bond together. So in metals, atoms lose electrons that can move freely. These free moving or delocalized electrons form a sea around the positive ions. This sea helps metals conduct electricity and heat. So the reason metals can conduct electricity is because these free electrons or delocalized electrons. The positive ions are arranged in layers and the electrons can move easily between them. So the electrons can move easily between them and carry that charge, carry that electricity. So this nicely links in with what we covered yesterday in the topic of electricity. Okay, 3 states of matter: solid, liquid, and gas. So with solid, liquid, and gas, I am going to read through it, and what I want you to do is ask questions where if something comes up that you don't understand. I'll explain where it needs to be explained, but if you have any gaps in your knowledge, please keep asking. So solids. Now, one thing before I start, this is where students lose unnecessary marks. Because as students, we've been covering things, or students have been covering things from year 7 linked to solid, liquid, and gas. Most students kind of assume they already know the answer. And what they end up doing is they write the correct answer, but they don't use the correct terminology. And it's one of those topics you think, oh, this is really easy, forget this. I don't need to revise this. And because they don't bother revising properly, they don't put down the correct words. So here it says solids, particles are packed closely together. Now what some people might write for solid is where all the atoms are together, where all the particles are together. But they've missed out packed closely together. Liquids, particles are close, but they can move around, they can flow. And gases, particles are far apart and move freely. You can show that as solid spheres. Again, This is now our better description. Particles are tightly packed with a regular pattern with no gaps. Don't write with no gaps because they are still in one respect a gap. Particles are closely together, randomly arranged. There are, I suppose, there will be some gaps, but I understand why it says no gaps, but I would avoid that, writing no gaps. Particles are far apart and they're randomly arranged. In fact, there's no arrangement. They have so much energy that they've broken free from each other. So use the correct terminology. So what are the limitations of modeling particle arrangements in this way? So number one, particles are shown as solids when they're not. Remember, if you think back to what we've already covered in chemistry, then what is most of an atom? And what did Rutherford discover? He discovered that most of the atom is empty space. So it shows them as solid spheres, so that can't be correct. It doesn't show how fast the particles are moving. It doesn't show the force between the particles. It doesn't show bonds. It doesn't show movement of particles. It shows the particles as spheres. I've pointed ticks here, but I should be pointing crosses. And it's only two-dimensional, not three-dimensional. What's the difference between melting point and boiling point? So melting point is the temperature at which solids turn into liquids. And boiling point is the temperature where they turn from liquids into gases. What determines the boiling point and melting point of a substance? To change state, the forces between particles must be broken. Stronger forces between particles mean a higher melting point. So the stronger the force of attraction, or if there's more force of attraction, means more energy is required to break those bonds. So which substance is a gas at room temperature? Based on its melting and boiling points. So melting and boiling point, room temperature is 20 degrees. So this is the kind of question you can cover up and have a go at. Now, fluorine, it melts at minus 220. So at 20 degrees, have we already gone past minus 220? Yes, 20 degrees is higher than that. So it's melted. It boils at minus 188. So have we gone at 20 degrees? Have we gone past minus 188? Yes. So therefore, it's a gas. It's boiled. Chlorine, 20 degrees, we've already gone past minus 101. At 20 degrees, we've gone past minus 35. So this is also a gas. Bromine, it melts at minus 7 degrees. So at 20 degrees, we've already gone past the melting point, but its boiling point is 59 degrees. So at 20 degrees, we've not reached the boiling point yet. So it's melted but not boiled. So it's a liquid. Water, 0 degrees. At 20 degrees, we have gone past 0 degrees, so it's melted. But 100 degrees, we've not got to yet, so it won't boil yet. So it's only going to be a liquid. Now, mercury, the only metal which is a liquid. So at minus 39 degrees, it has melted, but it doesn't boil till 357 degrees, so it's still going to be a liquid. Now sodium doesn't start melting till 98 degrees, which means at room temperature it won't have melted and it won't have boiled, so therefore it's a solid. Okay, state symbols. In some questions you might be asked to add state symbols. If it's a solid, you put an S, liquid, an L. If it's a gas, you put a G. But if it's aqueous, that means it's something that's dissolved in water. Now, what's usually dissolved in water are salts. Now, when we say salt, we mean an ionic compound. We don't just mean table salt. So, for example, table salt is sodium chloride. When we add that to water, we would, as a solid, as in what we've got in our salt shakers, it's a solid. When you add that to water and you mix it in water, then it becomes aqueous because it's dissolved in the water. Same thing if you had, let's see, lithium fluoride. Lithium fluoride as a salt is a solid, but if you dissolve it in water, then it becomes lithium fluoride but aqueous. So what are the typical properties of ionic compounds? They have high melting and boiling points. They don't conduct electricity when in a solid state. They can conduct electricity when melted or dissolved in water. So they've got high melting and boiling points. They don't conduct when they're solid. So they won't conduct electricity when they are solid. But when they are dissolved in water, because when they are solid, the ions are not free to move. The ions they've got strong electrostatic forces. The ions are not free to carry the charge. But when they dissolve into water, the ions separate. They dissociate. And that's something that we'll cover in more detail when we do acids and alkali. Now that these ions are free, they're free to carry the electricity, they're free to carry the electric current, the charge. So high melting boiling points, that's because these ionic bonds are really strong and they need lots of heat to break them. They don't conduct electricity because the ions are not free to move. And it says here stuck in place, better to say the ions are not free to move, so there's no electricity, so electricity doesn't flow. Once they've dissolved, or they've been molten state or melted, the ions are able to move freely, allowing electricity to flow. Okay, so this question is just asking you to explain the conditions needed for sodium chloride to conduct electricity. So they conduct electricity when they're molten or dissolved in water. So sodium chloride must be molten or dissolved in water. Why? Because remember, this is an explain question. So we started with a describe. We've described what we can see. The sodium chloride must be molten or dissolved. Why? Because then the ions are free to move. Properties of small molecules. So these are simple covalent molecules or simple covalent structures. Now the simple structure, small molecules have a basic structure, making them easy to move. Low boiling and melting points. They have weak forces between the molecules. What that means is, if, for example, that's a nitrogen molecule there. I'm just going to draw it as a circle. Another nitrogen molecule here as a circle, another one here as a circle. What do we have in between each molecule? Firstly, between the nitrogens, between one nitrogen and the other, we've got a strong covalent bond. That bond is very difficult to break. In fact, it's not just one bond, it's a triple bond. So it's really difficult to break. You need about 2000 degrees. That's why nitrogen only breaks apart and becomes nitrogen dioxide when you have lightning, or in car engines where you've got combustion occurring and really high temperatures. But between each individual molecule, you've got weak intermolecular forces. And for that reason, there's low melting and boiling points, because it doesn't take much energy to break these weak intermolecular forces. By the way, I was trying to make just the bonds, the weak intermolecular forces disappear, but the whole thing disappeared. Never mind. That's the power of the eraser. Now, solubility. Some dissolve in water or liquids, making them useful in many processes. They're non-conductive. And why they're non-conductive? Because they do not have free electrons. The electrons aren't free to move. Why can a solid ionic compound not conduct electricity? Because the ions are not free to move. When can the ionic compound conduct electricity? When the ions are free to move, when they're dissolved. Now with a covalent bond, can they conduct electricity? No, because they do not have any free electrons. Where are all the electrons? They are inside the covalent bond. There is one non-metal, or there's a category of non-metals that conduct electricity, which is the graphites. Graphite and graphene, they conduct electricity because they have a free electron. But that's about to come up and we will go through that. Explain why iodine has a low melting point. So iodine is a simple covalent structure. So if it's a simple covalent structure, so it's a molecular substance, so simple covalent is the better thing to write. And what do we have in between each iodine molecule? We have weak. You can write van der Waals forces or you can write intermolecular. Both will get you the marks. And in different schools, you will have been taught one or the other. So you don't actually need much energy to break that bond. Let's see if the eraser erases the right thing this time. So not much energy is needed. It didn't just erase, it took me to another slide. Okay. Oops. I need to get used to using that eraser properly. Polymers. What are the properties of polymers? So the intermolecular forces between polymer molecules are relatively strong, and so these substances are strong at room temperature. Now, polymers are huge molecules made up of smaller units. The atoms in the polymer are connected by strong bonds called covalent bonds. But what we're actually saying here is, let's say you've got a long chain polymer. Lots and lots of carbons attached to each other. And how will that long polymer attach itself to another polymer of a similar length? It will attach itself to that polymer with weak intermolecular bonds. The bigger the carbon chain, the more intermolecular bonds. The more intermolecular bonds, the stronger the overall bond. Even though they're weak, even if you've got lots and lots of them, then it takes more energy to break them. So polymers still make relatively strong solids because there's so many intermolecular forces, bonds between them. So the longer the polymer chain, the more intermolecular bonds, the stronger the overall attraction between the two, the stronger the bond. Giant covalent structures. Substances with giant covalent structures are solids that have very high melting points. So what we're talking about here is diamond, graphite, and okay, so these are slightly in the wrong position. It's because it comes from an activity where you draw lines between the correct ones. So let me erase it and I'll just draw lines to the correct ones. So I'm assuming you all know where each diagram goes. So we've got graphite, well, diamond, which is this one. Graphite, which is this one. And silicon dioxide, which is this one. But all of them are giant covalent structures. And because you've got so many covalent bonds here, it takes a lot of energy to break them. So they've got very high melting and boiling points. Diamond, in fact, is the melting and boiling points are estimates. So why do they have high melting and boiling points? Because this is now talking about metallic bonds. They've got strong metallic bonds that need a lot of energy to break. Why are they good conductors of electricity and heat? Because delocalized electrons move freely carrying the charge and the heat. Why are they malleable and ductile? Because the layers of atoms slide over each other, allowing metals to be bent and stretched. This is why Rutherford was able to use gold leaf, because he was able to take a small amount of gold and hammer it, or get someone to hammer it over and over until it was only a few atoms thick. They're strong and dense because it's got closely packed ions that makes them hard and durable. The shiny and sonorous part don't matter too much, but you do need to know the property. Knowing the property from a foundation perspective is still important, but you don't need to know the explanation per se. Now, why are metals good conductors? Give 2 other properties. So this is just a question. You could put any of these properties as part of the explanation. So this is more of a foundation question. No, actually, so this would be higher as well, because it's asking why. Why are metals good conductors? So you're explaining the why. And the giving 2 other properties is more from a foundation perspective. So you could give any of these properties. Why are alloys harder than pure metals? So alloys are harder than pure metals because the atoms in alloys are of different sizes. This makes the layers in the alloy distorted. Because the layers are distorted, they cannot slide over each other easily. So metals have a regular arrangement like this, which means pure metals, for example pure gold, is actually relatively soft. You could squeeze it slightly in between your fingers. So for example, if I put a force here, it will cause all the atoms to move across because they roll over each other. Now when you add something to the metal, another metal that has a different size, the atom has a different size, it then distorts that regular arrangement. And by distorting the layers, it now means if I, for example, push here, that atom isn't going to roll because it's going to collide with that big one. If I push from here, then this blue atom, the larger atom there, stops that metal from rolling in that direction. Now, because the layers are distorted, they cannot slide over each other as easy, so it makes it stronger. So whenever someone goes to, for example, to a jewelry shop and buys pure gold, in reality, it's not pure gold. It's an alloy. They would have started with 24 karat gold, but they have to add a small amount of alloy to it. So usually it's 22 or 23 carats. They have to add that small amount of another metal to make it strong enough to become jewelry. Otherwise, if they made a ring purely out of gold and nothing else, it would lose its shape quite quickly. Because an alloy is a mixture of 2 or more metals, or a metal and a non-metal. So what percentage of this alloy is that one? So all we're doing here is counting how many of the blue circles divided by everything. So there's 3 blue circles, but there's 15 atoms altogether. So it's just 3 divided by 15, which is 20%. So the platinum and iridium alloy is harder than pure platinum. So this just showing you the kind of common questions that come up. So explain why alloys are harder than pure metal. So exactly what we've already gone through. Alloys have different sized atoms, disrupting the regular arrangement. This prevents layers from sliding easily, increasing hardness. In the platinum iridium alloy, iridium atoms make it harder than pure platinum. Now, this goes back to what I was explaining with the whole AMAL method. The AMAL method is activate your brain, understand everything. So once you've understood, you can look at a question like that and you understand why an alloy is harder than pure platinum. So why platinum and iridium alloy is harder than pure platinum. However, it's only through memorization that you write down the key answers. And how do we know what to memorize? It's through past paper practice. So the more past paper practice, so this is the A part again. So A, activate your brain, understand. M for memorize, so you memorize this key understanding. And then A again from AMAL is to apply. And when you apply, you start to understand what needs to be memorized. You start to understand the examiner's mindset. If you do all of these 3 things, you have learnt. Not only have you learnt, you've taken yourself much, much closer to a grade 9. Any of my students that have ever got that, well, thankfully a lot of my students do get grade 7s and above. In fact, last year, for the students that I taught in AQA, I would say it was around 90% of my students got 7s, 8s and 9s. The only ones that didn't are the ones that came to me that were foundation students. So I'm expecting the same for my students this year. I'm hoping and expecting the same from anyone that's listening in at the moment. But what method did they follow? The AMAL method. They activated the brain, they memorized, they applied. So I've had students that have gone from grade 3s, 2s and 3s, they've gone all the way to grade 8s and 9s. In fact, just a few years ago, the student at the beginning of the year, she was completely failing. She wasn't even in top set. She was heading towards getting around a grade 2 or grade 3 just from following this method. And she only used this method for around a month. So it doesn't take more than 4 weeks to use it. So we've got 4 weeks left. This is the prime time to use it. In those 4 weeks, what she did was anything she didn't understand, she came to me and said, I don't understand this. In fact, it wasn't just her. There's a group of 15 students. And all these 15 students that came to me every morning, they all got grade 7s and above. But this particular student got grade 9s. And she went from a 2 or 3 to a grade 9. Whereas the others went from maybe a 3 or 4 to a grade 7, 8. So what they all did was they came to me and said, we didn't understand such and such topic. Can you explain it to us? So I explained it to them. So that was activate. As I was activating their brains, we were understanding, I was telling them bits to memorize. And they were doing around 50 marks worth of questions every day. Meaning, because these students were in combined, they were finishing a paper. They were finishing about I'd say 6 papers a week. So they're doing 2 biology, 2 chemistry, 2 physics papers a week. And if they didn't understand something from those questions, they again came back, and they came back to the activate phase, and they went back because that question showed them what they weren't understanding. But the more questions that they did, the better they got, and the less questions they were asking of me, because they started to understand what the examiner was asking. But like I said, if they didn't get a question right and they couldn't understand the mark scheme, they came straight back to me. So you guys can do a similar thing. We're doing the activate phase now. If you're going to start memorizing as well, you're going to start now looking at past papers as soon as the AMALathon's finished. If there's still something that doesn't make sense, just send us a message through the YouTube channel. And when we do our pre-exam workshops, I'll make sure I cover and I help answer those questions. So you can shape what the pre-exam workshops look like. So metals as conductors. Why are metals good conductors? Metals are good conductors of electricity because they have free moving delocalized electrons. So this is something we've already understood. Let's move on to diamond. Now, with diamond, each of these carbons forms 4 bonds. Then the next carbon also will form 4 bonds. It doesn't show it here, but it'll form 4 bonds. Every single carbon is forming 4 bonds. So that means you've got a lot of covalent bonds. Covalent bonds take a lot of energy to break. And for that reason, diamond is very hard. It has a high melting boiling point. All the electrons are stuck within these covalent bonds, so it can't conduct electricity. So let's have a look at this. Now, properties, I would memorize the explanation, particularly those doing higher, those in for grade 9. Why is it hard? Because of all the covalent bonds. Why can it not conduct electricity? Because the electrons are not free. Why has it got high melting point? Because of all those covalent bonds, which means you need a lot of energy to break the covalent bonds. And transparency, diamond is transparent because light passes through with little absorption. You don't need to know that for the AQA spec. However, it's just an additional piece of information that we've added. Graphite, a giant covalent structure where each carbon atom is bonded to 3 others. So now if we look at this structure here, you're going to see 2 things. You've got 1 layer there, 1 layer there. Now each carbon is attached to 3 other carbons. So we're specifically looking at this one here. The fact that it's attached to 3 other carbons, carbon is in group 4. It's got 4 electrons. So it's got 1 electron here, 1 electron here, 1 electron here, and this 4th electron is free to move. It's delocalized. It's not bound to a bond. It's not part of a bond. Because it's not part of a chemical bond, it's free to move. Now, for that reason, graphite can conduct electricity. Now, it's got lots and lots of covalent bonds, which means it's got high melting boiling point because lots of energy is needed to break those bonds. As you can see here, it's in layers. So you've got a top layer there and another layer underneath. Between those layers, you've got weak intermolecular bonds, which means the layers can slide over each other. In fact, that's how your pencil works. So your pencil, the lead inside your pencil, when you're using the pencil, what you're actually putting on the piece of paper is lead. It is the lead that you're putting onto the piece of paper. And when you push down on it, your force of your hand is enough to break the intermolecular bonds and then release layers on to the actual surface. And that makes it soft. It makes it a good conductor of electricity because it's got the free electron. It's got high melting and boiling points because of these strong covalent bonds. It can be used as a lubricant because they slide over each other. So it's soft and slippery because the layers slide over each other. It conducts electricity because it's got free moving or delocalized electrons. That's why when we do electrolysis, we use them in electrodes. We use them in electrodes because for two reasons. We use graphite as electrodes because they can conduct electricity. And two, we use them as electrodes because they are unreactive. So they don't interfere with the electrolysis process, with the reaction that's occurring. High melting point because they're strong covalent bonds. Graphenes and fullerenes. So graphene is a single layer of carbon atoms. So it's like graphite, except just one layer of graphite. So just like graphite, it's got carbons which are attached to only 3 other carbons, which means it's got one free delocalized electron. So just like graphite, it can conduct electricity. So it's got a high melting and boiling point because it's got lots and lots of covalent bonds, and that requires a lot of energy to break. So you can see I'm saying the same thing over and over again. You just need to know what it applies to. But if something's got higher melting and boiling point, if it's ionic compounds, it's because of the strong electrostatic attraction between the ions. But when we're talking about covalent bonds, it is because it takes a high amount of energy to break the covalent bond. The covalent bond is a strong bond. It's got high strength, again because of all the covalent bonds, and electrical conductivity. It has a free moving or delocalized electron that allows it to carry that electrical charge. What are fullerenes? So fullerenes are molecules made of carbon atoms arranged in hollow shapes. So you could say nanotubes, such as spheres, tubes, or cages, made of carbon atoms bonded in hexagonal, sometimes pentagonal or heptagonal rings. That is Buckminster fullerene, a buckyball. So that's C60. Now, they're made of carbon rings as hexagons, pentagons, heptagons. So that means we can use, because they're quite small, and you've got, essentially you can make little kind of football shaped balls out of them, like the Buckminster fullerene. You can make tubes. It means we can use them as in the form of nanotechnology, and we can use it in electronics. And within that nanotechnology, actually it mentions it here, you can use it to deliver medicine. They're strong and lightweight. So carbon nanotubes are stronger than steel, but they're very light. So they're used in sports equipment and aerospace materials. They're good conductors of electricity because some fullerenes have delocalized electrons. So you can use it in electronics and solar panels. They've got high surface area. So that means we can use it to deliver medicine. They're slippery and low friction. So for example, the Buckminster fullerene molecules, they roll over each other like ball bearings. So for that reason, they can be used as lubricants in machines. Okay, now what are carbon nanotubes? So carbon nanotubes are tiny cylindrical structures made of carbon atoms arranged in hexagonal rings. They are a type of fullerene and are extremely thin but strong. So this essentially is similar to what we've just covered, but it just is specific to carbon nanotubes. They're very strong and lightweight, so they can be used in bulletproof vests, sports equipment. They conduct electricity, so they can be used in electronics. They conduct heat well, so they can be used in cooling systems. They're flexible but tough, so you can use it in wearable tech. It's got large surface area, so you can use it in drug delivery. Okay, we're going to stop there. Oh, actually, there's only 2 slides left, so I'm just going to finish it and then we'll take a 5-minute break. So, size of particles and their properties. So what are the key terms used related to size of particles. So when we say nano science or nanoparticle, nano means times 10 to the power of minus 9. So we're talking about things that are very, very small. A coarse particle is usually something that's a micrometer, 1 times 10 to the minus 6. And a fine particle is usually around times 10 to the minus 8, but it's bigger than a coarse particle. So it's smaller than a coarse particle. Generally speaking, it's somewhere between a nanoparticle. So if you think of a coarse particle is times 10 to the minus 6, anywhere in between a nanoparticle. So nanoparticles are very small. So I should have done it the other way around. So let's say we've got times 10 to the minus 6 is your larger particle, that's coarse. Then you've got times 10 to the minus 9, that's your smaller particle. Anything in between we would refer to as a fine particle. Okay, now how does particle size affect surface area to volume ratio? Reducing a cube side by 5 increases the surface area. So that's reducing a cube's size by 5 increases the surface area to volume ratio by 25 times. Now what that means is if you've got a large cube and each side of that cube is 5 centimeters, if you break that cube down, yes, apologies, I forgot to mention this, 2.4, this whole part is triple award, is separate science only. So apologies, I completely forgot to mention that. Thank you, Dean, for mentioning that. Now, if I now break that up. Because that's 5 by 5 by 5. If I break that up, what I'll end up with is small cubes that are 1 by 1 by 1, but I'll end up with 25 of them. So I've increased my surface area by 25 times. Smaller particles, sorry, I won't end up with 25 of them, sorry, but if you make smaller versions of them and you compare the surface area of the two, it'll be 25, as in be squared of however much you made it smaller. Smaller particles have a higher surface area relative to their volume. So nanoparticles often require smaller amounts to achieve similar effects. Now, if you make something smaller and give it a larger surface area, then it makes it more effective. So you don't actually need to use as much nanoparticles as you would if it was larger. So it's more effective and you don't have to use as much of it. So what are the different uses of nanoparticles? This is the last slide. We've already covered this. We can use it for medicine, we can use it for catalysts, and we can use it in electronics, in cosmetics, in deodorants, and in sunscreen. Okay, brilliant. Excellent, everyone. Thank you very much. I know I've gone over the time, but I'm happy to do that a little bit extra so we can finish. I do want by the end of the AMALathon to have covered everything. So we are going to recommence at 12:15. So I'll see you all in 5 minutes. Thank you, everyone. Assalamualaikum, Sarah. I know it's not me, and I really am. I'm against the use of it, to be honest. I just don't like. Let me show. Okay, welcome back everyone. And for those that have just joined us, good afternoon. Thank you for joining us. This is now the third segment of AMALathon Day 3, where we're going to finish off physics. So the aim of today's lesson is to finish off the topic 2, physics, hopefully with some good understanding. Now I know this is a topic that for a lot of people can sometimes come across as a little bit more challenging. So if anyone has any questions, please keep asking. And if I'm not able to answer the question straight away, then I will make sure I answer it tomorrow morning in our question and answer session. Sorry, I'll just make sure everyone can see. There we go. Okay, so how can we investigate the relationship between the length of a wire and its resistance? So this is what I explained to you yesterday, but I'm going to just do a quick recap. So what they're actually showing you here is if you have your circuit in place, and it's going to show you this afterwards. If you've got your circuit in place, and within that you put a variable resistor, then you put either your filament lamp or bulb, or you put your diode within this complete circuit. Make sure it's got an ammeter which is in series and a voltmeter in parallel. Then as you keep changing the resistance, you take readings from your ammeter for the current and from the voltmeter for the potential difference, and you can then make an IV graph. And this is quite typical of a filament bulb, and this is quite typical of a diode. And that's because the resistance of a filament lamp increases with the filament's temperature. So when you have an increase in resistance, it's like what I was saying to you yesterday. It makes it more difficult for the electrons to get from place to place, to get through the wire. So that means at a certain point, you can keep increasing the potential difference, which is what we can see here, but the current stops. It can't go beyond a certain point. So remember, potential difference is like the push in the circuit, and the current is the electrons carrying the charge. Here, because there's such a high resistance, not many electrons can get through, or get not beyond the point that's already reached. Now with a diode, current only flows in one direction. That means you've got a massive resistance on this side, and it shows high resistance in one direction, and in the other direction it's an easy flow for the electrons. Hence you've got a line going upwards like that. So a small increase in potential difference causes a massive increase in current. Okay, so let's have a quick look at the thermistor. Now the notes that are on the screen are good, but I'm going to explain this in my own words. I'll read through what it says here first. The resistance of a thermistor diminishes as the temperature rises, meaning the resistance decreases as the temperature increases. So as temperature increases, the resistance of the thermistor decreases. So on a hot day, as the temperature gets hotter and hotter, the resistance gets lower and lower. So on a hot day, you have low resistance. On a cold, cooler day, you've got higher resistance. So at high temperatures, a thermistor has low resistance, allowing increased current flow. At high temperatures, a thermistor shows low resistance, which enables increased current flow, which is the same as what's written there. And thermistors are often used in thermostats. So this is a symbol for thermistor, so make sure you learn it. Now, a thermistor is a temperature dependent resistor. So it is a resistor, hence you have the rectangle shape to show that it's a resistor. its resistance changes with temperature, which I must have said 4 or 5 times now. It's a type of sensor, and it's often used in things like thermostats, ovens, fridges, and even phones and computers to monitor temperature change. Now, a common question that I always get asked, but you don't need to know for GCSE level, is why does resistance go down when temperature goes up? I'm going to explain it because I know a lot of students don't like it. Becomes a bit of a kind of sticking point that, do I only need to know this point here, that when the temperature goes up the resistance goes down? And for GCSE that is sufficient. However, it's always beneficial just to understand why it happens. So at higher temperature, the thermistor's material gains more energy. So the material's got more energy. That means more charge carriers, like electrons, are released inside the thermistor. So at higher temperature, the thermistor's material, so whatever it's made up of, gains more energy. That means there's more electrons carrying charge released within the thermistor. This means current can flow more easily, so resistance drops. So what we're saying here, as the temperature goes higher, the more electrons are released, and because more electrons are released, the resistance is less because we've got more electrons flowing through. So in a real world example, I suppose, when it starts, for example, if a computer starts to overheat, the thermistor senses the rise in temperature. That means its resistance drops, which triggers the fan to turn on, cool down. So let's try to understand this. We've got a computer. I'm just going to draw an old-fashioned type of computer. It starts to heat up. Inside there's a fan. There's a thermistor attached to a fan. The thermistor increases in temperature, but now the resistance has decreased. Why? Because we've got more electrons flowing through. More of those electrons reach the fan. So now the fan's got more charge reaching to it, so the fan starts working and it cools down the computer. Now I get asked similar questions linked to LDRs, as in, how does an LDR resistance change with light intensity? But students always ask me, how does it actually work? And it's actually similar to a thermistor. But I'll read through what's on the screen first. So the resistance of a light-dependent resistor, an LDR, so look, it's still a resistor, hence the rectangle in the middle, lowers as light intensity increases. The circle part there represents the light. Now in low light, a light-dependent resistor has high resistance, allowing minimal current flow. In bright light, a light-dependent resistor has low resistance, allowing increased current flow. So we use LDRs to activate lights automatically in dark environments. Now what's there is sufficient for your exam. However, I am just going to explain in just slightly more detail so everyone understands it. So an LDR is a resistor whereby resistance changes depending on how much light shines on it. So we use it for things like street lights, automatic night lights, light meters in cameras, alarm systems. But why does the resistance drop in brighter light? So when more light shines on the LDR, more electrons are released, or more electrons and materials gain energy. That means more electrons are released, so the current flows more easily and resistance drops. So let me repeat that. In brighter light, you have more flow of electrons through the wire. So you've got more flow of electrons, you've got more charge carriers, more electrons, so the current flows more easily. It increases. So that means, in other words, the resistance has dropped. So in real life examples, which we've already said, during the day there's lots of light, so LDR has low resistance, so the lamp stays off. At night you've got low light, the LDR has high resistance, which triggers the lamp to switch on. So now, the bit that we're covering is what we started yesterday. Apologies, I'm just having a look at the live chat. Okay, now to measure the current, so this is what I just explained to you just a few minutes ago. If you've got this set up, all you've got to do is change the component that's here. And if you have a resistor, what you should get the following IV, that means current voltage profile. So this is how you would carry out the experiment. So this is what I explained before. How can you investigate the IV characteristics of a resistor? Same with a filament bulb or anything that you add to a circuit. So you set up the equipment according to the diagram. You record the current voltage and readings. You modify the variable resistor for 4 additional times. So here you're changing the resistance of the circuit, noting the new current and voltage. So you're recording the current and voltage every time you change this variable resistor. And with that, then you reverse the connections here on the battery so that both the ammeter and voltmeter display negative readings, and you do exactly the same thing again. That means you're getting 4 positive readings and you're getting 4 negative readings. And then again you measure the current and voltage and you can draw the graph. So this is the graph that you'll get for a resistor. So your potential difference is your x-axis and your current is your y-axis. So you do exactly the same thing, but when you use a light bulb or filament bulb, you do 4 positive, 4 negative. But where we had the resistor before, we're putting the bulb there instead. And again, you change the resistance 4 times using the variable resistor, take your recordings, then you reverse the positive and negative. So you get 4 negative readings and you draw your IV characteristic graph. It's important to know what these graphs look like. I've seen questions before where they've drawn this first half and it was 1 or 2 simple marks and it just said finish it off, and all you have to do is draw the second half. In fact, I'm sure it's in this presentation, so we'll go through that. How can we investigate for a diode? So exactly the same thing. You add the diode, change your variable resistor 4 times, take your 4 readings of current and voltage. Then reverse your positive and negative, take 4 more readings, and you end up with a graph similar to this, because a diode only allows current to flow in one direction. So when you reverse the charges, when you reverse the battery or the cell in one direction, however much you change the potential difference, however much you change the resistance, the current doesn't increase, it stays 0, because you've got such high resistance. Now, a student investigated how the current in a filament lamp varied with the potential difference across the filament lamp. So the diagram shows part of the circuit used. Complete the above diagram by adding an ammeter and a voltmeter. So the thing to remember here is, how do we add ammeters? We add ammeters we add it in series. And how do we add voltmeters? We add them in parallel. So we would add it like this. Now the way this was originally drawn, you would lose marks because you've not actually attached the voltmeter to the circuit. So here's another one. The student reversed the connections to the power supply and obtained negative values for the current and potential difference. So draw a line on the graph to show the relationship between negative values of current and potential difference. So we've already got this part here. Now all you're doing is taking the coordinates and you are just doing them again, but negative. So here we've got 0.08. So we would do the same thing there, but negative, and that's 1. That's 1, so we would draw our first cross there. And then that would help us draw the line. So everyone would know that the line looks like this, but you've got to make sure that you get the right coordinates as well. Okay, series and parallel circuits. So what are the similarities and differences between series and parallel circuits? So now this is something that's important for us to understand the difference between. So there's 2 ways to connect electrical components. You can either connect them in series or you can connect them in parallel. So certain circuits incorporate both series and parallel components. So the first thing we need to be able to do is identify which one is which. So we know that this is series and this is parallel. So when you've got multiple circuits added in one, that's when you call it a parallel circuit. One loop is series, multiple loops is parallel. Now, in a series circuit, and these are the key points to remember here, each component experiences the same flow of current. That means wherever I put an ammeter here, I'll get exactly the same number. So if there was room to put in between the 2 bulbs there, I would get the same current. So if the current here was 2 amps, what would it be here? It would be 2 amps. What's it going to be here? It's going to be 2 amps. The current stays the same. The total resistance of 2 components is found by adding up their individual resistant values. So whatever the resistance of this bulb is and whatever the resistance of this bulb is, that's the resistance of the total circuit. So how do you find out the resistance within a series circuit? You add up the overall resistance. The power supply's total potential difference is shared among the components. So if we say this is 6 volts, I'm just going to use the eraser to make a bit of space here. So if we say 6 volts comes from our power supply, if, for example, 4 volts is used by our first bulb, that means the remainder, 2 volts, will be used by the second bulb because it's shared among the components. Okay, now, so if I just quickly go back to that, you need to remember that the current is the same. So I stays the same. R is this here, that's the total that shared amongst the components. And the voltage is also shared. So whatever the potential difference is coming from the power supply, that's shared amongst the components. So potential difference is shared. Now in a parallel circuit, it's the other way around. The voltage across each component remains consistent. So that means the voltage remains the same. So if I put a voltmeter here, if I put a voltmeter anywhere in the circuit, it will give me the same answer. The total current in the circuit, so if over here, the current is 5 amps, what happens when we reach a branching point is, if, for example, if 3 amps go this way, that way, and in this direction, the 2 amps go. So at the branching point, the current splits. So all the way in this direction, we've got 2 amps flowing through, and across the middle there we've got 3 amps flowing through. When they rejoin, if we measure the current here, it'll be 5 amps again. So if we measure the current, using that same example, if we measure the current, so we started with 5 amps. If we measure the current here, it'll be 3 amps. If we have an ammeter here, it'll be 2 amps. But when they reconnect here and we measure the current, it'll be the sum of both, so it'll be 5 amps again. Now, the combined resistance of 2 resistors, so this component here has a resistance, this component has a resistance, every component naturally has a resistance, but the combined resistance is lower than the smallest resistor among them. So let's say this first component here has a resistance of 10, and the second component here has a resistance of 5 ohms. The overall resistance within the circuit is actually less than 5. Now I'm going to explain that in a bit more detail, because again, this is something that's poorly understood. Once it's understood, it makes sense. Now, so what we're saying is whatever the resistance of both these components is, the overall resistance is less. Now, in a parallel circuit, the combined resistance of 2 resistors is lower than the smallest resistor among them. So here, if this bulb has a resistance and both these bulbs have resistance, whatever the lowest resistance is, the overall resistance is less than that. Now, what does that actually mean? So let's say we've got 2 resistors here. One is, let's use 6 ohms instead. So this is 6 ohms and this one is 3 ohms. You might expect that the total resistance, you might say it's 9 ohms, or you might say it's somewhere between 6 and 3, but in reality it's less than 3, because it's less than the smallest one. But why is that the case? Because in a parallel circuit, current has the electrons, so we're talking about the electrons here, the current has more than one path to flow through. So the current can flow this way, or the electrons of the current can flow this way. So because the parallel circuit, the current has more than one pathway to flow through, it's kind of like extra lanes on a motorway. Or if we think of the example I gave yesterday, in your cafeteria, if you had to get from one side to the cafeteria and to the other side, if there was only one path, that would mean there's always going to be high level of resistance. However, if there was another pathway where you could turn right, and you could, for example, walk down a corridor, turn left down another corridor, turn left down another corridor, and end up in exactly the same place, you've now got 2 pathways to get to the same place. Or it's a bit like when you're on if someone's driving a car on an A road and there's only one lane, they can only go at a certain speed. But when you go to the motorway, there's loads of extra lanes. So what happens when you have lots of extra lanes? It reduces the traffic, because everything spreads out. And that's what's happening here. The current is being spread out. Some's going one direction, some's going in another direction. So when you add something in parallel, you're actually giving the current another direction to go down. This actually makes it easier for the current to flow overall. So what happens to the resistance? It drops. That means the more branches you have, the less resistance. So I hope that makes sense. Overall though, I hope that makes sense, helps you understand what we mean by current. Because again, current is, resistance. Again, because resistance, current, voltage, there's sometimes parts of electricity which are not always understood very well. Okay, Dean, if you can just I think I've just answered your question. Can you please explain the resistance of parallel in detail? Did that make sense? If you don't mind answering back, so I know before I continue. Well, I'm going to continue, but I'll circle back to it if it still doesn't make sense. Now, what is direct and alternating current? Now I'm going to explain this in detail as well. What's on the slide in front of you is what you need to know. But this is the understanding phase. This is the activate phase, so it's important that you just have that understanding because it makes it easy to apply. So direct current. Excellent, thank you, Dean. So what is direct and alternating current? Direct current, a current that passes in the same direction. Current produced by cells and batteries. An alternating current is current that changes direction. Current in the national, for example, current in the national grid. Let's go through these one at a time. Let's look at AC first. Now, alternating current, the current switches direction repeatedly. It goes forward, then back, many times per second. Now, I'll go into a bit more detail afterwards, but because it's constantly moving back and forward, it means it's almost like it's oscillating. So in the UK, mains electricity, when it's alternating current, it has a frequency of 50 hertz. It means it changes direction 50 times per second. So alternating current is one that constantly changes direction. Voltage in alternating current also changes continuously from positive to negative. So if you look at it as a wave, on an oscilloscope screen, it looks like this, like a sine wave. Because the voltage is constantly going positive, negative, because the current is constantly changing direction. Now, in direct current, actually, I'll explain alternating current first. So, but why does alternating current constantly change direction? It's something that we're going to learn in paper 2. So alternating current, so in power stations, electricity is generated by rotating turbines. Now, what that means is you've got turbines that turn coils of wire inside a magnetic field. So you've got, for example, something like that, and it's got loads of coils of wire around it. And around this, you might have magnets. Now, if you've got all these magnets, now this is just one design, by the way. There's lots of different designs for these. Now, that coil of wire is attached to, for example, the wind turbine. When the wind turbine spins, this coil of wire spins as well. When that coil of wire breaks the magnetic field, because these are all magnets, you have a magnetic field between them. They're all going to have magnetic fields. So this whole region is a region of magnetic field. So as soon as you break that magnetic field, within that coil of wires, what you're actually creating is a current, so an electrical charge. In other words, you're making electricity. But because when something is spinning upwards, current is made in one direction. When it's spinning downwards, current is made in the opposite direction. So when something spins, when it spins up, you make current in one direction. When it spins down, you make current in the opposite direction. So this is Fleming's left-hand rule that helps you understand which way it is, and that will come in the second paper. That's why the alternating current alternates. So that back and forth movement causes alternating current. That's why we have, in the UK, our alternating current has a frequency of 50 hertz, as in it's changing direction 50 times per second. So it's going positive voltage, negative voltage, positive voltage, negative voltage, 50 times per second. Now, direct current is when the electrons flow in one constant direction. So that comes from things like batteries, solar panels, USB chargers, etc. Voltage stays at a steady level. So if we look at this, if we were to look at direct current, it would just be a line at the top or just a line at the bottom. If we were to compare it to our oscillating wave there. Now, why does the direct current flow only in one direction? That's because inside cells or batteries, there's a chemical reaction, and inside the battery that pushes electrons in one direction only. So inside a battery, you've got a positive side and a negative side. So the current flows steadily from the negative side of the battery to the positive side. So it's only going in one direction because you've only got one negative side, you've only got one positive side. That means the voltage stays constant. It means that electricity flows out of it, the electrons flow out of it in one direction as well. So you can look at it as water flowing through a pipe. It's impossible for water to flow in both directions through only one pipe. So I hope that makes a lot more sense out of AC and DC. Because again, this is one of those common questions where students ask me quite often that when you're saying AC, what do you mean? And from a GCSE perspective, you only need to know that it as in the current changes direction. And for direct current, you need to understand that it goes in one direction or the same direction. But it's always good to understand it from from the higher level. Okay, potential difference. What's the potential difference in frequency? So this is something we have now automatically covered. In the UK, the primary electricity supply is alternating current. So it operates at a frequency of 50 Hertz and a potential difference of 230 volts. You need to memorize that. It does come up as a question. Not always, but I have seen it as a question. So it's best to know. Best to have learned from beforehand. Okay, so how does a plug work? Now, I wonder if anyone can see which color is wrong here. So most electrical appliances connect to the mains using a 3-core cable. The insulation on each wire is color-coded for straightforward identification. So we got earth, which is yellow and green, so that's correct. Neutral, which is blue, and live, and I'm hoping someone answers this before I say it, which is brown, which is shown in that picture as the wrong color. Never mind. Now, the live wire transmits the alternating current from the power source at 230 volts. The neutral wire completes the electrical circuit. It's neutral, so it's around 0 volts. And the earth wire provides safety by preventing the appliance from being electrically charged. So in a sense, if something becomes electrically live, the earth wire carries the electricity, that charge away, so that we don't get electrocuted. Now, why a live wire may be dangerous even when a switch is open. A live wire can still be dangerous even when the switch is open because it remains connected to the power source, carrying voltage. So contact with it can still cause an electric shock. Why is it dangerous if a live wire and earth wire are connected? A live wire and an earth wire must never be connected to each other, because if they do become connected to each other, what they do is complete the circuit. And this is because contact would create a complete circuit from the live circuit to the ground. And that may lead to electric shocks or fires. Although we do have safety mechanism in the house, so as soon as that does occur, our electricity switches off. And also we have fuses inside the plugs as well. And a fuse has a wire in the middle of it, and if for whatever reason the voltage or the current flowing through it increases beyond a certain amount, more than it should, the wire melts and it breaks the circuit. Okay, power. How do we calculate power? Power equals work done divided by time. So we've got a few here. We've got power equals work done divided by time, which means power also equals energy divided by time, because power is measured in watts, and watts are the same as joules per second. Watts. So work done, this W here, is measured in joules. Energy is measured in joules. Now another formula is power equals potential difference times the current. So power equals IV. Power also equals I squared R. Why does it equal I squared R? I'm just going to show you over here. Power equals IV. Now V equals IR. Now if I substitute V equaling IR, that means P equals I, and then what does V equal? IR. In other words, there's two I's there, so P equals I squared R. So I hope that makes sense. So now we've got another one here. Power equals I squared R. So you need to know all four. I know that you're going to be given a formula booklet, but you need to be familiar with every single formula so that it's at the tip of your tongue. It's at the tip of your hands, and you're writing and working away straight away. You're not spending loads of time just going through this revision book, this formula booklet, trying to find the correct formula. So energy transfers in appliances. The amount of energy an appliance transfers is determined by both its power rating and the duration for which it's switched on. So we can work this out using power equals energy transfer divided by time, or energy, if we rearrange this, we can use a triangle. Energy goes at the top and power and time go at the bottom. So if you want to find out how much energy is being transferred by a particular device, you can do using this formula as well. So let's look at an example. An item within a home intended to facilitate the transfer. What are domestic electrical appliances? So what are domestic electrical appliances? An item within the home intended to facilitate the transfer of energy. So all of these devices need energy to work. So a toaster has a power rating of 800 watts. So we know P equals 800. And the toaster's used for 1 minute. So time equals 1 minute, which equals 60 seconds. And it's asked us to find out the energy transferred. So now we've got E, P, and T. If you forget what connects these three symbols, these parts of the equation, then what you can do is look at E, P, and T and see if it's in your formula booklet. But we already know that energy equals power times time. So we're going to do 800 times 60. And our answer is going to be 48,000 joules. Now here's another example. I'll leave that for you to, so if you're watching this live, or if you're watching the recording of this, you can pause it and have a quick look through. What is electrical work? So now, if you consider the equations that we've discussed so far, then power equals, so let's write all these out again. So let's understand them. We've got power equals IV. We've got power equals I squared R. And we explained why, because V equals IR. So if we replace the V with IR, we end up with IIR. And we've got power, energy transferred equals power times time. And here's a new one. Energy transferred also equals charge Q times the potential difference voltage. And we've also covered Q equals IT. So here we've got a question. Calculate the energy transferred when the charge flow is 30 coulombs. So the first thing we know here is we've got Q equals 30 coulombs. And we have a potential difference of 2 kilovolts, which is, we're going to times that by 1,000, to get to 2000 volts. Now we've just covered energy transferred equals QV. So all we're doing here is multiplying the two numbers, which is 30 times 2000. So we end up with 60,000 joules. Okay, I'll just leave this question, so if you're watching live or if you're watching the recording, then you can pause and quickly go through the answer yourself. Now, this is a slightly more trickier question with multiple steps, which is why it's a 6-mark question. So when the charger is connected to the battery, the potential difference is 15 volts. So let's write down all the information we've got first. So we know voltage is, the potential difference is 15 volts. We know the total energy stored is 0.81 megajoules. Mega means times 10 to the power of 6. So that means we times that by 1 million. So that's 810,000. 810,000 joules. And the next bit of information we've got is the average current, I, equals 3 amps. And it wants us to calculate the time taken to fully charge the battery. Now, looking at that initially, there's nothing that connects all three automatically. So think of what you can work out. So one thing we can calculate here is Q. It's because we know that E equals QV, which means Q equals energy divided by voltage. So that's 810,000 divided by 15. Now that we've got Q, so let's put Q here. So I've taken this in a slightly different direction to what the my way works as well, but I'm going to show you what the way the actual. So with this question, there's more than one way of answering it. So I'm going to show you one way now. So I'm going to very quickly work out 810,000. So 810,000 divided by 15 equals 54,000. So we now know that Q equals 54,000. Now we can use a second formula. Q equals IT. So the fact that Q equals IT, we can now do T equals Q 54,000 divided by I, which is 3. So 54,000 divided by 3 equals 18,000 seconds. Okay, so I'm just going to rub all of this out. So the way I did this was using this formula, E equals QV. I got Q and I did Q equals IT. Now, in this mark scheme, they did it slightly different. Both are, it doesn't matter how you get to the answer, a mark scheme will always have the different ways. Now, this particular mark scheme did it slightly different. What it did was, it looked at the I and the V. So it said power equals IV. So power equals, I is 3 and voltage is 15. So 3 times 15. which means power equals 45 watts. Now that we've got power, power equals energy divided by time. So if you rearrange this, what does time equal? Energy divided by power. So in other words, we're doing 810,000 divided by 45. And in the end, you still end up with 18,000. So either way is fine. I'm just going to erase my writing so you can see the mark scheme answer. Now, this is only showing one part of the mark scheme. This kind of question would have had more than one section, one part to it. Okay, so what's the relationship? What is the relationship between power ratings and changes in stored energy? A device that has a higher power rating will convert stored energy into other forms at a quicker pace. Okay, the national grid. So once we finish the section of national grid, the remaining part of it, of this is triple, which we'll look at tomorrow. Now, electricity is conveyed from power stations to consumers through the national grid. A network of cables and transformers that connect power stations to consumers. So what a transformer does, and this is something that will come in a different topic, this will come up in the paper 2 topic in more detail, is it increases, a step-up transformer increases the voltage. Now when you increase the voltage of something, you automatically, so a transformer, a step-up transformer increases the voltage. And as soon as you increase the voltage, you decrease the current. Now a step-down transformer does the opposite. It decreases the voltage and it increases the current. So when we're transporting electricity to someone's home, so initially if this is the power station, it will increase the voltage using a step-up transformer. At a higher voltage, it travels faster, and because it's got lower current, it loses less energy. Now the step-down transformer here reduces the voltage, and then that safely reaches people's houses, because when the step-up transformer takes it up to 400,000 volts, 400,000, so 400 kilovolts, whereas in our homes we only want it to be 230 volts. So 400,000 volts would melt everything. So step-up transformers are used at power stations to increase the potential difference voltage before electricity is sent through the transmission cables. When the voltage goes up, the current goes down. Lower current means less heat is lost to the surroundings through the cables. This makes the energy transfer through the national grid much more efficient, because that means less is wasted. So the pros and cons of overhead transmission cables and underground transmission cables. Overhead are easy to maintain and repair. They're cheaper. The cables are cooled by the air. Greater risk of electric shock. Greater visual pollution. But it's a hazard to aircraft. The underground ones are hard to maintain. They're more expensive. They need cooling systems. But there's a lower risk of electrocution. They cannot be seen. There's no hazard to aircraft. Now this is what we've already covered, but let's quickly discuss it. Describe how the step-up transformer enhances the efficiency of the national grid. So it decreases the current. So that means lower current means less heat energy is lost, so less wasted energy. It reduces energy loss. So the step-up transformer increases voltage and reduces current for efficient transmission. The transformer lowers the voltage before supplying it to residences. What's the purpose of this process? So why reduce the voltage? Safety, primarily, because lowering the voltage reduces the risk of electrical hazards. And efficient usage. Household appliances are designed for lower voltages. So our household appliances don't need 400,000 volts going through them. So it's just better usage to lower it down. Okay, 2.5 onwards is now triple award. Now, I am going to cover this tomorrow. I'm going to start covering it at 9:30. So if anyone, it will be recorded for those that miss it. But for those who are doing separate science, I will be covering this at 9:30 tomorrow morning. The time is supposed to be 9 o'clock till 9:45, but I'll only need about 15 minutes to cover this. So I'll be covering static charge, and I'll be covering electric fields. And it should take me around 15 minutes, 20 minutes, and then I'll answer questions. There's one question that I've not been able to answer. So, Kiruba, I will answer your question tomorrow morning, hopefully. Okay, thank you everyone for joining us live. Hopefully have a lovely evening. Hope you see you all tomorrow. Welcome back to day 3 of the AMALathon. We're now covering Edexcel content. Today in biology, we are hopefully going to finish off topic 2, which is, and we're going to continue from stem cells. So like I've said before, if you have any questions, ask the questions in the live chat. I'll answer what I can, and if I'm unable to, then I'll answer it in our time, which has been put there specifically for questions and answers. 4:30 to 4:45. So we'll be doing 1 hour biology, 1 hour chemistry, 1 hour in physics. So specification point 2.6. Explain the importance of cell differentiation in the development of specialized cells. So stem cells which are undifferentiated divide by mitosis and differentiate to become specialized cells. So this is something which I've already touched on and covered yesterday, but what we're going to do now is explain it in a bit more detail. So it's something which I've already touched on. So how do stem cells become specialized? Cell differentiation is crucial for the formation of specialized cells, which allows multicellular organisms to have complex structures and functions. So your stem cell, animals and plants contain stem cells. Stem cells are cells which are undifferentiated, which means they have no specific job other than to multiply. And how do they multiply? And this is a key point here that will come up in tests. They multiply due to mitosis. Stem cells multiply by mitosis, and when they do multiply, specific genes in the stem cell are turned on or off. Now I'm just going to make this quick point now, but this is something that triple students need to understand, not combined, but it links, so I'm just going to quickly explain it. Each separate part of the DNA is a gene. So every single cell has the genes to make every single protein within the body. Now, for an undifferentiated stem cell to become specialized, one of those genes has to turn off, some have to turn off, so that those particular proteins are made. Now, how do stem cells become specialized? For example, stem cell could become a nerve cell, it could become a red blood cell, it could become a muscle cell. Okay, understanding how percentile charts are really important. In fact, a question on this came up last year in the 2024 paper. So everything I'm about to go through, my advice, memorize it. If it comes up again, they are really straightforward questions. So what are percentile charts? Percentile charts are a tool used to monitor growth by comparing an individual's measurements such as weight, height, and head circumference to a reference population. So percentile charts are used to compare growth of an individual against a standard growth pattern. So what this means is, from birth, things like weight are checked, height are checked, and they're compared to the rest of the population. So if we look at this example here, if someone is on the 50th percentile, that means roughly 50% of the UK or the reference population, because this would be done in different countries, 50% of the reference population is smaller and 50% is larger. Now, if the baby is born in the 25th percentile, that means 75% of the population is taller and 25% will be shorter. Now, so babies' growth is measured frequently, especially in the first few years of life, to ensure they're developing within a healthy range. So if a baby's measurements deviate significantly from their previous percentile, for example, the drop from the 75th to the 10th, it may indicate a health concern prompting further investigation. So this is an answer to a question. If a child's measurements deviate or they change significantly from a percentile perspective, what does that indicate? It indicates a health concern. It indicates something that needs to be prompted further and needs to be looked into by a doctor. But for example, if a child remains on the 10th percentile since birth and they're still on the 10th percentile 2 or 3 years later, that's not a concern because they're on the trajectory they were supposed to be on. So this is what percentile chart looks like. And let's understand how to use this percentile range, percentile chart. So a boy is 10 years old. And the boy's height is 140 centimeters. So the first thing we're looking at here is that the boy is 10 years old and he's 140 centimeters. So that means it falls right there. So he falls between this line and this line. So he is somewhere in between the 50th and the 75th percentile. So as long as he's, if now this 10-year-old child comes back 2 years later and is still in between those two percentiles, then we know that child's growing in a healthy manner. So I'm just going to rub this all out so you can see it's already been done. So the child is in between the 50th and the 75th percentile line. Now, let's say this child suddenly drops from that 50th to 70th and over the next 2 years is still the same height. So the child is here. Now the percentile is on the 25th. Now that would be a concern. So if a doctor sees that the child's gone from between 50 and 75 to the 25th percentile line, then that would be a cause for concern. That would be an issue, and they would investigate what's happening. Now, how percentile charts used? Percentile charts are a tool used to monitor growth by comparing an individual's measurements such as weight, height, and head circumference, and they're comparing it to the reference population. So this is a past paper question. So why do we use them? Why do we use percentile charts? Because it helps us monitor growth and to compare an individual's measurement to a reference population. Now that was only a 1-mark question. And as it was only a 1-mark question, you have to write one of those things. Describe, there's 3 types of stem cells: meristems, embryonic stem cells, and adult stem cells. And meristems are only found in plants, but embryonic stem cells and adult stem cells are found in animal cells. So they all have differences, and you need to know the differences between the 3, because all 3 can come in an exam, and you can get a comparative style question as well. So an embryonic stem cell, as we're going to get to, an embryonic stem cell is formed when the egg cell is fertilized by a sperm cell, and that initial cell is called a stem cell. Now that stem cell's only job is to duplicate, and it will keep duplicating till it gets to around the 8-cell phase, and after that it will start to specialize. And how does it duplicate? Via the process of mitosis. So embryonic stem cells, like I just said, are produced when the egg cell is fertilized by the sperm cell. Now, embryonic stem cells become specialized when certain genes are switched off or on. That's more for triple award. Now, most cells in a human embryo become specialized after the 8-cell stage. So once we've got to around 8 cells, a lot of those cells start to specialize, meaning the stem cells start to become nerve cells, muscle cells, red blood cells, epithelial cells. I think this one is the muscle cell, and this is cardiac cells, using cardiac tissue. And once the thing with embryonic cells, however, is that once they have specialized, that's it, it's permanent. They have permanently specialized. And alongside that, some cells remain. These are known as adult stem cells. And the important thing to know is the difference between the two. So embryonic stem cells can become any cell in the body. Once the genes are turned on or off, then they're permanently that cell. Meaning that one cell will only divide through mitosis and become another one of those cells, another one of those cells. A bone cell can't suddenly become a muscle cell because there's a need for muscle in a particular area. So once it's specialized, it has specialized. Now adult stem cells and also embryonic stem cells are only around at that time around conception, so around the time when egg cells are fertilized by the sperm cell. Now adult stem cells are found in the bone marrow and they are within us, but the issue is that they only become a limited number of cells. So they can become red blood cells, different types of white blood cells, platelets. They're not as versatile. But an embryonic stem cell is versatile, but once it has specialized, it has specialized. It won't specialize again. Adult stem cells are unspecialized cells. They're found in specific areas of the body, including the bone marrow, skin, liver, and brain. They are stem cells that can only differentiate into a limited number of related cells. Now, meristem is the stem cell that you find in plants, and specifically we find them in the roots and the shoots. Now, meristems are versatile. Like embryonic stem cells, they are very, very versatile, but the difference is they are versatile for the whole life cycle of that plant. So only meristems in plants can undergo mitosis, so they still divide through mitosis, and the meristem produce unspecialized cells that can develop into any kind of plant cell. So the meristem is a type of stem cell that can become any type of plant cell, and we find in the roots and the shoots. That means these shoots potentially could turn into roots, and these roots could potentially turn into shoots. That's if we were to turn it upside down in the soil. Now, what could we use meristems for? Stem cells from meristems in plants can be used to produce clones of plants quickly and economically. So, if there's a particular trait within a plant, you can clone it using the meristem, and what that means is you've made more of that exactly same. More of those plants are exactly the same. So it means we could clone rare species to protect them from extinction, and we could take crop plants that, for example, have become disease resistant and just clone lots and lots of them. That would increase the farmer's revenue. It would reduce the amount of crops that are wasted. Now, the embryonic stem cell is found in early embryos. They're highly versatile, and they can develop into almost any cell type in the body. That's what we mean by highly versatile. And this ability makes them valuable for regenerative medicine and research. For example, for people that have got diabetes, they've got illnesses that could be reversed if you were to treat the damaged cells or replace the damaged cells. Now, adult stem cells are found in specific body tissues, such as bone marrow. So we find it in the bone marrow. But the issue is they can only differentiate into certain cell types, so they're not as versatile. So they can't develop into almost any cell type in the body. They can only develop into a few. They are less versatile than embryonic stem cells, but they're still essential for tissue repair and regeneration. So meristems in plants are located in regions called meristems. They're found at the tips of the shoots and the roots. Plant meristems allow for continuous growth and enable plants to regenerate parts. So the meristems produce unspecialized cells that can develop into any kind of plant cell. All stem cells multiply and divide due to mitosis. Now this is a past paper question. Describe the function of meristem in in the growth of a plant. So this is what we've just learned. And from a mark scheme perspective, any two from the following. Meristem stem cells are undifferentiated, but you can write unspecialized. And meristem cells divide to produce more cells, so they divide. And they divide due to mitosis. So we've covered all 3 points there. How do stem cells help animals to grow? Stem cells can divide by mitosis, and then they can differentiate to form lots of different specialized cells. So your answer should have included, this is the actual mark scheme, stem cells divide by mitosis, so that's what we've written initially. And stem cells are undifferentiated, and then they can differentiate to form any type of specialized cell. So we covered the first two and the last bullet point there. Okay, 2.9. Discuss the potential benefits and risks associated with the use of stem cells in medicine. So potential medical applications of stem cells and the potential benefits and risks of using stem cells. That's what we're going to be looking at now. Stem cells have the potential to treat a variety of different diseases and conditions. So stem cells could treat diseases like type 1 diabetes, multiple sclerosis, which is a disorder that starts to break down the nerve cells. Spinal cord injuries. Now that's a big one because spinal cord injuries are usually permanent. Also, they're in areas which are very sensitive. So if more damage is caused during the operation, it could cause further problems. Parkinson's, blood cancers. So stem cells could treat these things. Therapeutic cloning. So when an embryo is produced with the same genes as the patient. So essentially what we're doing here at this point is when you have your small group of cells that's going to make an individual, it is taking one of those cells. Now this is the point when they're still stem cells. So taking one of those stem cells and putting it to one side, or allowing those stem cells to multiply, and then when the rest of these cells become a baby, it could be when that baby's a lot older, the baby has an issue, for example, with the kidneys. And if they have the issue with the kidneys, what they could use that stem cell for is grow another kidney and replace the damaged kidney with a new one. Now, the embryo is full of stem cells which are genetically identical to the patient. So all it means is taking one of those stem cells. But the issue with taking one of those stem cells is it could cause a miscarriage. So it's not an easy thing to do. The embryonic stem cell can be used to provide the patient with new organs, like I've just explained. And the new organ will be genetically identical to the patient, so it won't be rejected. So how does therapeutic cloning work? A nucleus is taken from one of the patient's body cells. The nucleus is transferred into an egg cell that had its own nucleus removed. So we've got an egg cell, we remove that middle part. So I've explained to you one way it can be done. Here we remove that central part. We go to, so this is the egg cell. Whoops. So let's start with the body cell. So we've got, this is the body cell. We remove that nucleus. So now we've taken the nucleus out. From the egg cell, I'll do that on this side, we've removed the nucleus. So now there's nothing in the central, there's still cytoplasm. The egg cell is stimulated. Now that nucleus there, this one, is added inside the egg cell. So that's been added there. The egg cell is then stimulated, so it starts to divide and form an embryo. After a few days, stem cells are extracted from the embryo. These stem cells can then be used to grow tissues or organs that match the patient. Now, there are some ethical concerns here, and the ethical issues are that these stem cells could potentially grow to become another human being. Even if it is a clone of the person, they could potentially become a human being. They can't speak for themselves, so there are certain religious groups that would consider this to be unethical, because that embryo, they consider it to be a living thing. So with some religions, they believe the soul comes into the body at a later date. Some religions believe it's as soon as the egg cell and sperm cell combine. So those people that believe the child is living, or is the baby at that point, for them, for those religions, they would consider this to be unethical, because those stem cells won't necessarily be given the opportunity to grow. Unless they put it into a womb, it's not going to grow. These are cells which are being developed outside of the uterus, just so the stem cells can be taken and grown into an organ that matches the patient's. So there are some advantages and disadvantages of therapeutic cloning. Now, the organs come from the patient's own stem cells. That means they won't be rejected. The stem cells could be used to treat different conditions such as paralysis and diabetes, so it's really, really good in that respect. Now, there's a risk of viral infection, because the cells that have been used could have a virus, and sometimes viruses, and this is something that I'll cover with triple, they can lay dormant within a cell. This is what I've already explained, so a bit of ethical or religious objections, because to access the stem cells, an embryo must be destroyed, and that embryo could eventually grow into a baby. So stem cells with the properties of embryonic stem cells can be produced from a patient's own skin cells. Discuss the advantages of using these stem cells to treat the patient. So what we're saying here is stem cells, so these are adult stem cells that have come from a patient's own skin, can be made with embryonic stem cell properties, as in, what can an embryonic stem cell do? It can become any cell in the body. So this is what they're trying to take out of here. So what are the advantages of using stem cells to treat the patient? So embryonic stem cells, so essentially what you're writing here is the fact that if it's come from your own skin cells, then genetically it's going to be similar. The DNA is going to be the same. If it's the same as the embryonic stem cell, it's going to be versatile. It can be used to become any cell in the body. And are we destroying an embryo here? No, because it's come from a person's own skin cells. So embryonic stem cells. So first, when we talk about advantages, first talk about embryonic stem cell advantages. So questions like this, look at what it's actually saying. Break open the question. So embryonic stem cells can differentiate or specialize to form any type of cell. Advantages of using embryonic stem cells that have been made from skin cells include, it's the patient's own stem cell. So it's genetically the same, so it will not be rejected by the patient's body. As these stem cells come from the patient's own skin cells, they won't be rejected. Some people have ethical or religious objections because to access stem cells, an embryo must be destroyed. In this case, you're not having to destroy an embryo, so there's no ethical issues here. And as an embryo does not need to be destroyed, as the embryonic stem cells have come from the patient's own skin cells. So the key here is looking at the key facts. Now, if you've memorized these advantages, if you've memorized therapeutic cloning, you've understood the advantages and disadvantages, then this kind of question is easy to open apart. You're looking at embryonic stem cells and looking at the advantages. Your stem cells have come from your own skin cells, so again, you're looking at the advantages and you're comparing to the usual disadvantages. Now remember, if we want to succeed in our exams, then number one, we've got to activate. In other words, we need to understand. So let me just quickly answer this question. Therapeutic cloning in Edexcel is part of the combined syllabus. So the first thing we've got to be able to do is we have to activate. In other words, we've got to understand everything. If we understand everything, then in questions like this, we can understand that skin cells that have turned into stem cells are genetically identical and it's different to embryonic. We've got to memorize key information. Like we need to answer this question, we needed to have memorized the advantages and disadvantages. And thirdly, we've got to apply what we've learned. The more application we do to past papers, the better we understand what the examiner wants from us. The better we understand, the more then we can test our understanding. So we can test our understanding, and we can understand what needs to be memorized as well. So all three steps: activate, memorize, apply. All of these equal learning. This is what takes us to grade 9s. And just to give you an example, students that I've taught in the past that have followed this simple process, they're the ones that have gone from grade 2s and 3s and reached up to grade 9. I've seen this every single year. The ones that follow this process. So a few years ago, or even last year, I had a student that came to me every morning. In fact, I had a group of students. Every year I always have a group of students that follow this process to the letter. And they came to me every morning with everything they didn't understand. So I helped them activate. I helped them understand everything they needed to understand. When they'd go home and revise, if they didn't understand something, they'd come back and we would activate. We would understand everything. And alongside that, I would make it clear to them what they should start memorizing. And then at home, they start, and this is something that can be done in the last 4 weeks, just before an exam, because we've got 4 weeks left. So we're now doing the activate phase. While we're going through this, I'm telling you which bits to memorize. And in terms of application, as soon as this week's finished and the AMALathon's finished, my advice is to do 2 biology papers a week, 2 chemistry papers a week, and 2 physics papers a week. That way in 4 weeks, you'll have covered 7 or 8 papers before the biology exam. And that will get you ready for all the paper 1 content. So if you do 2 biology paper 1s, chemistry paper 1s, and physics paper 1s across each week, that's 1 paper per day. That's 1 day where you're checking your understanding and you're checking your memorization. Now, if something comes up in one of those papers that you haven't memorized, then you know you need to memorize it. You do that on a daily basis, you'll be ready. So it works out to be 1 paper per day. Now, if you're doing combined, that's only a 60-minute test. And for those doing triple, that's only 1 hour, 100 minutes, 1 hour, 40 minutes. So this is the way to get ourselves to a grade 9. It is still possible. We've got 4 weeks. We can still get ourselves to a grade 9. Okay, now this is triple, so we're just going to skip through this. I did go through this yesterday at 4:30. So if anyone missed yesterday's session and their triple award, if you go to day 2, if you go to the AMALathon day 2, it's got all the timestamps there, so you can go straight to triple content as soon as you're ready to. Now we've got 2.13 and 2.14 to go in topic 2, and then we're going to move on to topic 3. I hope what I've just said makes sense. It's a 4-week plan. Everyone can get themselves to a grade 9. Everyone that's ready to put the time and effort in can get themselves to 2 grade 9s in science. So I'm going to quickly recap that. This, in part of the AMALathon, we are activating the brain. I'm telling you what to memorize. Now what you've got to do is start applying all of that learning to your past paper practice. Try and do 1 exam paper a day. So 6 papers a week. Across 4 weeks, you'll have gone through all the papers which are available from 2018 onwards. So let's have a look at the nervous system now. What is the function of the nervous system? So the nervous system enables humans to react to their surroundings and to coordinate their behavior. The structures of the nervous system leads to fast, short-lasting responses to a stimuli. So as soon as we react to something, the brain sends the message, or our spinal cord, which has a relay neuron that sends a message straight to our muscles contract, and we react to it straight away. So how is the information actually passed on? So number 1, we have a stimulus. A stimulus is a change in the environment. It could be light, it could be smell, smells a chemical. It could be a taste, which is also chemical. It could be that we touch something hot, we touch something sharp. So all this is a stimulus. We hear something. Now, in our eyes, we have receptor cells. So those that are doing triple award is what we covered yesterday, cone and rod cells. They're the receptor cells. In our ears, we've got receptor cells. In our nose, for chemicals, we've got receptor cells. In our tongue, on our skin. So we've got receptor cells everywhere that detect that stimulus. They then send a message through the sensory neuron. So the receptor sends a message through the sensory neuron or neuron to the coordinator, which we call the CNS. And the coordinator is made up of either the brain or the spinal cord. Now, if it's not an emergency and it's nothing dangerous, it goes through the brain only. If it's something we've got to react to fast, then it goes through the spinal cord as well. And that helps protect us from any harm, any danger. So from the coordinator, it then goes to the effectors. Now, how does it get from the coordinator to the effectors? It goes through the motor neuron. The effector is either a muscle or a gland. So the muscle either contracts or the gland releases a hormone. And then we have a response. And that is how our nervous system reacts to everything. So what is a stimuli or stimulus and receptors? So stimulus is, as I just said, a change in our surroundings or environment. And the stimulus is detected by receptors. Receptor cells detect a change in our surroundings and then they send a message to our CNS, central nervous system. So here's our stimulus. We've seen an apple. So the light from the apple reaches our eyes. And once it's reached our eyes act as a receptor. They send a message to the brain. The brain then sends a message through the motor neuron. So it goes through the sensory neuron to the brain, from the brain to the motor neuron. And then our muscle, the effectors, contract and we grab hold of the apple. Now, if it's something that's causing us danger, it goes to the spinal cord instead, where we have a reflex, like a relay neuron. Otherwise, the process is the same. Now, 2.14 will go through that in more detail. So, if there's no danger, the receptor sends the message straight to the brain. If there is danger, it sends the electrical impulse to the spinal cord, which has a relay neuron inside it. So let's go through a few examples. So this man has seen something he likes. I want to taste. So his nose has smelt the flavor, or he's seen the food with his eyes. They sent a message to the brain. The brain's made a decision. We're going to walk. So the brain sends a message to the legs and the person starts walking. And they walk to the food and they eat it. So in this case, the receptor is the nose. The stimulus is the chemical change. It's the smell. The receptor sends a message through the sensory neuron to the brain. The brain makes a decision. I want to taste. And it sends a message to the legs. And the person starts walking to the food. Let's see another example. The receptor is the skin. The stimulus is the heat. So the person touches the cup. It sends a message to the sensory neuron to the brain. The tea's at the right temperature to drink. It won't burn me. So it sends another message. So the brain has made a decision now. It's not hot. I'm going to drink it. So the brain sends a message through the motor neuron back to the arm. And the person has a drink. Example number 3. Receptor, the ear, the stimulus is sound. So a person has pressed a doorbell or something's not loud enough. They can't hear it. So a message goes to the brain. The brain makes the decision that I want it to be louder. So they send... So the person is trying to hear a sound. maybe from the radio, from TV, from anything. They can't hear it very well. So a message goes from the ear to the brain. Can't really hear this very well. So the brain now sends a message through the motor neuron to the hand, and the person's hand touches the button where the volume is and increases the volume. Okay, there's lots of examples here. So you can have a look at these. So if those are watching live, afterwards you can look through this and you can look at each example one at a time. And it's the same process every single time. We have a stimulus, which is detected by the receptor. How does the receptor send the message to the brain? Through the sensory neuron. Then it goes to the brain, which is the central nervous system. The brain makes a decision. It sends a message to the effector muscle. In this case, this guy's arms, because he wants to grab hold of the camera to take a picture. And then the muscles contract and we have our response. So I won't go through any more examples, but I will just show you this example so you can have a look at it in your own time. When someone reacts to a stimulus, messages from the brain are sent to their muscles. Describe how messages are sent from the brain to the muscles. So this is something we've already covered. How does the brain send messages to the muscles? It sends it through the motor neurons. So this is just a past paper practice question. It sends electrical impulses along the motor neuron. Now there's a really good question here. It says, will the paper 2 revision also be on YouTube Live? It will be on YouTube, but it will be based on membership. So if anyone wants to join the AMALathon 2, we'll have pre-exam workshops prior to every single exam, so the weekend before each exam. And we'll also have another 1 week session like the one we're doing at the moment, like an AMALathon, where we'll spend 18 hours going through all the paper 2 content. There will be a small cost, so if you want to join our WhatsApp channel or you want to register using the link below, both of them will make it, both ways. That way we'll have your contact details and we'll be able to contact you close to the time when we start taking payments. So let me just repeat that. We'll have AMALathon 2, and AMALathon 2 will start in May. We'll have pre-exam workshops, so that's 3 hours before each exam, that's 18 hours of revision. And we'll also have an 18-hour AMALathon, similar to the one we're doing now in the May half-term holidays, where across 18 hours I will cover all of the paper 2 content. So that's 36 hours of learning, and I believe, you'll have to check with the admin staff, but I believe the price is £108 if you register yourself right now during the 6 days AMALathon. Otherwise the price is not that much more, but it'll be slightly more. So this is what the nerve actually looks like. It's got myelin sheath, which speeds up the transmission of the impulse. So it acts as an insulator. And what do we mean by that? It keeps the electricity in. So the electrical impulse is kept inside with the myelin sheath. It keeps it insulated. Similar to a wire. If you look at the plastic, the wires that we've got, they've got the rubber plastic on the outside. Inside is the metal wire. Why do we have the outer sheath? Or why do we have this outside rubber plastic part? It's to keep the electricity in, it's to keep it insulated. Now the dendrites are the branched ends which receive electrical impulses. The dendrons carry electrical impulses from the receptor towards the cell body. Now it's not common, but the questions on this do come up, so it is important to know. And you've got the axon. The axon is the actual, what we just call the metal wire. And it's not made of metal, but it's similar to a metal wire. It carries the electrical impulse through the body. So in this question, you've been asked to add an arrow to the diagram above to show the direction of travel of an electrical impulse. So the electrical impulse always travels towards the dendrites. This means the electrical impulses move from left to right. So the electrical impulse would go that way. And then it says name both structure K and L. So K is the myelin sheath and L is the axon. Now, this is something that's really important. and it comes up in lots of exams. This is particularly comes up in 6-mark questions. Now a synapse is a gap. So I'm going to draw my own synapse here. And what the synapse does is, synapse is a gap between two neurons, two nerve cells. So this is nerve cell number 1 and this is nerve cell number 2. When an electrical impulse arrives at nerve cell number 1, these chemicals, and you can see them here, they're called neurotransmitters. They are released and they diffuse across the gap. And when they reach the other side, if you look really carefully, you've got these orange bits here. Those are receptors. Now, when the neurotransmitter fits into the receptor, it causes the electrical impulse to continue. Now, what this does is it ensures the electrical impulse is only traveling in one direction, because only one side is releasing the neurotransmitter and only one side has receptors that take in the neurotransmitter. So when an impulse reaches the end of a neuron, a chemical neurotransmitter is released across the gap. The chemical then diffuses across the synapse, and that ensures that it only goes in one direction. This needs to be memorized. It comes up nearly every single year. But you'll notice that as soon as you do past paper practice. That's the question here. Explain how impulses are transmitted at synapses. So the synapse is a gap between neurons. When an impulse reaches the end of a neuron, a chemical neurotransmitter is released across the gap. The chemical then diffuses across the synapse. When the chemical reaches the next neuron in sufficient quantities, this starts another impulse. So that is your ideal answer. And this was an actual past paper question. Explain how impulses are transmitted at synapses. And this is the mark scheme. This is why past paper practice, this is why the application phase is really important, alongside the activate phase and the memorize phase. If you activate, memorize, apply, you can still get yourself to a grade 9. There's still 4 weeks plus left till your exam. I think it's 5 weeks left till your exam. It's still enough time. Now the synapse is a gap between neurons. Impulse stimulates the release of chemicals, neurotransmitter, and the neurotransmitter or the chemical diffuses across the gap, the synapse, and it stimulates an impulse in the next neuron. 2.14. Now, the reflex arc is important, and we need to be able to describe what happens during a reflex arc. Now, why are they important? Because it doesn't go through the brain, it goes through the spinal cord as well. When something goes through the brain, we take a split second to think about it. It still takes, for example, when we react to things, it still takes 0.3 seconds to react to something. But a reflex arc is even faster because there's no thought process there. It means it allows us to have a reflex towards something. So it means it allows us to react fast. It allows the response to be automatic, and it protects us from harm. So it means if something, for example, someone throws something and it's coming towards us and we haven't realized, and the second we see it, we can move our head out of the way. Now, if someone threw something towards us accidentally and it's coming towards us, if it went through our brain first, our brain would look at it and it would work out, is it dangerous? Is it not dangerous? Do I need to do something? Do I not need to do something? There'd be such a thought process that by the time the brain has made a decision, which might be 0.3 seconds, but that's enough time for that projectile or thing that's been thrown to hit someone. Whereas if it goes through the spinal cord, then you have an automatic fast response. So here we've got a receptor. It detects the stimulus, the change in the environment. It sends a message through the sensory neuron to the spinal cord. In the spinal cord, we have this, which is a relay neuron. And the relay neuron automatically then sends a message through to the motor neuron, which goes to the effector muscle, and we have a quick response. Now, if you look carefully here, I'll have to erase some of my blue pen. There's gaps between each of the nerves. So this makes sure, so those gaps are called synapses, and it makes sure that the nerve, the information is going in this direction. In one direction only. So that is your reflex arc process. So this is now on this slide is just recapping what I've just said. A stimulus is any change in our surroundings or environment. The stimulus is detected by receptors. Receptor detects a change and generates an electrical impulse. The impulse travels along the sensory neuron, and then it goes across the synapse and into the motor neuron, the relay neuron, and then it goes across the synapse and goes across the motor neuron to the effector muscle, at which point a person reacts. So this is just an example. Why do we move our foot away if we step on something sharp? So our foot has touched something sharp. So it sends a message through the sensory neuron to the spinal cord where the relay neuron is. The relay neuron sends a message through the motor neuron to the effector muscle. The muscle contracts and it moves the foot out of the way. Now if it had gone to the brain first, then the person would have stepped into the needle first. Okay, so this is a question and there's the answer. I'm going to leave this just for a couple of seconds. So afterwards, when you're revising using this, using the recording on YouTube, then you can pause here and have a go at the question and answer. And here is the 6-mark answer to the question. So by the end of the AMALathon, you'll have done 6 hours of biology with me, 6 hours of chemistry, 6 hours of physics. And that will be your activate phase done. So your remaining time, I would advise, is on memorization and application. Okay, that's the end of this PowerPoint. I'm just going to quickly load up topic 3. And then we're going to continue for just a little bit longer. Now, real big thank you to everyone that's been supporting us, that's subscribed and been sharing our videos, that liked our videos, because our AMALathon day 1 video has already gone past 1100 views, and our AMALathon day 2 videos already close to 700. So we really appreciate all the support that we've had. So based on where we are right now, we have completed key concepts in biology. So as soon as it's uploaded, and we finished cells and control as well, and we've now moving on to genetics. So this is complete. It's better I use a highlighter. That's complete. This is complete. Cells and control is complete. So we're in line with where we want it to be. And from a triple perspective, we've covered that content, we've covered this content. And today at 4:30, unless we have some questions and answers, I'll be moving straight on to B3 genetics, which is triple only. And those that are able to watch, fantastic. Those that are not able to watch, not a problem. You can watch the recording afterwards. Okay, so let's move on to genetics now. Now 3.1 is triple only. Now I am going to very quickly go through this. So those who are doing combined, if you can be patient for just a couple of minutes, it doesn't take long. This 3.1 and 3.2 really doesn't take long at all. So asexual reproduction just means that there's one parent. So like I said, this is triple only, but asexual reproduction means there's only one parent, meaning clones are made. So it only involves mitosis. There's no fusion of gametes. So I'm using the wrong pen here. There is no fusion of gametes. That's what they're showing here. There is no fusion of gametes. They're only clones. So it's a type of cell division that produces identical cells. Now the advantages are it's faster. Now there's loads of questions that come up on this that are always comparative, so it's important to remember this. Only one parent's needed. And that means it's faster because only one parent's needed. It means that one parent doesn't need to spend time trying to find that perfect partner to have a child with. And organisms are able to reproduce if separated. Meaning it doesn't require two, it only needs one. Now, the disadvantages is that there's no variation in the population. So if one gets ill, they all get ill. If the environment changes, this could put all organisms at risk. So if suddenly there's a new virus, a new bacterial cell, something that causes harm to one, it causes harm to all of them. So for example, banana plants, banana trees were at one point were heading towards becoming endangered, as in there were the concerns that they'd have to chop down a lot of trees, and in the end they had to, because banana trees are grown as clones. So they take a sapling, a cutting from one, and they continue to grow it, and that's because of consumerism. So people prefer a certain shade and a certain type of banana. So once that perfect banana was found, they just kept cloning it. And like I said, it's really quick. We're on 3.2b already. And this is biology only as well. So 3.3 will be for both, for everyone, combined and triple. But this is I just want to quickly go through this. What is sexual reproduction and what are its advantages and disadvantages? So here, animals use sperm cells and egg cells. Plants use pollen and egg cells, or an ovule. Now, sexual reproduction involves the joining of male and female gametes. So it's the egg cell and the sperm cell. And that makes your initial zygote or initial ball of stem cells that eventually becomes the embryo and then is born as a child. Gametes are produced through meiosis. So meiosis is something we're going to go through in a bit more detail. There should be an E just there. A type of cell division. So gametes are produced through meiosis. It's a type of cell division that results in genetically unique cells with half the number of chromosomes. In other words, they're haploid cells. So the egg cell, this part is valid for combined as well, so everyone can be listening to this. The egg cell is made through meiosis, which means the cell splits twice. So we end up with half the number of chromosomes in the egg cell and half the number of chromosomes in the sperm cell. And what do we call half the number of chromosomes? Haploid. So our body cells have got 46 chromosomes, but our gametes have 23 chromosomes. That's the egg cell and sperm cell. So when gametes fuse during fertilization, the genetic material mixes. And this restores the diploid chromosome number and leads to variation. So half chromosomes are here, half chromosomes are here, but from those 46, we don't know which 23 have gone into the gamete. From those 46 chromosomes in the male, we don't know which 23 have gone into the gamete. So what you end up at the end is something unique and different. It's a mixture of both. And that is what we call variation. So what are the advantages and disadvantages of sexual reproduction? So the advantages are that it produces variation in the offspring. Now a lot of this overlaps with topics that are coming up. So this, like I said, is okay for combined as well. Now if the environment changes, organisms with advantageous variations have a survival advantage by natural selection. And in that respect, it potentially helps evolution. Variation allows natural selection to work, driving evolution over time. Now, the disadvantage of sexual reproduction is that it is energy intensive. It is slower than asexual reproduction, meaning for the reproduction to occur, for example, in human beings, it takes 9 months. Whereas in bacterial cell, that just makes clones of itself, splits through a process similar to mitosis, it takes 20 minutes. So it's energy intensive, it takes a long time, it requires a lot of food and nutrients. And in other words, it's much, much slower. If an individual becomes isolated, they will be unable to reproduce. So if there's only one human being on an island, they can't reproduce. It's not always easy if individuals are isolated. So now let's look at the comparison between the two, and then we're moving into the combined bit. So asexual needs one parent, it uses mitosis, and there's no variation. But in sexual, it requires two parents. The sex cells are made through meiosis, and there's lots of variation. So now let's have a look at mitosis, and then compare it to so let's look at meiosis and then compare it to mitosis. So meiosis is, as we just said, a type of cell division that produces gametes, like sex cells called sperm and egg. So sex cells, for example, sperm cells and egg cells in animals. So meiosis, cells in reproductive organs divide by meiosis to make gametes. Now, we don't need to know this whole process. What we do need to know is the outcomes of this process. So if you look at this whole process, we start with one daughter cell. It splits here. So that's the first split. And then it splits again here. So it splits twice. We start with one parent cell and we finish with 4 daughter cells. So 1 becomes 4. We start with a diploid cell and we end up with a haploid cell. So we start with 46 chromosomes, a full number of chromosomes, and we end up with half. This causes, so we have, with our cells, daughter cell at the end, we have genetic variation. So let's do a quick comparison between the two. Mitosis, number of daughter cells produced, 2. Meiosis, 4. Number of chromosomes in each daughter cell, 46. How many in meiosis? 23. So let's compare the outcomes. Mitosis produces 2 diploid cells that are genetically identical. So here's the key points here. 2 diploid cells, they're genetically identical to the parent cell. And the daughter cells are genetically identical to the parent cell. Also, we've got 46 chromosomes and they're both haploid. And comparatively speaking, meiosis produces 4 haploid cells. Sorry, they're not haploid. Let me delete that. They're diploid. But that all is already written there, so I didn't need to say it again. The thing that's missing was just the 46 part. Here it's got 23 chromosomes, or half. Meiosis produces 4 haploid cells, daughter cells that are genetically different. Daughter cells are genetically different from each other. So let's have a quick look. Now, this is an actual past paper GCSE question, and these are the kind of answers that would have got you marks. Mitosis produces 2 cells, meiosis produces 4 cells. We mentioned that. Mitosis produces genetically identical, and meiosis produces genetically different cells. We've mentioned that. Mitosis produces haploid, mitosis produces diploid, and meiosis produces haploid. We would have got that mark too. Now we missed this one out. Mitosis produces body cells, but meiosis produces gametes and sex cells. Okay, we're going to stop there and we're going to take a 5-minute break. So we'll re-continue at 2:35. Okay, thank you everyone. See you in 5 minutes. Okay, welcome back everyone. Let's get started with the chemistry phase of day 3. So we're going to hopefully finish off all of topic 1 now. Now topic 1 is really important because topic 1 is going to come up in both paper 1 and it will come up in paper 2 as well. So it is important that you've got a good understanding of all of topic 1 for both papers. Now, what is the structure and bonding of graphene? Now, if you look at graphene is just like graphite, except it's just a single layer. So all the properties of graphene are similar to the properties of graphite. So you've got one atom. So you've got one atom that's bonded to 3 other carbon atoms, 1, 2, and 3. So if that carbon's bonded to 3 different atoms, it's using 1 electron in the covalent bond there, 1 electron in the covalent bond there, 1 electron in the covalent bond there. Now remember, carbon is in group 4, so it's got 4 electrons. So if you've got 1 here, 1 here, and 1 here, there's 1 delocalized electron. That 1 delocalized electron means graphene can conduct electricity. There's loads and loads and loads of covalent bonds here, which means that the graphene can conduct electricity. And the fact that graphene conducts, that means graphene conducts electricity. Now you've got lots and lots of covalent bonds, and that means the covalent bonds make it mean that it's got a very high melting and boiling point because you provide enough energy to break all those covalent bonds. So fullerenes are molecules made of carbon atoms arranged in hollow shapes such as spheres, tubes, or cages. Most made of carbon atoms bonded in hexagonal, sometimes pentagonal or heptagonal rings. So it makes these kind of shapes. This is called Buckminster fullerene. That's C60. And that's really good for, that is used in medicinal deliveries. So fullerenes are strong and lightweight because they're made of carbon. You don't need to know it from a nanotube perspective if you're doing combined. They're good conductors of electricity because they've got delocalized electrons. They provide a large surface area, which means they're good for drug delivery and useful for trapping other molecules. They're slippery and have low friction. So something like Buckminster fullerene can be used as a lubricant because it's kind of a ball shape. The balls roll over each other. Now the nanotube part, I'm going to move past because we don't need to know that for combined, and it is something that is covered in topic 5 for those people doing triple award. So when I cover topic 5 in more detail, that will automatically be covered as well. So explain why graphene will be a good conductor of an electrical current. So carbon has 4 outer shell electrons. So carbon's got 4 outer shell electrons, but only 3 of them get used up. So 1 gets used here, so 3 covalent bonds are made, but this 4th one is delocalized, it's free to carry electricity. So 3 electrons used in bonds, so 1 electron is free to move, it's delocalized, it can carry the charge, the electrical current. Now this is a 6-mark question, and this is a very common type of 6-mark question. Each year when they do ask this kind of, when it does come up, they might change the 3 different things they're looking at. But now consider these 3 substances, and what in this particular question we're considering diamond, graphene, and fullerene. Explain in terms of their structures, their bonding, their relative melting points, strengths, and abilities to conduct electricity. So you're doing a comparative between the 3. In other words, just answer those things that are underlined for each one. So diamond, but because it says explain, you've got to say why something happens. So you can't just say diamond can't conduct electricity, you've got to write why it can't conduct electricity. So diamond has a giant covalent structure with strong bonds, giving it a high melting point and hardness. Now, why has it got high melting point and hardness? I know it doesn't say it there, but I want everyone to be able to say this. Why has it got a high melting point and hardness? Because it's got lots and lots of covalent bonds. So to melt it, you've got to break all those covalent bonds. You have to provide enough energy to break the covalent bonds. Now, it does not conduct electricity because it has no free electrons. Now, graphene has a single layer of hexagonally arranged carbon atoms, giving it high strength and a high melting point. So in other words, it's got lots and lots of covalent bonds. Because it's got lots and lots of covalent bonds, it requires a lot of energy to break those bonds. It can conduct electricity, and this links to the structure here, because whereas in diamond, each diamond has 4 covalent bonds, in graphene, each carbon has 3 covalent bonds and 1 delocalized electron. And that delocalized electron can conduct the electricity. Then we've got fullerene. Fullerene has a molecular structure with weak intermolecular forces, resulting in a low melting point. It conducts electricity poorly because electrons are not free to move between the molecule. So these kind of questions, when you enter the application phase after we finish the AMALathon, then it's important you look at 6-mark questions like this. And these are the kind of 6-mark questions where you need to memorize the mark scheme. Okay, polythene. What are the properties of polymers? Now, really quickly, a polymer, this is a polymer in front of you, and it's made up of repeating units called monomers. So this is the repeating units here, monomer. Now this is more of the monomer polymer part is more of a triple topic, but to understand what I'm saying, you need to understand this. Now, if you've got a long chain of carbons, and this will come up in more detail when we look at paper 2, and we look at crude oil and the different fractions, and how the melting point and boiling point is bigger as the molecule becomes bigger. And that's because the larger the molecule, the more carbon atoms it has, and the more weak intermolecular bonds you have between the molecules. So this is one whole molecule there. That's another polymer with a big molecule, and between it you have weak intermolecular bonds. Now if I've only got a small molecule, I'm going to have less intermolecular bonds. So this is going to take less heat energy to break. This one will take more heat energy to break. So this will have a higher melting point, boiling point. This will have a lower melting point, boiling point. Properties of metals. So firstly, how do metallic bonds form? Now, metallic bonds form like this. So if you look over, let's go recap our ionic bonding. So let's say you've got sodium. It's got 1 electron in the outer shell. Now, when metallic bonds are formed, here you can see the electron in the outer shell. The electrons become free to move. So you end up with, now if that sodium loses that electron, what does it become? It becomes a positive ion. So now, we've got a positive ion, and in between the positive ions, you've got a sea of delocalized electrons. The electrons are free to move around, and it's the attraction between the positive ion and that sea of electrons that holds the metal together, holds the structure together. So it ends up forming a giant lattice. Now I've just received a really good question. Is graphene a simple covalent molecule? No. Graphene still comes under giant covalent. Because graphene is like graphite, except it's just one layer of it, which is still significantly big. Whereas usually you know the difference between a simple molecule and a large one when you can't add numbers to it. So for example, we just call diamond. We don't say C 1 billion. because there's so many atoms you can't really make a ratio. Whereas if you look at Buckminster fullerene, it's C60, so we know there's just 60 carbons there. When we say H2O, then we know there's 2 hydrogens, 1 oxygen. So, properties of metals. Now, pure metals form a giant lattice. That's regular layers of metal atoms. And they form strong metallic bonds. So there's positive ions held by a sea of delocalized electrons. So to melt it takes a high temperature because you've got to overcome that attraction. Now, they've got delocalized electrons. That means the delocalized electrons can move freely and they can conduct electricity and heat. And they are malleable and ductile, which means the layers slide over each other, making metal soft and easy to shape. So why does it have high melting and boiling point? Because the strong metallic bonds need lots of energy to break. Why can they conduct electricity and heat? Because they've got delocalized electrons that are free to move and carry that heat and charge. Malleable and ductile, because metals have layers, and layers of atoms slide, allowing metals to be bent and stretched. So if you look at the Rutherford experiment, why did Rutherford use gold, use a piece of metal? Because it's very easy to hammer it into a really thin sheet. So he used gold leaf. It was hammered so thin that it was only a few atoms thick. And it's strong and dense. That's because the closely packed ions make the metals hard and durable. Now, shiny and sonorous. You need to know them as properties, but you don't need to know why. The why part is not necessary. But for the rest of the why part is necessary. If we're aiming for grade 9s, or if we're aiming for a grade 5 at least, then we need to know this. Now, we've covered this already. Why are they good conductors of electricity? Because they've got free electrons. They've got delocalized electrons. Why? When can an ionic compound conduct electricity? Not when it's solid. Why can it not conduct electricity when it's solid? Because the ions are not free to move. But if the ionic compound is dissolved in water, then the ions are free to move and it can conduct electricity. What about non-metals? When they've got covalent bonds, can they conduct electricity? No, because the electrons are part of the covalent bond. They're not free to move around. Why can graphite conduct electricity? Because that's still non-metal, because it has one delocalized electron. And then we're back to metals. Why can metal conduct electricity? Because it has a delocalized free electron. So sodium, like all metals, conducts electricity. Why can it conduct? Explain how it can. Because it has delocalized electrons which can move throughout the metal. And look what it says here, reject ions. Because ions are only linked to ionic compounds, salts. Limitations of the model. So sometimes you can represent ionic compounds or compounds as two-dimensional, like this, as ball and stick, like this, or 3D. Now the issue with 2D is it only shows the flat view, not the full structure. So it could lead to a person assuming that atoms don't have dimension. The ball and stick, the atoms shown as too far apart, and they're not to scale. Bonds are sticks, but real bonds aren't rigid rods. So the bonds are shown by the sticks, but they're not rigid in real life. And there's no massive gaps between the atoms like that. And 3D, it's hard to see individual atoms clearly. And if you look at this really careful, I mean, it's complex and difficult to draw, but if you look at each of these atoms carefully, they look like they're next to each other. If you had all these positive ions next to each other, what would they do? They would repel. So it's a very difficult thing to draw, represented that way. Now this is what we've already covered. You need to know the properties of the metals, and you need to know the explanation as well. Okay. Now today's lesson was all about the mathematical side of things, and we finally made it there now. So we are now covering all the numeracy elements that will come in paper 1 and paper 2. When I cover topic 5 with triple award, then I will go through the triple award maths as well. That's all part of topic 5. So relative formula mass, RFM, is the total mass. So excuse me one second. So relative formula mass is the total mass of a compound calculated by adding up the relative atomic masses of all the atoms in the chemical formula. Now I'm going to explain what that means. Let's show it here first. So let's look at H2O. So if you've got your periodic table with you, you can do this much quicker, and you can even start answering the questions in the live chat if you want to. So H2O is made of hydrogen, and oxygen. What we're doing here when it says relative atomic mass, we are looking at the mass number. So if you look at hydrogen, it's got a mass number of 1. Oxygen has a mass number of 16. So what we're doing here is 1 plus 1 plus 16. So the relative formula mass of H2O is 18. 1 plus 1 plus 16. Let's look at the ones on this next slide. Let's look at CO2. So that means CO2 is C, O, and O. Now carbon has a mass number of 12. Oxygen has a mass number of 16. So what we're doing now is 12 plus 16 plus 16, which equals 44. We've done H2 already. Let's look at MgOH2. So MgOH2, another way of writing it would be this, Mg, and there's 2 OHs. So now magnesium is 24. So if you look on the periodic table, the mass number is 24. We already know O and H. So that means 24 plus oxygen and oxygen, which is 16 plus 16. Then we've got 2 hydrogens. So plus 1 plus 1. And what does that equal? 58. Empirical formula. So this is the ratio. The empirical formula of a compound is the simplest whole number ratio of atoms of each element in that compound. It does not show the actual number of atoms, just the simplest ratio. So we're looking at simplest ratio. For example, if I had propene is written as C3H6. Can we simplify that? Well, let's look at 3 and 6. If that was a ratio, 3 to 6, how would we simplify that? That would be simplified, we'd divide both by 3, so it becomes simplified to 1 to 2. And that's exactly what we're going to do here. 1 carbon, 2 hydrogens. It's just we don't need to put the 1 there. So if you've got C6H12O6, C6H12O6, then we've got a ratio of 6 to 12 to 6. So what divides in all numbers? 6. So 6 divided by 6 is 1. So we've got 1 carbon. 12 divided by 6 is 2. So we've got H2. Oxygen divided by, 6 oxygens divided by 6 is 1. So we've got 1 oxygen. So in the end, it would be CH2O. Now we can deduce the chemical formula, the empirical formula as well. So a compound contains 40% carbon and 6.7% hydrogen, 53.3% oxygen. Now when you do these questions, what you do is this. You take the percentage or the mass, whatever they give you, and you divide it by the MR, the mass number, the mass number of that individual element. So let's look at carbon first. Carbon, it's carbon 12, the mass number. So carbon, we're going to do 40 divided by 12. And 40 divided by 12 equals 3 1/3. Now let's look at hydrogen. Hydrogen has a mass number of 1. So we're going to do 6.7 divided by 1, which is 6.7. And let's finally look at oxygen. Oxygen has a mass number of 16. So oxygen, we're going to do 53.3 divided by 16. And that equals 3.3 as well. So what we've now got is carbon, which is 3.3, oxygen, which is around 6.6 or 6.7, and we've got, no, sorry, that's the middle one, should be hydrogen. So the middle one should be hydrogen, and then we've got oxygen again as 3.3. Now, if we divide each one of those numbers, again, we treat it like ratios. So 3.3 divided by 3.3 is 1. So we've got 1 carbon. 6.6 divided by 3.3 is 2. So we've got 2 hydrogens. 3.3 divided by 3.3. So oxygen is also 1 as well. So that's our ratio. 1 carbon, 2 hydrogens to 1 oxygen. CH2O. The top number came from the periodic table. So if I'm showing you oxygen, if you look at the periodic table, it will look like this, 16 and 8. So the top number is the mass number. So this is a quick experiment on how to deduce empirical formula. So how can the experiment be used to deduce the empirical formula? So what we can do is this. And it's shown really nicely in this diagram. You've got this crucible, that's a crucible. We've put magnesium inside it, and you can see the mass has increased. It's gone up by 12. Now we go to step 3, we heat it up, and because what we're saying here is that the mass of the magnesium was 12. Yeah, but we don't need to worry about that too much actually. Now, no, actually that's fine, 12, it was 12 grams. Now, we've burnt it, and after burning it, it's reacted with the oxygen, and so now it's made of white magnesium oxide powder, and now we're at 44.2 grams. So we've increased by a further 8 grams. So that means the magnesium weighs, how much does the magnesium weigh? 12 grams. We know that because it was, when it was empty, it weighed 24.2, and when we added magnesium, it was 36.2. Now at the end, we've increased it, we've gone from magnesium to magnesium oxide. So if we take away 36.2 from 44.2, we've got 8 grams. That represents oxygen. So now look, we've got the mass, we divide it by, now magnesium's top number is 24, its mass number, so 12 divided by 24, that equals 0.5. Now the 8 grams, which represents the oxygen, if you look at your periodic table, oxygen's got 16 on the top, 8 at the bottom, so 8 divided by 16, that also equals 0.5. So if we divide both by the smallest ratio, in other words, what's the ratio here? It's 1 to 1. And that shows us that when magnesium and oxygen react with each other, it makes MgO. So in this ratio, it's always 1 magnesium reacted with 1 oxygen. So they're explaining it here now. Weigh the empty crucible. The crucible container is placed on a balance, and its mass is recorded. So we've done that. We've recorded the mass of the crucible. That's this bit. we've got the mass of the crucible. We weigh the crucible with the metal inside. So that's this bit. We weighed it with the metal inside. Then we heat the metal in oxygen. So the crucible is heated with Bunsen burners to allow the metal to react with the oxygen in the air. Now that it's combined with oxygen, it's going to be heavier. Once the reaction is complete, the new mass of the crucible with the metal oxide is recorded. This way, we now know that magnesium weighs 12 grams. Its AR value, so its mass number is 24. Mass divided by its mass in grams divided by its mass number gives you 0.5. Oxygen weighed 8 grams. We're dividing it by its relative mass, its mass number 16. We also get half. That shows us we have a 1-to-1 ratio. That one magnesium reacts with one oxygen. So the overall formula is magnesium oxide. Now, which is the empirical form of butene? Now, butene is C4H8. So when it says what is the empirical formula of butene, this is the past paper exam question. The way we calculate this is we just need to put it in the simplest form. So think of it like a ratio. At the moment, it's a ratio of 4 carbons to 8 hydrogens. So how do we simplify that? Divide both by 4. So it simplifies to C1H2, which we just write as CH2. Now another question here. So let's do this one step at a time. Titanium ore was analysed. So we've got titanium and we've got oxygen. A titanium ore was analysed and found to contain 12 grams of titanium atoms. So first bit of information we've got here is we've got mass. So how much titanium do we have? 12 grams. How much oxygen do we have? 8 grams. Now we divide that by the relative atomic masses. So you can put MR or AR. M usually stands when it's a molecule, so really AR is what I should have written, but it's fine. Either way is fine. So what's the MR or the AR here? So it should be AR. Titanium is 48. So 12 divided by 48 equals 0.25. And with oxygen, it has an AR of 16. 8 divided by 16 equals 0.5. Now we're looking at these two numbers as ratios, 0.25 to the ratio of 0.5. So let's divide both by the smallest one. So we can divide both sides by 0.25. So 0.25 divided by 0.25 is 1. So that means we've got 1 titanium. And 0.5 divided by 0.25 is 2. So we've got 2 oxygen. So the overall formula will be TiO2. Okay, now I'll erase all this so you can see the mark scheme answer. Okay, so this should be, I'm just going to do some corrections in the actual question, but I'll leave this so people try it in their own time. So hydrocarbon X was cracked to form 1 molecule of hexane. So that's C6H14. The empirical formula of Y is CH2. And here the mark scheme goes through that in detail. Okay, conservation of mass. Large numbers in front of chemical symbols are multipliers. These numbers are subscript. You do not change these when balancing equations. So when you balance an equation, you can change the number in front, but you can't change the small numbers. So when it says these numbers are subscript, it's talking about these smaller numbers. These smaller ones. Okay, the subscript, those ones you can't change. Now, conservation of mass, what it means is that whatever the number of atoms you have on the left hand side, as in the reactants, you have the same amount in the products. So if we do a quick comparison between what's in our reactant and what's in our product, we will see that it's exactly the same because this has been balanced. So when we say 2H2, we mean we've got H2 and we've got another H2 plus O2. In other words, we've got 4 hydrogens and we've got 2 oxygens. On the other side, 2H2O means we've got H2O, we've got one of them, and we've got a second one as well. So that means we've got 4 hydrogens, and we've got 2 oxygens. So it's exactly the same amount of hydrogen, oxygen on both sides. So some reactions, especially when you've got a reaction like this that's open, some reactions may appear to change in mass, but this can be explained. A reactant or product is often a gas, so its mass is not always measured. If a gas is produced and it escapes into the air, the total mass seems to decrease. This does not mean it's lost, it's just contained within the system. So this is an open system. When you have an open system, what it means is that as it's reacting, some of the gas leaves. Now, if you were to add up this liquid that's left and add up all the gas, it will still add up to 17.32. So it doesn't mean it's lost, it's just in a different place. It's not within the same container anymore. Now, if we put a lid over the container, then that'd be a closed system and the mass wouldn't change within that system. Okay, calculating masses. Now there's a question there. I'm going to explain how I do it, and we're going to have a quick look at how because there's 2 ways of doing this. The way I'm going to show you, and there's also the method that's being used in this presentation as well. So we'll start with the presentation method. Whichever one works for you, whichever method your teachers taught you is fine, as long as you can get to the correct answer in the end. Calculate the mass of hydrogen produced if an excess of acid is added to 100 grams of magnesium. So first thing, write down the RFM, the mass number. So hydrogen on the periodic table has a 1 mass number, and magnesium has a mass number of 24. Now we do mass divided by the RFM. Actually, this method's fine. Mass divided by the relative atomic mass. Now we're trying to find hydrogen. We already know that we've got 100 grams of magnesium, so that's the numbers that we know. That's what we're working with. So we're doing 100 divided by 24 because we're looking at initially the magnesium. So 100 divided by 24 equals 4 point something. Now, whatever that equals, you multiply it by the relative formula mass of the thing that you're trying to find. So times it by 2, and now you've got 8.3 grams. Now that's one way of doing it. My preferred way of doing it, and you just pick the one that you prefer, is we've got magnesium, we've got hydrogen, I put mass there, AR, and moles. Now moles equals mass divided by the mass number. So mass divided by mass number. So here, do we know the mass? We know that we've got 100 grams of magnesium. Do we know the relative formula mass? Yes, because magnesium is 24 and hydrogen is 2. Now, 100 divided by 24 is roughly 4.2. Now, 1 magnesium gives you 1 hydrogen. So whatever the number of moles of magnesium are, hydrogen is exactly the same. Now, if we rearrange this formula, if N equals mass divided by MR, if you want to work out the mass equals moles times MR. You have to multiply them. So you have to multiply these two numbers. So you end up with 8.3 reoccurring. So you just have to go with whatever works for you. So let's look at this next question here. It says calculate the mass of magnesium needed to produce 250 grams of magnesium chloride. So the information that we've got, we know magnesium has a relative formula mass of 24. Now magnesium chloride, they've given us 95, but you would usually have to calculate that. So magnesium MgCl2, magnesium is 24 plus chlorine is 35.5, so you have to add that twice and you end up with 95. 35.5 times 2 is 71 plus 24. Now, again, we're doing mass divided by the relative formula mass. So we've got information about magnesium chloride, so that's the one we're working out for. 250 divided by 95. And then what do we multiply it by? The MR of the element or substance that we're trying to find the mass of. And that equals 63.1 grams. Now that's the initial answer. 1 to 2 decimal places is sufficient. Now, you can see from that question, this one is the type of question that needs practice. So again, when you're in your application phase, where once you're in the application phase, you need to spend a little bit of time going through the maths, because in chemistry, at least up to, so those doing combined out of 60 marks, potentially at least 10 marks in each paper, if not more, will be maths based, will be questions like this. Now 10 marks is halfway to your grade 4, so they're not marks that can be missed. Yeah, you're looking around maybe 12 marks out of 60, and in the higher paper on 20 marks out of 100, so you're looking at about 20%. That means you're only about 6 marks from your grade 4. Not that anyone's aiming just for grade 4, what we're aiming for is a grade 9. So let's have a go with this in this question. There's several stages to the production of sulfuric acid in industry. Calculate the maximum mass of sulfuric acid, H2SO4, that has an MR of 98, produced from 400 tons of sulfur trioxide. So we're going to deal with the sulfur trioxide first. So because we know sulfur trioxide, we're going to do 400 times by its MR. So we're doing the mass, which is 400 tons, times by 80. So that is 32,000. And then what do? We multiply that number. Sorry, I've done that the wrong way. We're going to do mass divided by MR. So we're doing 400. So let me just drop that out. This is not the methodology that I use commonly, but I think it's a good methodology. So initially we're going to do mass divided by the MR, which is 400 divided by 80. And how do we know we're dividing 400 by 80 and not 400 by 98? Because we've got 400 tons of sulfur trioxide. So we're only looking at SO3, sulfur trioxide. Now that equals 5. Now, what do we multiply that 5 by? Now we multiply with the MR of the mass that we're trying to find, which is 98. So 5 times 98, which equals 490 tons. So let's just check if that's correct. Yeah, and the answer is 490 tons. So I'll erase this. So afterwards, if you're watching and using this to revise, you can check the answer. So everything we're going through right now, the recordings will be available on YouTube as soon as we finish. Now in terms of notes, there are going to be some notes available. There's just some issues at the moment, so once that's corrected, we will send them out. If you do want the notes, what you'll need to do is contact us. You can either join our WhatsApp channel, or you can register and we'll contact you via the WhatsApp channel. The best way is to join us through join the WhatsApp channel. Majority of our information will be through the WhatsApp channel. So if you can join us, that'd be really helpful to yourself and to us as well. Now, because we don't have the notes right now, what I would advise is making a note of any topic that isn't making sense, or asking questions in the chat, linked to anything that you're unsure about. Now, as part of our initial phase where we're understanding, where we're activating, as part of our initial activate phase, what's really important to do is work out what you do and don't know. So if there's something that's not making sense, ask questions, or ask me to go through it again. And then when we have our question answer session, I can use that time, or I can use it during this session as well. So when we're working out the concentration of something, it equals mass divided by volume. So concentration equals mass divided by volume. Now, one thing you've got to keep in mind is if you are given a volume in centimeters cubed, if you want to convert centimeters cubed into decimeters cubed, you have to divide by 1000, because the answers are always in grams per decimeters cubed. So here it's very important that mass is in grams and volume is in decimeters cubed. Now, what is a mole? So a mole is a measure of the amount of substance. Relative formula mass is the sum of the atomic masses of all atoms in a compound. The main thing we're looking at now is mole, is the measure of the amount of substance. Specifically, if we say I've got 1 mole of carbon atoms, that means I've got this many atoms. Avogadro's constant, 6.02 times 10 to the power of 23. That's the number of particles or atoms in 1 mole of a substance. Now, because that number is so big, instead of using that big number, we just say mole. So mole is the number, 1 mole is this many atoms. Now, if I say I've got 1 mole of pens, that means I've got this many pens. If I say I've got 1 mole of footballs, or 1 mole of onions, I've got this many onions or this many footballs. So that's what we mean by 1 mole. Now if you get a question that says you've got 0.5 moles of a substance of, for example, let me just rub that out. Got 0.5 moles of CO2. And it says, how many CO2 molecules do we have? So what you do is, you've got half a mole. 1 mole is this much. So half 0.5 moles will be 0.5 times 6 times 10 to the power of 23. So I should have written 6.02. So you end up with 3.01 times 10 to the power of 23 molecules of carbon dioxide. So here's a really good past paper question. It says, calculate the number of atoms combined in 1 mole of C copper iodide. Now copper iodide looks like this. The question is asking me the number of atoms in 1 mole. So how many atoms do we have here? We've got 3. Now 1 mole contains 6.02 times 10 to the power of 23. But we've got 3 atoms. So we therefore multiply that number by 3. So we end up with 1.8 times 10 to the power of 24. How do we calculate moles from a chemical reaction? So we've got 1 mole of nitrogen reacting with 3 moles of hydrogen to give us 2 moles of ammonia. The amounts of the substances involved can be found from the balanced chemical equation. So it's telling you how many moles are reacting with how many moles of another substance. So here we've got 1 mole of nitrogen reacting with 3 moles of hydrogen to give us 2 moles of ammonia. So how do we calculate moles from a chemical reaction? What we can do is convert the masses in grams to moles. So if you get a question like this, where you're told how much of each one you have, you can use this formula. N equals M over MR. Now N, instead of N, you can write mole. If that makes it easier for you. So here what we're going to do is work out the number of moles for each thing. So butane, the first thing we've got to work out is MR. So carbon is 12. So if you look at your periodic table, carbon is 12. Hydrogen is 1. So what's 4 times 12? That's 48. Plus 10, 58. And we've got 88 grams of it. So let's initially work out the MR of everything. So C4H10 equals 58. O2 is 2 times 16, so that's 32. Carbon dioxide we did before, carbon is 12, each oxygen is 16, so that equals 44. And H2O we did before as well, that's 18. Now what we're going to do is do the mass. So look, mass, and divide it by the MR, so these numbers here. And if we do that, we get 88 divided by 58, which is 1.5 moles. We get O2, oxygen, which is 10 moles. And carbon dioxide, 264, so we've got, there's 264 grams divided by 44, which is 6 moles. And H2O, which is 144 grams divided by 18, which is 8 moles. And now, what we do then is turn this into its simplest ratio. So if we some of this part of the calculation is slightly wrong, but with the simplest ratio, what we can do here is if we treat the 1.5 moles as a 2, or we can double everything, so ignore this, so that becomes 3 moles, the 10 moles becomes 20, the 6 becomes 12, and 8 becomes 16, then we now know how much of each one is reacting. So we've got 3 of these reacting with 20 oxygens, and that produces 12 carbon dioxides and 16 waters. Now, you're not really going to get a question that's as detailed as that, but what you might get a question asking is, there's C4H10, we've got 88 grams of it, how many moles of C4H10 do we have? So what you would do is, you would find out the mass is 88, the MR is 58, so the number of moles equals 88, the mass, divided by the MR, which is 58, and that gives us 1.5 moles. Now, the second part of the question might say, how many molecules is that? So, if you want to know how many molecules of C4H10 in 88 grams, because now you've worked out the moles, 1.5, the next stage you do 1.5, and you multiply by, I'm hoping everyone's now learnt the number, but you're going to do 1.5 and you're going to multiply by 6.02 times 10 to the power of 23. And that will give you the number of molecules of C4H10. And that's the kind of question you will get in a higher paper. In a foundation paper, it'll stop at this point where you get 1.5 moles. In a higher paper, you'll have to work out, they might ask you a question to work out how many atoms it has or molecules it has as well. Okay, what is the limiting reactant and why is it important in a chemical reaction? So in a chemical reaction with 2 reactants, we often add extra of 1 reactant. Now the reason we add extra of 1, so here most likely we've got extra liquid. We've got more of the solution. We have the solution and we keep adding the solid until all the solid stops reacting. So initially we get a spatula of solid and we add it in, and you'll see it dissolve. Then we take another spatula of the solid and we add more, and you'll watch it dissolve. And you keep doing that until the solid builds up at the bottom. Now, when the solid builds up at the bottom, what does that tell you? That it's in excess. Now, so in a chemical reaction with 2 reactants, we often add extra of 1 reactant. Now that ensures the other reactants completely used up. Now, when it starts building up at the bottom, that indicates that all the acid is fully reacted now, or all of the solution is fully reacted. The reactant that is used up first is called the limiting reactant because it controls how much product is made, meaning there wasn't enough of it for the reaction to continue. Now, Avogadro's constant, just to answer one question, Avogadro's constant refers to atoms or molecules. It just refers to how many of them you actually have. So they could ask you how many atoms it has. Now, if it asks you how many atoms it has, so for example, if you've got CO2 and it says we've got 1 mole of it, and it says how many molecules you have, from a molecule perspective, you would say 6.02 times 10 to the power 23, because that's a molecule. If it said how many atoms within 1 mole, then you multiply it by 3, because you've got 1, 2, 3 atoms. You've got carbon, oxygen, oxygen. So then you would multiply 6.02 times 10 to the power 23, because the question is asking you for how many atoms in 1 mole. That's a really good question. Thanks for asking, because it's important to be able to differentiate between the two. So when zinc reacts with magnesium sulfate solution, solid magnesium is formed. So there's 2 possible equations for this reaction. Now this is the kind of question that really puts a lot of people off. They'll look at it and they'll get put off by the numbers. They'll get put off by the way it looks. But actually it's not too complicated a question. You've got 2 equations there, and and both of them represent zinc reacting with magnesium sulfate. Solid magnesium is formed at the end. So in both of them, we've got magnesium sulfate here reacting with zinc, and it makes magnesium and zinc sulfate. In B, it's the same thing, but it's got different balancing. So it's found that 12.5 grams of zinc react with 14.75 grams of magnesium. Carry out a calculation to decide if equation A or B represents the reaction taking place. Now, do you know all you've got to do is this? So you know how I've already told you N equals M over MR. Now what we're saying here in the equation number 1 in A is 1 mole of magnesium reacts with 1 mole of zinc. In part B, we're saying 3 moles of magnesium sulfate react with 2 moles of zinc. So in one, we've got 1 mole of magnesium sulfate reacts with 1 mole of zinc. In the other one, we've got 3 moles of magnesium reacting with 2 moles of zinc. So we need to work out which one's right. Now, we know mass here, and we know the MR. So let's work out each one. Let's do zinc first. Zinc is going to be 12.5 grams divided by 65.4. I'll work that out in a second. And for magnesium sulfate, magnesium, not magnesium sulfate. So one mole of zinc gives you 1 mole of magnesium, but in B, 2 moles of zinc give 3 moles of magnesium. So in part A, it's a 1-to-1 ratio. In part B, 2 zinc give you 3 magnesiums. So it's a 1-to-3 ratio. So the magnesium will be 14.75 divided by 24.3. So with zinc, when you do 12.5 divided by, apologies, there's an issue with this. There's one incorrect element to this question. Let me just drop this out so I can show you. The comparison should be between magnesium sulfate and magnesium. So we've got zinc, which equals 0.0.199 Now we are also doing magnesium sulfate. So this shouldn't say magnesium, it's 14.75 grams of magnesium sulfate. So the MR of magnesium sulfate is 24 plus sulfate is 32, and then we've got 4 oxygens, which is 64. So when we add that up, if you do 14.75 divided by 120, then you also get 0.191, meaning this is 0.191, the zinc, and the magnesium sulfate is also 0.191, so it's a 1-to-1 ratio. So the answer has to be A. So let me just show you with the mark scheme. It would work out 0.1229 is because I didn't use the 0.3. if you do sulfur as accurate as into one decimal place, then we would end up with 0.191. Now, so that means zinc gives you 0.191, and the magnesium sulfate gives 0.191. So it's a ratio of 1 to 1. So it's reaction A. Apologies, there's an issue there. Now, this is the last slide, or last few slides. So we'll continue from here tomorrow from 1.53. So we're going to take a few minute break. We're going to take a break for a few minutes, and we're going to restart at 3:40. So see you guys in a few minutes. Thank you very much, everyone. Okay, welcome back, everyone. So the session's going to finish hopefully as soon as we finish topic 3, physics. We should be able to do that in hopefully by 4:30. And now what we're covering is the particle model. Sorry, I've opened up the wrong PowerPoint, wrong presentation. Okay, this is the correct one now. So we are going to try and finish the whole of this content. There's a good chance we should be able to do it by 4:30, otherwise we'll be finishing this off tomorrow. So this topic is conservation of energy. Okay, so let's make a start. Now, gravitational potential energy is the energy stored in an object due to the position above the ground, meaning the higher up something is, the more energy it has. So if we drop something from a small hill, or we drop it from a slightly bigger hill, the person on the slightly bigger hill, whatever they drop has got, at that point, the thing that they're holding has got more gravitational potential energy because it's higher up. What this person at the smaller hill is holding has got less gravitational potential energy because it's lower down. And the way to calculate it is mass times gravity times height. So M stands for mass of the object, which needs to be measured in kilograms. G is gravitational field strength, which is newtons per kilogram. And on the Earth, that's usually 9.8. And H is the height above the ground, which is in meters. And gravitational potential energy needs to be measured in joules. Now, what's really, really important is that whenever you do these kinds of questions, you have to have it convert everything to the correct units. So here's an exam style question. We've got a 500 gram object and it's lifted to a height of 1.2 meters. Calculate its gravitational potential energy, assuming that gravity is 9.8. So the first thing we've got to do is convert units. So in this case, we are going from grams to kilograms. We're going from smaller units to bigger units. If you're going from smaller units to bigger units, then you have to divide by 1,000. Now, if anyone's struggling with this as a topic, and they're struggling with the conversion of units, there's a AMALearning video on our YouTube channel, and it's 1.4/1.5. It is a biology video, however, it helps with the conversion of units. It's not a particularly long video, but you will definitely understand it a lot better after you've watched it. So we've already got height in meters. Fantastic. Now, like I said to you guys yesterday, we should write everything down at one side. So we know that mass is 0.5 kilograms. We know the height is 1.2 meters, and we already know that G is 9.8. So now all we've got to do is substitute our numbers in. There's the formula. Substitute in the values and do the maths. And obviously add, round it up to one decimal place, but 2 significant figures is good. And always add the units. And that's an easy 3 marks, 4 marks. Then we've got kinetic energy. Kinetic energy is the energy an object has due to motion, due to its movement. So the faster and heavier an object is, the more kinetic energy it has. So the formula for this is kinetic energy equals half times the mass times the velocity squared. Now, where most people go wrong with kinetic energy is they forget to square the velocity. So the velocity is there, but they just forget to square it. And that one mistake leads to a loss of a mark. So kinetic energy equals mass times the velocity squared. Mass in kilograms again, velocity in meters per second, and kinetic energy in joules. Now, quick question answer on kinetic energy. So we've got a 2 kilogram ball, that's in the correct units. It's got a speed of 3 meters, that's in the correct units. All we now need to do is substitute it in. So half times, so kinetic energy equals half times the mass times the velocity squared. So mass equals 2, velocity equals 3, and let's not forget the squared part. So, and that's incorrect, so we'll do that ourselves. No, that's not incorrect, sorry. Half times 2 is 1. 3 squared is 9. So it equals 9 joules. It's already rounded up and we've already added the units, which is joules. Energy transfers. How could this... So this is nothing to do with equipment. This is the wrong title here. Now, part of the issues we've had with the resources is we've transferred them from PowerPoint and we've moved them onto Canva, and so that it's got our branding on it, and we've ended up with lots of different things in the wrong order and little kind of mistakes like this. So I'm not sure why that's happened when we've converted it across, and we're just correcting that before we start giving them out to people. What I don't want is people to end up with stuff that's got the wrong information on the wrong slides. The information is not wrong, it's just there's a few things out of place, out of sync. So we were expecting things to be out of sync where, you know, the alignment changes, but we've had not just alignment change, we've had things skip from page to page somehow. So the Sankey diagram, this is a way of showing energy transfer. Sankey diagrams start off as one arrow that splits into 2 or more points. So this arrow here represents the energy that it enters. So this is the total energy. And this part here represents useful energy. And this part here, this arrow here, represents wasted. Now, if you add up useful and wasted, it equals a total. So energy is conserved. The width of the arrow is drawn to scale to show the amount of energy. Now, you won't have to draw Sankey diagrams. What you might have to do, however, is interpret them. So, for example, when you get an efficiency question, and it will come up, efficiency equals useful energy divided by the total energy. So if you've got this Sankey diagram here and it says useful energy is 200, but the total energy was 500, then automatically for this formula you would do 200, because that's useful, divided by 500. Okay, conservation of energy. All this means is the total energy before equals the total energy after. So for example, we've got our hill top again with our person stood at the top. They're holding on to something that they want to throw. That thing that they want to drop has a certain amount of potential energy. Let's say it's got 100 joules of potential energy. When it actually drops, just before it hits the ground, when it's at maximum velocity, it will have 100 joules of kinetic energy. Now, this is assuming that there's no particles hitting it. So this is assuming no energy has been lost. So let's use a different example, a roller coaster ride. At the top, it's got maximum gravitational potential energy. Going down, the gravitational potential energy converts into kinetic energy. It turns into kinetic energy. At the bottom, you've got max kinetic energy and low GPE. And so the GPE, the gravitational potential energy, has converted into kinetic energy. So there's nothing lost. However, some energy will be lost as heat, sound, due to friction. Now, when we say lost is the wrong word. Wasted would be the correct word. So it hasn't disappeared and gone somewhere else. It still exists, it's just moving in a different way. So if you start with 100 joules of energy, you have 100 joules of energy at the end. So let's say this was actually 99 joules, and due to friction, we would then know that 1 joule of energy has been lost. But that 99 plus 1 still equals 100, so it's still being conserved. Excellent. The energy has been dissipated. That's dissipated energy is something that comes up again and again in past papers. Now, I just want to answer a question quickly before I continue. Currently on the AMALearning, with the AMALearning animations, we've almost completed biology paper 1. By next year, so those of you who are year 10, when you're year 11 and you're revising, by that point you will have one animation for every single specification point, and that will be for Edexcel and that will be for AQA. Over the next few months we'll be completing Edexcel, and after that we'll start completing AQA. We did temporarily pause because we started to plan for the AMALathon, but we'll continue as soon as the AMALathon finishes. Now, a system is 3.5 transfer of energy. What is a system? A system is a set of parts that interact and exchange energy. It can be open, closed, or isolated. Now, we don't need to know about isolated, based on energy and matter flow. What that means is an open system is energy and matter can enter and leave, for example, boiling kettle. Now, I'll use the example of a cup of tea. If it's got hot tea in there, then some of the water within the tea can turn into a gas and it can exit. And some of the heat energy can anyway, but some of the matter, some of the particles can. Now, if I had a closed system, that's like a thermos, or in this cup of tea, I could just seal the top if I had a lid. Now the particles can't escape, so that's a closed system. Now some energy will still transfer out, but particles can't transfer out, so that's a closed system. Now, there's a few different types of energy transfers that we need to be familiar with. One is magnetic. So this is a non-contact force, but it transfers energy as well. The energy stored when attracting poles are pulled apart or when repelling poles are pushed together. So if you get two negative sides of a magnet and you push them together, what you've got there is magnetic energy that then pushing each other apart. Internal energy just means the total kinetic and potential energy of particles in an object. Chemical energy, the energy stored in chemical bonds. So we saw chemical bonds before. If you've got a covalent bond, energy is stored inside that bond. So if you've got a lump of coal and it's made up of carbon atoms, when you burn it in oxygen, you're breaking those bonds and then making new bonds with oxygen. When new bonds are formed, that releases energy. So making bonds is exothermic. Breaking bonds is endothermic. It has to take energy in to break bonds. Now, kinetic. Kinetic energy is the movement of energy of a moving object. And gravitational potential is the energy stored in an object at height. And elastic is the energy stored when an object is stretched or compressed. So that's like an elastic band. When you pull it, when you let go of it, it releases the elastic energy. And nuclear. So that's energy that's stored in the nucleus of an atom. And electrostatic, the energy stored when repelling charges have been moved closer together or when attracting charges have been pulled further apart. Now electrostatic is a triple only topic. So if you're not doing triple, you don't need to worry about knowing that in too much detail. So let's look at some scenarios. So an object is projected upwards. So the energy changes that we have here is kinetic energy converts to gravitational potential energy. Because when you throw something upwards, you're causing it to move upwards. That means it's speeding up. But the higher, as it moves upwards, it starts to slow down. So initially you give it enough energy to start moving upwards. It's got more kinetic energy, but as it gets to the top, the kinetic energy decreases because it converts to gravitational potential energy. At the top there, you've got maximum gravitational potential energy. Then it starts to drop and it gets faster and faster because gravity accelerates it. Remember, gravity accelerates it at 9.8 meters per second squared. So meters per second per second. So every second it increases its speed by 9.8. So as it drops, it gets faster and faster. So the gravitational potential energy converts back into kinetic energy. Next scenario. An object is projected up a slope. I know that shows it going down, but it should have shown it going up. So as this roller coaster, let's draw the rest of the roller coaster. So it can be shown as going upwards. So now as it starts to go up, we've got kinetic energy, and then the kinetic energy, as it goes higher and higher, converts into gravitational potential energy. So here we've got a scenario where a moving object has hit an obstacle. As soon as that moving object, the fact that it's moving means we've got kinetic energy. When it collides with that wall, you hear the collision, so that's sound waves. And as I said to you guys yesterday, whenever particles collide with each other, what do we create? Thermal energy due to friction. So friction is a force, but it's a force that releases thermal energy. So please don't ever get these two things confused or mixed up. Friction is the force, but it releases thermal energy. Now, here we're talking about a boat that's moving. It's moving because it has fuel inside it. Now, they're right, this depends on context. But what we're saying here is that it's got fuel inside. And what's that fuel? Chemical energy. Well, fuel is full of chemical bonds. And what's that? When those chemical bonds get broken and reformed in a different format, it gets converted into kinetic energy. So that fuel, when these bonds are broken, they react with the oxygen to make carbon dioxide. And when the bonds in carbon dioxide are made, that releases energy. That energy then is converted into kinetic energy. In paper 2, we'll cover endothermic and exothermic in a lot more detail. So that will make a lot more sense. Next scenario. Someone jumping out of a plane. So they've jumped out of the plane. While they were in the plane, let's see if I can draw a plane. Nope, I can't draw planes. Now, when the person is in the plane, they've got lots of gravitational potential energy because they're really high up. But as this person starts to fall, that gravitational potential energy converts into kinetic energy. A vehicle slowing down. So we've got kinetic energy, and to slow the car down, so the car's moving, it's got kinetic energy, the brakes now touch the wheel. So what have we got between the brakes and the wheel? We have got friction. So these particles are colliding with each other, and that releases thermal energy. Okay, we're gonna move past these examples now. Now, definition of conservation of energy. So the law of conservation of energy states that energy cannot be created or destroyed. It only transfers from one form to another. The total amount of energy in a closed system remains constant. So energy, according to this, can't be created or destroyed. We just need to know that they convert from one form into another. Now the question says that with these type of examples, how would these be questioned in the exam? So you can get scenarios where it might say such and such a thing has happened, describe how the energy has changed. So it won't necessarily be those specific examples, but those examples are good examples. What I'll try to do for tomorrow is something that we'll definitely be doing in AMALathon 2 in the pre-exam workshop. We'll be going through lots of past paper practice. And just to remind you all, when we do the pre-exam workshop, the questions will be linked to the highest leverage topics. So I'll be covering the topics that I feel, or our analysis shows, is more likely to come up. Now, in the past, I have always been around 80% accurate. I don't think I've ever been lower than 70%. So usually my students always go into the exam with me having covered 80% of the content that actually came up. Now, some of those students don't get grade 9s, and that's because they haven't followed the AMAL process, meaning they didn't understand everything from beforehand, they didn't memorize, and they didn't do lots of past paper practice. So me doing a 3-hour last minute, going through the stuff that's going to come in the exam, what that allowed them to do was to sit in the exam and say, oh, Mr. Tanvir just mentioned that 5 minutes ago. But because they hadn't practiced, because they hadn't fully understood, they weren't able to then apply. So the activate, memorize, apply has to start from now as a minimum. 4 weeks is still enough time to get the best grades. Okay, so this means that whenever there are energy transfers in a closed system, there is no net change to the total energy. So the amount of energy that goes in is, if you go back to the Sankey diagram, the amount of energy that went in, some of it was useful, some of it was wasted, but the amount of energy that initially went in was still the same. So I'm not very good at drawing Sankey diagrams. So the amount of energy that goes in, some of it is useful, some of it is wasted. But if you add the useful and the wasted, it always adds up to the original amount. So energy in equals energy out. Now, what's wasted energy? 3.7. So energy is typically wasted in the form of heat, sound, or light. So it's wasted in the form of heat, sound, or light. Let's use a really common example, a light bulb. Now, it gives out light. Would we consider light to be wasted for a light bulb? No. Sometimes you can hear a light bulb. So sometimes it can have sound, which is wasted energy. If you touch the bulb, it'll be warm or hot. Older light bulbs will be very hot. But the newer light bulbs will be slightly warm, so that means light bulbs also give off heat, which is wasted energy. So let's look at these examples as well. Friction converts useful energy into unwanted heat. For example, in car engines. For example, when a tire is braking, a lot of heat energy is released because of friction. That's wasted energy. Air resistance, that causes kinetic energy to be lost as heat. So if a car is moving, if we're running, for example, the faster we run, the more wind you feel in your face. That's because particles are hitting your face. And that means some of the energy is being converted to thermal energy. If a car is moving and lots of air particles are hitting it, that means some of the kinetic energy is being converted to heat, so the car slows down. Electrical resistance, so wires heat up. This is something that we'll cover in more detail when we cover the electricity topic. But wires heat up when electricity passes through them. And when they heat up, the ions inside the metal wire move around more, stopping the electrons from going through. They impede them, they slow them down. Then we've got unwanted sound and light. Devices like engines and light bulbs produce unnecessary noise or light, leading to wasted energy. Well, light bulbs don't produce any wasted light, but they do produce wasted heat. Now, how energy is dissipated in everyday devices. This is something that's really important. So it comes up every year. Every year you're going to see questions that talk about energy that's been wasted. And another word for wasted is dissipated. Wasted or lost. So for example, a car. Fuel energy is lost as heat from the engine. In TVs, if you ever go to the back of the TV, you might feel it to be slightly warm. That means some of the energy has been lost as heat. In light bulbs as well, some heat energy is lost. Now, whenever heat energy has been lost, what do we call that? We say it's been dissipated. It's been lost to the surroundings. lost to the air in the environment, in the air. So how can we reduce energy wastage? We can use LED light bulbs, which are more efficient. We can use lubricants to reduce friction, or we can insulate electrical devices, and that reduces heat loss. Increasing efficiency. So that's what we've just covered in the previous slide. We can reduce friction by using lubrication, so that reduces friction. Ball bearings reduce contact between moving parts in machines. But the main thing here is lubrication. We can reduce heat loss with insulation, with double glazed windows. We can use shiny surfaces. So for example, in thermos flasks, on the inside you've got a shiny surface that then reflects the heat back inwards. Now, I wouldn't worry about this too much, not till we get to the electricity topic, but this part here. How can we reduce air resistance? We can make something more streamlined. So for example, if you have a car that looks like this, all the air particles are going to hit all these flat surfaces. Now if you have a car like this, then the air particles have less of a surface to hit. In fact, if anything, they flow over the top. Smooth surfaces also reduce air resistance. Okay, rate of cooling. Heat transfer through a material. So what is conduction? It's heat transfer through a material without the material actually moving. It happens by particles vibrating. So it happens in solids, best in solids. It can happen in liquids and gases as well, but it occurs best in metals. It happens best in solids, but out of all the solids, it happens best in metals because they've got free electrons. And what do the free electrons do? They carry the heat energy, exactly the same way they carry electrical charge. It doesn't happen as well in liquids and gases because particles are farther apart. Well, I should say further apart, because the particles are further apart. So with a gas particle, the thing with conduction, the way it occurs is like this. If you've got a solid, and solid has a regular arrangement, if I start heating up this side, as soon as this side heats, the particles got more energy. So because they've got more energy, they start vibrating, they pass the energy on to the next part. We start vibrating and pass the energy on to the next part. We start vibrating and pass on the energy to the next part. So in a liquid, because the particles are far apart, if the heat energy goes into this particle, even if it starts vibrating more, moving around more, it's still not guaranteeing that it's going to collide into the next particle because they're further apart from each other. So it takes longer for the heat energy to transfer through a gas. That makes the gas a really good insulator. That's why now with a metal, if you have a metal, it has free electrons, delocalized electrons. So the electrons can carry the heat energy and they can carry it through the metal. So metals are really good conductors, whereas things like wood, plastic, air and wool are better insulators because they don't transfer the heat energy as quickly. Okay, so this I've already explained. Heat makes particles vibrate. The vibrations pass energy to nearby particles. Metals conduct well due to the free electrons. Now, in a solid, atoms are in fixed places but are free to vibrate. Now, this is what I've already covered and I've already explained on the previous slide, so I'll move on. Now, metals, and I have explained this, but we'll explain this just again. Metals have got delocalized electrons, which are able to move freely. When a metal is heated, these electrons gain kinetic energy and move faster. So, when a metal is heated, the electrons gain energy and they move around faster. They collide with metal ions, causing them to vibrate faster. So they pass on heat energy onto the ions, and they themselves are also able to transfer the energy through. The collisions of these electrons cause the rate of conduction to increase. Now, does the thickness of a conductor affect cooling of a building? The thickness and type of material used in walls, roofs, and windows significantly influence how quickly a building loses or retains heat. So if you've got thicker walls in a house, so if you think about really hot countries, sometimes you've got thicker walls, but that's because it stops the heat energy from coming into the house as quickly, so it keeps the house cooler. But if you've got the heating on inside your house, if you've got thicker walls, then it takes longer for the house to cool. It'll stay warm for longer. So more resistance to heat loss, it keeps buildings warmer in winter and it keeps them cooler in summer. So that's a good thing in a hot country. Thinner walls equal faster cooling, so there's less resistance, so heat escapes or enters more quickly. Now, if the thickness and type of material, so the type of material makes a difference. If you're using a material like metal, they're good conductors. Glass is not a good conductor. That means with the metal, you've got fast heating and you've got fast cooling, because the heat energy is absorbed by the conductor and it's emitted quite quickly as well. So metal roofs heat up and cool fast. Now, a good insulator is things like wood, foam. They slow down the cooling. The heat transfer is slowed down, keeping temperature stable. For example, insulated walls maintain comfort. Now, efficiency I've already gone through. Efficiency is the useful energy divided by the total energy. And you can times it by 100 and give it as a percentage, but that's not necessary. You do not have to give it as a percentage. So let's go through an example. So LED light bulbs are 90% efficient. That's because most energy is converted to light. 10% is still converted to heat energy and sound. Traditional bulbs were actually only 10 to 15% efficient, meaning 90% was lost as heat. That's why if you touch one of the old style light bulbs, you burn your hand. So let's have a go at an exam style question. A machine is supplied with 600 joules of energy, but only 240 joules is converted into useful work. Calculate the efficiency of the machine. So we know efficiency equals useful divided by total. So in this question, we know the machine is supplied with 600 joules of energy, so we know total is 600. And 240 is converted into useful work, so that goes at the top. So we therefore know that is 0.4. Now if you want to multiply that by 100, then you'll get 40%. Both are valid. Both are absolutely fine. Okay, so how can we increase the efficiency within a house? How can we stop energy being lost? So we increase the thickness of the walls. Well, that wouldn't be an easy thing to do, but potentially you could build a house with thicker walls. You can reduce the thermal conductivity of the walls by changing the materials. You can add double glazing, draft excluders, things like curtains and things that, for example, go at the bottom of the doors to stop air coming in. You can insulate the building, such as cavity wall insulation. You can put insulation in the roof as well. You can put down carpets on the floor, on the inside. All of this would increase efficiency. So what can be done to decrease heat loss from buildings? That was what we were showing here, but we're going to talk about it in a bit more detail now. So to decrease heat loss from a building and increase efficiency, you can put carpets and rugs down because they trap the heat and keep the floors warm. Underfloor insulation, that stops the heat escaping into the ground. You can use smart thermostats. They control heating to save energy. And heat recovery systems, so you could reuse heat that would normally be lost. Now that's not a straightforward thing to do. That would have to be done whilst building the house brand new. Okay, energy sources. So now what we're looking at is non-renewable and renewable energy sources. Now, so this is the kind of topic that usually comes in foundation, but it can come in higher as well. Now, what are the different non-renewable energy resources on Earth? Non-renewable energy resources are natural resources that cannot be replenished. So if something non-renewable, this is a common question actually. I've seen this in higher papers and foundation, and people get it wrong because they put the wrong wording down. Now, some examiners might give a benefit of the doubt mark, and some of your teachers might, but if it's not correct, it's not correct. So you've got to think about the correct wording. Non-renewable energy resources are natural resources that cannot be replenished within a human lifetime. They take millions of years to form and will eventually run out. Burning them releases greenhouse gases, contributing to climate change. So the types that we've got fossil fuels like coal, which is a solid fuel burned to make electricity, oil, which is a liquid fuel used in transport and heating, and natural gas, which is used for heating, cooking and power. And then we've got nuclear fuel, which is also a type of non-renewable energy resource. Uranium is the main fuel in nuclear power stations. Plutonium is made from uranium, and that's also used in reactors, but you do not need to know about plutonium. So, advantages and disadvantages. My advice, memorize this slide. So when it comes up, it's a nice, easy question. It's not going to take too much. It'll be a nice, easy question for you. So, coal, there's cheap and there's plenty of it. It will eventually run out, but there's still plenty of it. It's easy to store and transport, and it's reliable. Reliable means the generators, the power stations used for coal, you can run them 24 hours a day. They can provide energy 24 hours a day, so they're reliable. The issue with some of the renewable stuff is they don't work at certain times of day. So for example, solar panels don't work at night. Wind turbines don't work when it's not windy. Coal can work all the time. As long as you've got it, as long as you've got lots of coal, it's going to keep working. Then you've got oil. It provides lots of energy. It's used in transport and industry. There's a good infrastructure for it. Meaning, if someone's car runs on petrol or diesel, there's lots and lots of petrol stations everywhere for you to keep filling your car up. And natural gas. It's cleaner than coal and oil. It's efficient and widely used. It's good for heating and power. Now, when we say it's cleaner than coal and oil, natural gas releases less carbon dioxide, so it is cleaner. But it still releases carbon dioxide, so it still has that as a disadvantage. Now, nuclear is very efficient. It doesn't produce carbon dioxide, and when it's running, it produces no carbon dioxide when running, and it is reliable energy. So all of these are reliable. As long as you've got a good store in the back, you're good to go. It'll last 24 hours a day, 365 days a year. Nuclear, you don't need a massive amount of, because a piece of a radioactive substance provides, especially something like uranium, it has a half-life of what they think is a few million years. So if you have enough purified uranium for a long time, it's just going to keep releasing lots and lots of energy. It is, however, dangerous. So coal, lots of carbon dioxide is released. So lots of air pollution, acid rain, because a lump of coal isn't just made of carbon, it's mainly carbon, but in amongst that carbon there can be some sulfur. And when that sulfur burns in oxygen, you don't just get carbon dioxide, you get sulfur dioxide as well. That sulfur dioxide, when it reacts with the rain in the clouds, it becomes H2SO4. And it falls with the water in the clouds and it causes acid rain, which means it harms nature and it can lead to health problems as well. Now, when we mine for coal, it harms nature and health also. Oil is non-renewable and it's running out. Oil spills harm the oceans because they float on top of the water. They're less dense than the water, so it floats on the water. So anything that tries to land in the water or is towards the surface gets harmed, and it releases greenhouse gases. Now, natural gas, it still releases carbon dioxide. If there's a methane leak, that's very harmful, and things like fracking can cause earthquakes. Now, nuclear dangerous radioactive waste is released from it. As in, when you're trying to decommission it, you're left with really dangerous radioactive waste, which has to be buried deep in the earth with concrete around it. There's risk of serious accidents. So if you consider some of the issues that are happening around the world at the moment, if someone was to so for example, Ukraine has lots of nuclear power plants. If a bomb was to land nearby on it, causing the power plant to overload, that can be a serious incident. When something like that happened in Chernobyl, which was a nuclear power plant, where something went wrong, the amount of energy released from it meant that no one was able to live there for many years. Only now, recently, have people started to consider moving back. But it is very expensive to build as well. So nuclear power stations are probably the best source of energy, but there's the potential that it could lead to danger. That scares people. Now, what are the different renewable energies? Now, renewable energies we love because they do not release greenhouse gases. And a renewable energy resources are energy sources that naturally replenish over time and will not run out on a human timescale. So they are things that will keep replenishing. So we've got solar energy, wind energy, hydroelectric, biomass, geothermal, tidal and wave energy. So let's look at the advantages and disadvantages. And this is going to be the last part of today's session. And then I'll be continuing with the triple award, which will be looking at topic 3 biology. So we're looking at protein synthesis. Now, solar energy provides an infinite source, so sunlight. There's no greenhouse gases, it's no maintenance. However, it's weather dependent, so it works okay on cloudy days, but it doesn't work at night time. It's expensive initial setup, and it requires a large area for panels. So if you're going to provide energy for a city, you will need another city of solar panels. Wind turbine is fantastic, no pollution or carbon dioxide emissions. It's sustainable, it's cost effective over time. Land can still be used for farming around it, but the wind is unpredictable. It can be noisy and affect wildlife, birds, and it's expensive to install and maintain. People don't like living near them. So if you can imagine someone living by nice green sweeping hills, and then all of a sudden someone comes and builds wind turbines, then their view's been ruined. Also, when they spin, it releases a lot of noise. It is really noisy. So people don't like them. The ones that live local to them don't like them. Hydroelectric power. So this is where, for example, you've got a lake towards the top, you've got a lake towards the bottom, and as the water falls, there's like a stream in between them, and as the water goes from one to the other, it makes a turbine spin and creates electricity. Now, this is really reliable and it's consistent. No carbon dioxide emissions, so no greenhouse gases. It can store water for electricity demand, so it can be used as a backup as well. Now, when this part here, or this part, one of them is made, now in areas usually you have one lake. Now if you produce another lake or another area, that's usually a man-made. So and then you also need a dam to stop off the water in one part. Now, building of the dam disrupts ecosystems and fish migration because you're destroying a big area of land. It's expensive to initially build and is limited to areas that have rivers, because what you'd actually end up doing is you're damming up one of a potential river. So if there's a river there, you dam it up so the water stops and it's all stored, and then it can be used for hydroelectric power. Geothermal, biomass, tidal wave energy. Now, geothermal energy is, so if we look at the Earth and the Earth's crust is about 20 kilometers wide, but there are some parts of the Earth where the crust is a little bit narrower. So if you're in an area where there's volcanoes and the crust is a bit shallower, there's not as much of a crust, it means you can dig down quite deep and you're already closer to the mantle, which means if you dig down to a point where it's hotter, you can now pour water into that hot part of the earth. So you're still digging into the crust, but it just means you're close to the mantle, and that means it's going to be hotter. And what you can do then is use that heat to boil water. That steam is then used to turn turbines. So it's reliable, it operates 24/7, it's low environmental impact, no fuel needed. Is limited to certain locations, is high upfront costs, there's a risk of earthquakes due to drilling, so there are issues with this. But once it's set up, it's amazing use, it's a really cheap energy. Biomass energy is using organic waste, so that's really good, it reduces landfill waste. So any unwanted food and stuff that we throw out ends up in a normal bin. If we use it for biomass energy, it's a good use of it. It can be continuously produced and it's cheaper than fossil fuels. But the problem is when we burn it, it produces carbon dioxide and other gases. But the difference here is when a tree grows, it takes in carbon dioxide. When we burn that tree, it releases carbon dioxide. But the amount of carbon dioxide it took in is the amount that it gives out. So that's the same with anything that's organic. Anything that we eat, the amount of, when I say anything we eat, I'm talking about plants. So when we eat anything that's plant-based, the amount of carbon dioxide it took in when it was growing is the amount of carbon dioxide it gives off when we burn it. So it's carbon neutral. It does require large amounts of land areas. Now, and overuse can lead to deforestation. Now, the fact that it's using all that land, that means it's using valuable farm space. Now, considering we're a planet with over 8 billion people, using land just to make a little bit of energy is just using a bit of land just to make a bit of gas or energy is wasted. It should be used for food. Tidal and wave energy. So predictable and reliable source, no greenhouse gas emissions, long lifespan of equipment. So predictable and reliable source, no greenhouse gases and long lifespan of equipment. It's doesn't provide, that's a positive. Let's just put the same thing there. Right, tidal and wave energy again does not work at certain times of the day. So if you don't have a tide, it won't work. Okay, so now our energy resources is changing due to several factors. So shift towards renewable energy. So a lot of governments around the earth are shifting towards renewable energy to reduce carbon dioxide emissions, because in the past some people denied if global warming was happening or not, but no one denies it anymore, or not as many people. Energy efficiency improvements. So a lot of companies want to now make things more energy efficient, because that then reduces the overall energy consumption, it reduces the electricity bills. Energy concerns. So there's a growing awareness of climate change has led to stricter regulations and policies encouraging cleaner energy sources. Now, depleting fossil fuel reserves. So as fossil fuels start to decrease, then it's important that there's a backup of renewable energy. Now, many governments are providing subsidies and incentives. So governments are now providing subsidies and incentives for renewable energy adoption, meaning they're paying towards people's solar panels and wind turbines. Not the whole amount, but still something rather than nothing. And advancements in energy storage. Improved battery technology allows for better storage of renewable energy, making it more reliable. Okay, excellent. So today, big thank you to everyone. We are going to now continue with triple in 5 minutes. So we're going to take a 5-minute break and continue with triple. Thank you everyone that's doing combined. I hope you took benefit from the session. If you can give me some feedback or give me a rating out of 5, I'd really appreciate it. And if we can act upon it, we'll do our best to. I'm just going to quickly answer this question. It says, can we just say people are more aware about climate change so government uses renewables? Yeah, that would be an accurate summary of it. Usually with questions like this, you'll be given facts and numbers and you'll have to interpret the data. So, but yeah, that would be a summary of the understanding. And thank you for those that have given 5 out of 5. I won't be offended. Oh, thank you, Em. Appreciate it. Please like, share. If you've not subscribed, please subscribe. What we're finding at the moment is we're peaking at around 40 people watching live, but then afterwards a lot of people seem to watch it, so we end up with 600-700 views, which is really good. We really appreciate people watching, but it would be nice to have more people live. But if it's not, even if only one person turned up, I'm still going to teach exactly the same, so it makes no difference. So like I said, we're going to continue in just a few minutes. So those that combined, have a lovely evening. I'll see you all tomorrow. Okay, triple scientists, let's get started. Okay, this is only paper 1. And let's go through the triple content. Anyone that's been with us now for, this is coming on to the 4th hour, really appreciate it. I think we've had a few people that have been with us since quarter to 10, since 10 o'clock through the AQA and the Edexcel. So those that have been in both sessions, I really appreciate people that have been studying with us. Hope you're all taking lots and lots of benefit. Now, what I would advise for those who are doing triple award is what I'm going to go through is well, everything's linked to protein synthesis. Now, if anything comes up from protein synthesis that doesn't make sense, please ask, and I will go through it again tomorrow in even more detail, or I'll make time to go through it. But I would also advise that you check on the AMALearning. If you go on the AMALearning YouTube, which you're already on, if you watch the animations for all the triple award topics, it'll make it really clear as well. So all for 3.7b, there's an animation, there's one for 3.8, 9, 10, 11. These are all the triple topics. And the last two, I think it's 3.15 and 16, the last two specification points from topic 3 are also triple award. So that's what I'm going to go through now. Hopefully it won't take too much longer than half an hour, and then that will be the end of the day. Now, if you've got questions, I'll do my best to answer. If you want to go through this again, this video will be available as soon as I finish, but also the actual animations that we've already made for this are already uploaded as well. So 3.7b, explain how the order of bases in a section of DNA decides the order of amino acids in the protein, and that these fold to produce specifically shaped proteins such as enzymes. That's the actual specification point. It's quite a wordy, long specification point, but the main thing you need to understand from 3.7b bases is triplet coding. So how does the structure of DNA affect proteins made in protein synthesis? Now, the order of bases, each triplet code, so each set of 3 bases determines the amino acid order in a protein. Now, we're going to make sense of this in just a second. DNA contains sequences of bases, with each sequence of 3 bases called a triplet or a codon, coding for a specific amino acid. So there's our DNA. If we now look at the DNA, each 3 bases codes for amino acid. That's a triplet, that's a codon. So this will code for a specific amino acid. The next 3 will code for another amino acid. The next 3 will code for another amino acid. Now, once we made all these amino acids, what forms in between them is what we call a peptide bond. So the amino acid is the monomer, and when they join to each other, they become a polymer, which is protein. Now, depending on how each amino acid reacts with each other, because the weak intermolecular bonds form between the amino acids. So let's say this one here makes a weak intermolecular bond with this one here, that would cause it to fold over. So suddenly your chain of amino acids become a three-dimensional protein. So the main thing to understand from this part here, and it helps with protein synthesis massively, is that understanding that the amino acid order three bases is called a triplet or a codon, and that codes for a specific amino acid. So DNA carries the genetic code. The sequence of bases A, T, C, or G in DNA forms a code. This code determines the order of amino acids that are joined together to make a protein. Now, the triplet code, every three bases in the DNA sequence called a triplet or codon codes for one specific amino acid. So this is just reiterating what we've just gone through. So here is, again, this is just reiterating what we've just gone through. So proteins are made up of long chains of amino acids, and the order of amino acids determines the protein structure and function. So you can see here, this triplet or codon has made this amino acid. This codon has given us the next amino acid and the next one. And then the amino acids join together to make a protein. Now, why is the protein shape important? In other words, why is it important to have the amino acids in exactly the right order? Because the correct order makes a specific 3D shape, so a specific protein. So, and that's needed for enzymes, because enzymes have specificity, they've got specific shapes. Hormones fit into receptors around the body and have an impact. They have to have a specific shape. Antibodies have a specific shape so that they attach to a virus or a bacterial cell and then destroy them. If they don't fit into certain parts of the bacteria or the virus, they can't destroy them. So they have to have specific shapes. Okay, now this is a question that many people got incorrect when it first came out as a GCSE question. How many amino acids will be produced by the above strand of DNA? So let's have a quick look at this. 1, 2, 3, 4. So the answer is 4. Because we're looking at the triplet codes. Each triplet code makes an amino acid. So the diagram below shows an mRNA molecule. Okay, so we've covered this already. Okay, so now we can look at proper protein synthesis now. Now, the reason I would advise you to watch the animation after I've explained it is because in the animation, everything's moving around. So you can actually see, you can visually see what's happening. I'm going to be describing it and do my best to kind of bring life to the static pictures, but in the animation, it really makes it clear. Now, protein synthesis is the process by which cells produce proteins based on the genetic instructions in DNA. It occurs in two main stages. Number 1, transcription. And number 2, translation. So stage 1 is transcription. The DNA unzips around the gene. So you look over here, this is the DNA. I'm just going to highlight instead. So that's your normal DNA. But here you can see how it's unzipped. It's opened up. Now, so the first thing, the DNA unzips around the gene, and then it says RNA polymerase is an enzyme that works like a copy machine for the RNA. So here is the RNA polymerase there. And what it's doing, now this RNA polymerase is on the way out. So it's actually moving in this direction. But what it's done as it's gone past, I'm just going to remove the highlighting so it can be seen a bit more clearly. And what happens as it goes through is, you see where you've got T in the DNA there. So in the RNA polymerase, which is an enzyme, it attaches an A. So there's lots of free nucleotides here. And this is a nucleotide in the different bases, A, T, C, and G. And look, there's G, so what's attaching to it? A C. There's C, and what's attaching to it? A G. The only difference is, when you have A, what attaches to it, the complementary base, is going to be U. So T gets replaced with U. So this strand here, we call messenger RNA, mRNA. And the thing that's joining it together, that's attaching it, is called RNA polymerase. So the enzyme helps attach everything together. So in a sense, it acts a bit like a glue. Now that we've got an mRNA, if you look at the DNA, the mRNA is the exact copy of it. Not exact copy, as in it's the complementary copy of it. So here you've got, I'll remove all this. Yeah, this is why I was saying the animation is so much better, because you can actually see everything moving around. It helps really helps understand it. But I will do my best at the moment anyway. So you can see here, the DNA has got T, but the mRNA has got A. Then the DNA has got G and the mRNA has got C. So it's making a complementary strand. That complementary strand now will leave the DNA, leave the nucleus and go to a ribosome so it can be translated. Now, the whole of this DNA is too big to exit the nucleus. It's so big it can't leave the nucleus. So, and what we need to do is we need to take the information from this DNA and take it to the ribosome. So because the actual DNA is too big, then a small copy of that particular gene that's required is taken out instead. That then diffuses out of the DNA and makes its way to the ribosome. And the ribosome then translates this. And each of these three, it converts into an amino acid. So in stage 2, we've got translation. So essentially what's happened now in transcription is, it's like if you've got a really big recipe book and it's too big to pass on to someone, but someone says, I just need page 35. So it's like you making a copy of page 35 and then passing it on to that person. The second part is where someone looks at the recipe and actually cooks it up. So translation is like building a protein using the instructions from the mRNA strand. So it's like a recipe. So this is what's actually ends up happening. So translation here, the exact order of amino acids in a protein determines its unique structure and function. So this bottom strand is a little bit pixelated, but as it goes through now see this big blue bit here, that's the ribosome. As that strand makes its way through, so I'm just going to remove the thingy and just add a highlighter. As that strand makes its way through, for every 3 amino acids, for every 3 bases, so here we've got 3, the tRNA, so this is the tRNA, brings along the correct amino acid and then attaches it onto the next amino acid, to the next the amino acid that have already been created, that have already been made. So for each 3 set of bases, a new amino acid is attached. So now you've got a chain of amino acids, in other words, a protein. The exact order of amino acids in a protein determines its unique structure and function, emphasizing the specificity of proteins. So we use these then proteins for enzymes, hormones, antibodies, structural enzymes. So protein synthesis is a vital biological process that converts genetic information from DNA into functional proteins which are essential for life. So this is what we've already covered. Now, I'm just going to come back to this. So we've spoke about transcription. That's where the mRNA makes a copy of the DNA of a specific gene. Then in translation, each triplet coding gets converted into amino acid, and then the amino acids are attached to each other. And that occurs in the ribosomes. And the attachment is done by the tRNA. So like I said, if you're able to watch the animation, it really does add a lot more clarity to it. Now, within a protein, within DNA, you have coding regions and non-coding regions, and that's the next 2 specification points. So we'll go through that. Now, this is a past paper question. Describe how protein synthesis in the host cell makes viral proteins from this mRNA. So this is an application question. Not an easy question at all. But if you understand the whole process in protein synthesis, then it's really straightforward. So look, the viral mRNA produced in the host cell's nucleus. So what we're actually talking about here is translation. We're talking about stage 2, because what's happening in this question is stage 1 has already occurred. The virus has provided the mRNA. So it's provided a strand of mRNA which is full of bases. So DNA is a double strand, mRNA is a single strand. Now, as long as that goes through the ribosome will take each codon and make a specific amino acid. But this time, when it takes each codon and makes a specific amino acid, what has it actually made? It's made the virus. So the viral mRNA produced in the host cell's nucleus travels to a ribosome in the cytoplasm. The mRNA made by the host cell attaches to a ribosome. Molecules called tRNA bring the correct amino acids. So the tRNA, so I should have written this in capitals, tRNA, the tRNA, based on the triplet codons, so the ribosome reads the codons. Each tRNA has a triplet of bases, anticodon, that matches the codon on the mRNA. So tRNA has 3 bases already attached to it, and it's attached to an amino acid. You've got loads of different types of tRNA. So depending on these 3 bases. Now, when the tRNA comes and attaches itself here, then the correct amino acid is released. Then a different tRNA will attach itself to these 3, and a different amino acid will be released. The tRNA transfer RNA molecules bring the right amino acids to the ribosome based on the triplet of bases or codons. The ribosome then links these amino acids together, so it makes peptide links, peptide bonds, and the exact order is specified by the mRNA. So depending on the order of these bases, that will dictate which amino acids are made. So that then forms a polypeptide chain, in other words, a protein. Once the chain is complete, it folds into its unique shape to become a working protein, which in this case is a virus. So the virus has taken advantage of our own ribosome to make more and more of those viruses. So eventually the cell then gets full of viruses until the cell lyses, as in it breaks apart, and those viruses then go to other cells. So this is a really good question to understand how to apply this theory. Okay, describe how genetic variants in the non-coding DNA of a gene can affect phenotype by influencing the binding of RNA polymerase and altering the quantity of protein produced. So we have something called the non-coding part of the DNA. The non-coding part of the DNA doesn't code for any specific gene. What it actually codes for is how much of a protein should actually be made. So what is non-coding DNA? So think of DNA as a big instruction manual. Not all parts of it give direct instructions to make the proteins. So the main part of the DNA, so example here, you've got all these bases. You've got regions within here, like this part here, that has the information that actually acts a bit more like a control panel. And they decide when and how much protein is made. So it's very different. So not only does our DNA have how to make the protein, it also has the information on when the protein should be made and how much of that protein should be made. So you can appreciate if someone has a mutation in the non-coding part of the DNA, it can lead to issues in that suddenly too much of that protein is made and it causes an abscess, it causes a tumor. So there we've now got here our non-coding regions, and we've got in the center there and just at the end there we've got our coding region. So this part here makes the DNA, and this part here controls how much of that DNA is made. So sometimes the genetic panel gets a glitch called a genetic variant or mutation. It can actually have quite important effects even though it doesn't directly code for proteins, because if the proteins are overproduced or we end up with a protein shortage, both are problems. Overproduced can lead to a tumor. A protein shortage can cause other deficiencies and other issues. So most variants or mutations don't have any noticeable effect and are harmless. But some mutations may be harmful and cause diseases or disorders. Okay, we can move on from this bit. So this is an actual past paper question. Describe how a mutation in the non-coding region of the DNA can prevent a gene being transcribed. So the variant or mutation in the non-coding region, so the variant or mutation in the non-coding region of the DNA makes it harder for the RNA polymerase to bind. So if the variant or mutation in the non-coding region of the DNA make it harder for the RNA polymerase to bind, less protein might be made. So remember that polymerase is what helps make the mRNA, the single strand. Now if that mRNA can't be produced, as it can't be produced with the additional information linked to how much of a protein should be made, then if it doesn't bind properly, then less protein might be made. The RNA polymerase using the coding strand of DNA to make the RNA polymerase uses the coding strand of DNA to make complementary strand of mRNA, so it's important. But a mutation occurs in the non-coding DNA before a gene. That means less protein will be made. So describe how a mutation, so here's the actual answer. So the binding of RNA polymerase, so it prevents or reduces the binding of RNA polymerase, which means that mRNA cannot be produced or less is produced. So here, this part is not necessary, or this part, it's just that initial statement, that initial sentence or paragraph. So the initial sentence is all that's needed. 3.10b. What is coding DNA? So coding DNA is the part of your DNA that contains the instructions for making proteins. These sections of the DNA are called genes, and each gene provides a code for a specific protein. Now, what happens when coding DNA changes? It means the amino acid. So if the coding DNA changes because of mutation, that means the amino acid sequence changes, and that means the specificity goes. So mutations sometimes don't make any difference. Sometimes the same amino acid is still produced because just one amino acid might not make a difference. But sometimes a protein can be the shape can be completely changed, and at that point it won't work. And that mutation then disrupts the whole gene completely. So enzymes, so this part we're going to move on from. Because we know with enzymes, if you change the shape of the enzyme, the protein, then the substrate will no longer fit. So it takes away the specificity. Now, how common are these changes? So most mutations don't cause noticeable changes and are harmless. Some mutations can be harmful, leading to diseases, while others might be beneficial and give advantages such as better survival in certain environments. Each person is born with around 60 to 100 new mutations. So a coding mutation affects an essential gene, and that can cause a genetic disorder. So generally speaking, everyone has mutations. And mutations occur in everyone's cells, but it's not really an issue. But it is not really an issue. So excuse me just 1 second. Yeah, so those your general mutation is not an issue. The way the mutations occur, they don't lead to a new gene being produced. But every once in a while, it can lead to a new gene being produced, and at that point, it can lead to a genetic disorder. When the sequence of coding, so why alleles can produce proteins with different structures. So when the sequence of coding DNA changes, it's like altering the recipe. So you could make a small change and the recipe still, it doesn't make a difference. But if it's a big change, then that can lead to an altering of the protein shape. And sometimes that can lead to the production of a new allele, a new version of that gene. So this is an actual question. How can alleles produce proteins with different structures? So alleles have different DNA. So an allele is a different version of the same gene. So alleles have different DNA. mRNA strand has a different sequence within the allele. That means it codes for a different amino acid, and that means the protein folds with different structure. So what we're saying here is, why is it that you've got the same gene, they're both genes for eyes, but you've got one eye color gene here and you've got one eye color gene here? Why is it that different proteins are being made with different structures? So it's because the allele to make this one compared to this one, it's got a different set of DNA. That means when the protein is made, because this is all linked back to 3.8, when the protein is made, it ends up having a different mRNA strand sequence. So there'll be different triplets or codons. That means the different amino acids are coded for. So when the tRNA comes, different amino acids will be given. So the order of amino acids are different. And when the protein folds, it folds with a different structure. So this question, although it's linked to 3.10, goes right back to 3.8. So it's really important to understand 3.8 in a lot of detail. Okay, Gregor Mendel. Now, what we're going to cover with Gregor Mendel, and there's 3.17 and 3.18. I think, we should go through this. Now, Gregor Mendel, often referred to as the father of genetics. So the reason I'm with 3.17 and 3.18, I think those are better done in sequence. So what I'll do tomorrow is I'll cover 3.17 and 18 within sequence, but this is okay to be done separately because it'll still link to genetics. But it doesn't have to be done in the sequence. So tomorrow I'll be doing 3.17 and 3.18 as well, because I'll be covering topic 4 triple award content, but I'll add the 3.17 and 18 because I think it's better that we've covered the genetic stuff fully before we go through 3.17 and 18. So Gregor Mendel, often referred to as the father of genetics. Now, what did he do? He was a monk and a scientist. He did experiments on pea plants in the mid-19th century, and he discovered dominant and recessive alleles. So pea plants were his thing. So he grew lots and lots of pea plants, and he was actually a really good mathematician as well, which helped quite a bit. So Mendel observed the inherited characteristics, he observed that they're controlled by units. Now afterwards we've now called them genes, and they passed unchanged from parents to offspring. So he realized something was getting passed on. So when he took a tall pea plant and a slightly smaller one, or when he was looking at the seeds in different plants, he started to see that certain things were passed on, they were inherited. So things like flower color was inherited, the height was inherited, the seed shape and color was inherited. So he studied inheritance of different characteristics in pea plants, and he noticed when you had this color flower and this color flower, you always got this one. So he realized that this must be dominant. And when he then took two of those flowers, so he took two of these flowers, meaning he's got, so what I mean by this is we had a dominant plant that's purely pink, we had one that was white, but when these were bred together, all the offspring was pink. Now when he bred the offspring together, he found only 75% of it was pink, meaning that this pink one here in the middle must be carrying something inside it that allows it to have an offspring that's white. So why were Mendel's ideas not accepted until after his death? So he's understood that something's been passed on, but he faced many challenges. The biggest one being a lack of recognition. He was a monk. He wasn't seen as some big time scientist. So people didn't accept what he was saying. So Mendel's work wasn't accepted during his lifetime because DNA had not yet been discovered. So the physical mechanisms of inheritance was unknown. He'd understood that it was happening, but there was no explanation from a DNA perspective. His mathematical approach to biology was new and not widely understood. So people didn't understand the maths. And his findings were published in obscure journals, making them less accessible to the scientific community. In other words, he was unknown. Scientists didn't know about DNA, genes or chromosomes, so they couldn't understand his ideas. They couldn't relate to them, make sense of them. Now, Mendel established the principle of inheritance. So Mendel's principles to trace how diseases run in families and to predict inheritance patterns for conditions like cystic fibrosis and sickle cell disease, or even type 1 diabetes, came from his work. Now, 3.12 onwards is now what we'll be covering tomorrow. So this is all genetics until we get to 3.17 and 3.18. And it's better that we cover 3.17 and 18 as part of the flow. So that will be the end of the session today. So just want to say a really big thank you for everyone that stayed on. So 3.17 and 18, I'm just going to show you what they are. And you can go through this in your own time if you want to. The videos are available on AMALearning YouTube channel. But 3.17 and 18 are sex-linked genetic disorders and ABO blood groups. So I will go through this in more detail tomorrow. It's better done once we've finished the genetics section. Okay, thank you everyone. I hope you all have a lovely evening. Thank you for staying logged on, and I'll see you all tomorrow.